Good morning, sir. Nurachari, sir. Sir, your voice is not audible. Good morning, sir. Nurachari, sir. Sir, your voice is not audible.
Good morning, sir. Good morning, sir. Good morning. Okay, 5 minutes. నేను మ్యూట్ లోనే ఉంటాను. మీరు మాట్లాడండి, నేను వింటాను.
ఇది పెడతాను.
ఇప్పుడు శ్రీనివాస్ సత్యలక్ష్మి గారు ఇప్పుడు ఈ ఫస్ట్
నుంచి ఇదేంటి
ఫస్ట్ మీటింగ్ ఆఫ్ మీటింగ్ ఎంఓఎం 14/9/2020 అనే డాక్యుమెంట్.
हेलो हेलो अवनीत जी वस्तलेदे
सर आपकी आवाज आ रही है ऑडिबल
अरे नहीं आ रहा
आ रहा है सर. नमस्कार.
नमस्कार आई एम ऑडिबल
यस सर यू आर सर.
ओके ओके फाइन राइट अवनीत जी
Yes, sir. Yes, sir.
या
आपकी आवाज आ रही है.
आपसे आंसर नहीं आया तो मैं समझा ऑडिबल नहीं है.
No, it's perfectly all right, sir. Perfectly all right.
सर गुड मॉर्निंग. चारी सर गुड मॉर्निंग.
नमस्कार सर नमस्कार.
कैसे हैं सर? हाउ आर यू?
आपका चाय मिल गया?
मिल गया. आई जस्ट हैड इट एंड वेंट आउटसाइड.
वेरी गुड. जस्ट नाउ आई हैड माय टी. या ये अच्छा तरीका है.
हाँ जी हाँ. हमारे इधर बोलते हैं सर इन पंजाबी भी से हींग लगे ना फटकड़ी रंग चोखा. Without any expenditure you enjoy everything.
Good morning. Good morning, sir.
गुड मॉर्निंग गुड मॉर्निंग.
गुड मॉर्निंग
शर्मा जी नमस्कार.
Okay. Let's. Can we go with the session?
Yes.
Oh, you told 11.
यस यस.
You told 11.
So it has to be 11.
It has to be 11 because so till now, till then we can talk.
रामन जी.
Yes, sir.
So everything is okay?
Fine, fantastic, sir. 100%.
Oh, good.
So we the Punjabi University,
Yeah,
Patiala,
they do very well. They do very well. Yes, I know.
and the
नमस्कार
MS,
मैडम मनोज कौर मैडम नमस्कार.
yes sir.
under the presidency of Professor Rupam Kapoor, the MSI is also doing very well.
See, for the first time,
Yes, sir.
a woman mycologist has become president.
Yes
Yes, sir. Yes, sir. No, sorry, sir. That's not the first time, sir. Sorry.
Why? Who was
Because
there? Kalyan Sundaram was there?
Yes.
No,
Kalyan was. Yes.
Yeah, Kalyan Sundaram was there.
Yeah. She was the
Chandralata Raghukumar.
Chandralata Raghukumar. Yes.
Sir, yes, sir.
Yeah, you have made her president. Chandralata Raghukumar.
Sir, Rupam Kapoor is an excellent president and she gave an excellent remark.
Yeah.
Thank you sir. I need everybody's blessings and best wishes sir.
Yeah. I can see the of your guide, Professor Mukherjee, KG Mukherjee.
All these speeches were very good. All these speeches were very good.
Yeah.
Sir, good morning, sir.
Avinit Pal Singh or Kaur Madam, Rupam Kapoor and yourself.
Yeah.
Thank you, sir. Thank you.
Sir, only thing we missed this year is their food.
Hospitality.
Hospitality. Yeah, welcome.
Mativanan, how are you?
Yeah, fine, sir. I'm from the beginning. I'm here, sir.
Thank you.
I joined in the inaugural function.
Yeah.
It went on very well, thanks to all the organizer in the Patiala,
Yeah.
Dr. Kaur Madam and Avanit Pal Singh, Dr. Artri sir,
Khattar sir.
Khattar sir, and also the Dean who has delivered the presidential address, Dr. Rupam Kabur Madam for the presidential, the MSI presidential address, Professor Raman sir, and Manohara sir. So everyone really, it's nice to join in the webinar to see you all on the computer screen instead of physically, and we are meeting.
Madhivanan, on behalf
Yes, sir,
of
please,
MS, hearty congratulations to you, become the registrar of Madras University.
Thank you, sir. Thank you. Thank you, sir. Thank you.
All of you should know that Professor Mativanan has taken charge few months back as a registrar of a very prestigious university, the university which was established before the establishment of UGC. And that is really a pride moment for all of us that a mycologist becoming registrar of such a prestigious university. Congratulations.
Congratulations, sir. Congratulations.
Thank you, madam. Thank you. Thank you, madam. And also, I'm happy to inform you all, Professor Raman sir has nominated as a Professor Emeritus to the University of Madras very recently.
Congratulations,
very recently.
Raman and
Congratulations.
Madhivanan,
Thank you, sir.
for this accomplishment.
Thank you. Thank you. Thank you, sir. Thank you.
Avanit ji, so many good news are coming in Patiala conference.
Yes.
So many good news.
Yes, sir. Professor Manduchi also, she is working as the director, UGC HRDC, Punjabi University, Patiala.
Oh,
Congratulations.
madam.
And
Congratulations,
Professor
madam.
Khattar is the honorable professor in charge examinations.
Oh,
So
great, great. Joe.
And
additional
Professor
responsibility.
Khattar has been selected for Panitrai Lecture Award by the Indian Botanical Society.
Very good.
That's
Thank
great,
you.
sir.
Thank you.
Congratulations, ma'am.
Thank you, ma'am. Thank you.
Dr. Raman, can we, shall we start or wait for 5 more minutes?
You call the other co-chairman. Who is the co-chairman? Professor Atri is there.
Professor Atri is there, Hanji sir.
We are here.
First, the chairman and the co-chairman to stick to 30 minutes for each of the speakers, please.
Sorry, sir, I couldn't get you.
Timekeeping has to be there. That is what he's.
Yeah. I request the chairperson and co-chairperson.
Sir, may I briefly introduce the chairs first? Then you can start, sir.
No, I am not starting. I am just telling them. It is
Okay.
out of telling them to keep the time by 30 minutes.
Okay, sir. Done, sir. Done. So, respected mycologists, welcome back to the first session of this 3-day national conference, along with the 47th annual meeting of MSI. In this very first session, we have two very, very important lectures to be delivered. The first one is the presidential address, and second one is the Lifetime Achievement Award lecture. So for chairing this session, may I request Honorable Professor C. Manorachari and most esteemed Professor N.S. Atri to act as chair and co-chair for this session. Now,
welcome, sir. Welcome
to the session, sir. We have two eminent mycologists of the country with us. Professor C. Manorachari, born in Vevilala in Karimangar district of Andhra Pradesh on 1st of January 1945. He got his education. He was a student of University College of Science, Osmania University, Hyderabad. Did his PhD in 1974. Then he was awarded with DSc from University of Mysore, 99. He has a rich experience of postdoctoral research at UK, US, and Germany. He has been a frequent visitor to these countries. He served for a long time in the Department of Botany, Osmania University, Hyderabad. He is the NASI Senior Scientist and Platinum Jubilee Fellow for National Academy of Agricultural Sciences. Now he has been into various capacities. He has been head Department of Botany, Osmania University, Chairman Board of Studies in Botany, and Dean. He has been into UGC and Development Affairs, Emeritus Scientist of CSIR, and he has been the principal investigator of many research projects funded by various agencies. He has an endless list of the awards and honors. Now, to name a few, he has been awarded with Young Scientist Award way back in 1979, Best Teacher Award by Government of Andhra Pradesh, Birbal Sani Medal by Indian Botanical Society in 2000. He has been bestowed with the Lifetime Achievement Award by Botanical Society in 2016. He has been bestowed with every award which is there in the country. He has been a fellow of National Academy of Sciences, a fellow of Indian Phytopathological Society, Indian Botanical Society, Andhra Pradesh Academy of Sciences, and it is a matter of honor for MSI that this year he has been awarded with the Fellow of Mycological Society of India also. His research areas, they are primarily mycology, plant pathology, fungal biotechnology, and microbiology, and he has a huge list of publications to his credit. So with this brief biodata, we welcome Professor C. Manorachari to the very first session of this 3-day national conference. Welcome, sir. We have with us an eminent mycologist, a teacher from Department of Botany, Punjabi University, Patiala, who has been a source of inspiration to many like me and still inspiring all of us with his dedication, with his commitment to the field of mycology. Professor Narinder Singh Atri, he retired from Department of Botany, Punjabi University, Patiala this year. He finished his 5 years of re-employment with the Department of Botany, and presently he is settled at Solan in Himachal Pradesh. His area of specialization is mycology and plant pathology, with particular reference to systematics, evaluation, conservation, and cultivation of mushrooms. He has also contributed towards the studies of diseases of crop plants, avenue plants, and ornamentals. He is a highly proficient mycologist as far as mushroom collection, identification, and its cultivation is concerned. He has been instrumental in making the domestication of wild mushrooms like Lentinus squalosus, Lentinus conatus, Torulosus, Cladopus, Sajor kaju, and culturing of various other mushrooms like Calocybe indica, Pleurotus florida, Pleurotus albidus, and Pleurotus sapidus. To his credit, he has as many as 14 PhDs. 2 of the students, they are awaiting for their viva voce. 10 students have been awarded with MPhil, and he has more than 150 publications besides 4 books which are published. So he has delivered a number of invited talks. He has been the member of various professional bodies. While during his active service in the Punjabi University, Patiala, he has been the controller of examinations. He has been the additional dean research, besides being on many important committees. Large number of research projects funded by various agencies, they have been successfully completed. He has been the editor-in-chief of Kavaka till December 2020. And he was the person who was instrumental in making this journal online, and that has really increased the visibility of this Journal of the Mycological Society of India. He is also on the editorial board of Indian Journal of Forestry, Nelambo, and Mushroom Research, and Journal of Indian Society of Mycology and Plant Pathology. So With this brief biodata, we welcome Professor N.S. Attri. He has been kind enough to spare his valuable time and to be with us at Patiala, right way from Solon, for this 3-day national conference. We welcome you to this session, sir. So with this brief biodata of the chair and co-chair, I now hand over the proceedings to Professor N. Raman for the award session 1. Professor Raman, please.
Thank you, Dr. Avanith. On behalf of MSI, I request the chairperson, Professor Manogarachari, and the co-chairperson to Professor N.S. Attri to conduct the proceedings, please.
You have to introduce Dr. Rupam Kapoor, no?
Yes, sir. You can introduce her.
Okay.
You know her.
Fine.
That's why
Okay. Yes. Professor Rupam Kapoor, your presidential address is awaited.
Sir, would you kindly introduce Professor Rupam?
For Manogarachari, sir,
Yeah.
you introduce her, sir.
Yeah, Professor Rupam Kapoor, she is known for keeping alive the traditions of her teachers like late Professor K.G. Mukherjee. She did her PhD with late Professor K.G. Mukherjee and then started with the basic mycology, but now turned out to be a biotechnologist with reference to microbes and fungi. And she has done very good research work prominently on arbuscular mycorrhizal fungi with reference to their molecular taxonomy, ecology, sustainable agriculture, and other related aspects. She is known for her contribution in the form of more than 130 research papers, and all publications mostly they're cited heavily by the foreign scientists as well as Indian scientists. And she has got dedicated band of researchers, and she has been guiding not only on mycological issues but also the plant pathological issues faced by the country in order to achieve sustainable agriculture and food security. And as a bright and proud student of late Professor Mukherjee and Delhi University as a professor, she is sought after teacher by the students. I heard her in many platforms, and she has been very keen in encouraging youngsters. Even today also she has said that there must be a forum like MSI which can organize the systematics with reference to molecular taxonomy for the youngsters in the case of fungi and biodiversity. She has been responsible for training some of the students on modern technologies also at Delhi University. And with this kind of brief introduction, I wish her that she will enlighten us with her presidential address, which she is going to speak on arbuscular mycorrhizal fungi. I must say that in this modern era, she has become an authority on this group of organisms, that is mycorrhizal fungi. May I request Madam Professor Rupam Kapoor, please give your presidential address.
Thank you, sir. The advantage of this online meet is like all the stars of mycology have landed in this conference. And I congratulate the organizers for that. I may be missing out some, but I would like to acknowledge their presence and seek their blessings before I start with my presidential address. I feel like a student of nursery asked to recite a poem in front of my teachers here. I want to thank Professor Manorachari, who's more of a father figure to us, Professor Bhagyaraj, Professor Satyanarayan, he's also here. So namaskar, sir. Professor Lakhan Pal, who is also more like a teacher, but my Guru Bhai. Professor Rodrigues, Professor Yashpal Sharma, in whom I find a brother, and Professor Atri, Professor Bhatt, Professor Manruchi Kaur. The list goes on, and I think I will exhaust my half an hour here by acknowledging each one of you for your best wishes and blessings. Before I start my presidential address, in the capacity of president and also an ardent follower of mycology, I would like to thank Professor Atri for his long service as editor-in-chief. He delegated this responsibility with utmost sincerity, and we are totally grateful to you for that, sir. Thank you very much. With this, I would like to start my presentation. Is my screen visible?
Yes, ma'am. Yes.
Yes, ma'am. Perfectly all right, ma'am.
The crisis on food security has been looming largely because of mushrooming population. In last 5 decades, increasing food demand, increased requirement of food has compelled doubling of food grain production. And this was accomplished by employment of pesticides, chemicals, breeding of high-yielding crop cultivars, intensive irrigation. Nevertheless, exhaustive use of agriculture fertilizers have inadvertently resulted in depletion of the resources and environmental damage. The modern agriculture should accomplish 2 objectives concurrently. Firstly, to satiate the nutritional requirements of a swelling human population. And secondly, to reduce the damage that is caused to the environment. In this regard, a group of soil-dwelling fungi, arbuscular mycorrhizal fungi, can play a substantial role in fulfilling the United Nations 2030 Agenda for Sustainable Development, that is to end hunger, achieve food security, and improve nutrition and promote sustainable agriculture. Arbuscular mycorrhiza is a symbiotic relation between the fungi belonging to phylum Glomeromycota and roots of plants. As the name indicates, these are endomycorrhizal fungi that form intracellular structures called arbuscules in the cortical cells of the root. Some of the genera of arbuscular mycorrhizal fungi form balloon-shaped structures, vesicles that store lipids and glycogen. In the soil, the fungus subsists as spore, and the external mycelium that extends from the root act as root extension and increase the active surface of the root for the nutrient acquisition and water uptake. Arbuscular mycorrhizal fungi are the oldest symbionts of plants. They date back to about 400 million years ago, and they are present in all groups of plants, starting from bryophytes, pteridophytes, gymnosperms, and angiosperms. Most of the families of angiosperms, they participate in arbuscular mycorrhiza formation. However, some family, including to which the workhouse of genetics belongs, Arabidopsis belongs, do not participate in forming this symbiotic association. And we all know by now, this is because some of the components of signaling pathway, common SIM pathway that has been found, those genes are not expressed in them. As the arbuscule is formed in the cortical cells, it is delimited from the symplasm of the cortical cells by the invagination of the plasma membrane to form periarbuscular membrane. So the two membranes of the symbionts, the arbuscular membrane and the periarbuscular membrane, which is derived from the host cell, are separated by an apoplastic space through which the exchange of nutrient between the two partners takes place. The relationship between the two symbionts is depicted by in this slide by Bonfante and Yonner in 2010, which I liked among many descriptions. Here it shows that when in contact with the soil, the extra mycelium, they contain some high affinity phosphate transporters that take up phosphates from the soil, convert it into polyphosphate to prevent the buildup of phosphate concentration within the cells, and then translocate it, and then they are ultimately deported inside this periarbuscular space from which they are taken up by phosphate transporters that are present on the membranes of arbuscules. In contrast to plants that take up nitrogen mostly in the form of nitrates, mycorrhizal fungi are capable of taking up nitrogen in the form of ammonium, amino acid, urea. In return to all these benefits that fungus provides, it takes carbohydrates from the plant, and now it has been recently found that these fungi, they are not only dependent on carbohydrates, but they are also auxotrophs of fatty acids. So they also take fatty acids from the plant for their subsistence. This is a slide I always show those who have attended my presentations earlier. The concern of phosphorus in tropical soils. There are two issues with phosphorus. One, its movement in soil solution is very slow compared to the rate at which it is taken up by the roots. So after some time, a depletion zone develops around the root. And secondly, whatever phosphorus that is present in the soil, it gets converted into unavailable form by forming salts of calcium or salts of aluminum and iron, depending upon the pH of soil. In this regard, arbuscular mycorrhiza helps in uptake of phosphorus by improving the physical exploration of the soil, by entering the soil pore, which otherwise is not accessible to the roots, formation of polyphosphates, formation of certain organic acids such as oxalates that form complexes with calcium to form calcium oxalate, rendering phosphorus available to the plants. The hyphae of arbuscular mycorrhizal fungi, they occupy active site of decomposition, and they take phosphorus immediately after mineralization before it is converted into unavailable form. Mycorrhizae not only help in the uptake of phosphorus, they also help in the uptake of nitrogen and other ions such as copper and zinc, whose movement in the soil solution is slow. Consequently, there is increase in the yield. They maximize the nutrient uptake, they increase water uptake. They also help the plant to alleviate abiotic stress. The plants show enhanced disease resistance, and the work which we have focused on, they also influence the ability of plants to produce secondary metabolites. Lastly, Arbuscular mycorrhizal fungi help in maintaining soil fertility. In a small experiment carried out on tomato plants, we found that when these plants were colonized by arbuscular mycorrhiza and later challenged with Fusarium oxysporum, the plant showed an enhanced disease resistance to the pathogen. This was found to be due to dramatic increase in the levels of jasmonic acid, the activity of PAL, which is the first key enzyme for the synthesis of phenylammonia and is a key enzyme in the shikimic acid pathway, and there was enhanced production of phenol. Summarizing our observation, we found that not only it improved the disposition of plants to the pathogen, AMF showed synergism with other biocontrol agents such as trichoderma and Pseudomonas syringae. It imparts induced systemic resistance, so the plant, not only the roots, the entire plant is primed against subsequent attack by the pathogens. It protects the plant against pathogen by direct competition for site and nutrient, altered root morphology, and altered rhizosphere interaction. We also did certain experiments to study the mechanism by which AMF elevates abiotic stress, especially salt stress. And in an experiment where we coupled transmission electron microscopy with energy dispersive X-ray analysis, we found that the uptake of sodium and chloride ions and its translocation to the mesophyll cells were far less than the non-mycorrhizal cells, as it is evident in this slide. AMF helps in maintaining ionic homeostasis and preserving the ultrastructure of the cells. Due to osmotic stress, due to salt stress, osmotic stress as a result of salt presence, the plasmolysis of the cells results in recession of the plasma membrane from the cell wall, whereas in case of mycorrhizal cells of mycorrhizal plants, this is the term of mesophyll cells of mycorrhizal plants. The membrane remains closely oppressed to the cell wall. Also, there is pinching of certain vesicles that help in sequestration of sodium and chloride ions. The structure of chloroplast is preserved. The chloroplast in the mesophyll, in the mycorrhizal cells produce more amount of plastoglobules that are site for synthesis of alpha tocopherols, which are antioxidant molecules. When a plant is subjected to any kind of stress, abiotic stress or biotic stress, there is production of reactive oxygen species. And the production is far more than the ability of the plant to quench it by its antioxidant ability. This overwhelming production of ROS results in nucleic acid oxidation, membrane peroxidation, and protein denaturation. It has been found that in support with arbuscular mycorrhiza, there is high production of both antioxidant enzymes as well as non-enzymatic antioxidants. Mycorrhizal plants show high superoxide dismutase activity, which facilitate rapid dismutation of superoxide radicals to form hydrogen peroxide, which further prevents its buildup is stopped by activity of catalase, ascorbate peroxidase, and peroxidase. This diagram summarizes the various mechanisms by which arbuscular mycorrhiza alleviates salt stress. Due to paucity of time, I will not spend much time here. We also studied the effect of AM in alleviation of arsenic stress in wheat. That is the second most major point of concern in Indian agriculture. We found that not only there is decreased uptake of arsenic, its translocation to the above ground part and to the edible part of the plant is less. Arsenate in the aerobic soil, it is analogous to phosphate ions, hence it competes for its uptake. and it occupies and competes for phosphate ions inside the soil. Wherever the molecules phosphate is required, arsenic occupies that space. Mycorrhiza creates a favorable phosphorus-arsenic ratio for the plant. It prevents the antioxidant stress, like I said. The net photosynthetic rate in arbuscular mycorrhizal plants is far more than non-mycorrhizal plants. That results in higher biomass. Higher biomass of the plant eventually culminates into dilution of the heavy metal that is taken up by the plant. Not only this, in a recent work carried out by us, we found that arbuscular mycorrhiza also enhance the bioavailability of iron and zinc, which is a cause of malnutrition in the Indian population. Mycorrhizae helps in improving soil strength. It provides the scaffold to the soil particles to form macroaggregate. It releases certain glycoproteins, which are known as soil glues, known as glomalins, that help in binding the soil particles to form macroaggregates, hence improve the aeration and percolation of water to the roots. As 20% of the photosynthetes that are synthesized by the plant, fixed by the plant, 20% are used in subsistence of this symbiosis. So the roots that form mycorrhiza act as new sink and enrich the soil with organic carbon pool. Arbuscular mycorrhiza bind the heavy metals and the nutrient ions on its external hyphae and prevent their leaching to the groundwater, hence preventing the pollution of the groundwater. Coming on to the work to which we have dedicated almost 1.5 decades in role of arbuscular mycorrhiza, enhancing the production of terpenoids of high medicinal value. Terpenoids are they also are secondary metabolites besides primary metabolites in various forms, as they are the building blocks based on carbon-5 isoprenoid units. They form commercial use as flavor, fragrance in cosmetics and food. They have several medicinal properties and have immense ecological significance to the plant, as they help in plant to interact amongst themselves with microorganisms, with arthropods, and with herbivores. Terpenoids help in communication between the plants. The volatiles that are released as a result of herbivore attack results in production of volatiles that are communicated to the adjoining plant, which perceives these signals and gets primed against before it is challenged by the pathogen. Not only that, this herbivore-induced plant volatiles are also perceived by the carnivores of the herbivore, which is feeding on the plant, and hence the plant is able to save itself by releasing these terpenoids. In case of mycorrhizal plants, they form common mycorrhizal network. And this common mycorrhizal plants network, it helps in the communication between the population within a community, where they are able to communicate the danger signals and other signals, and also share resources through these conduits of common mycorrhizal network. When arbuscular mycorrhiza is formed, that results in change in the quantity and quality of terpenoids that are produced by the plant, and that results in inviting more amount of carnivores towards the plant that is attacked, and also the different spectra of terpenoids that are synthesized makes the plant more feeding disagreeable to the pathogens as well as to the herbivores. The major health challenge, of course, after COVID now, is that of prevalence of malaria. 93% of African population, followed by Southeast Asian population, are suffering from malaria. the Plasmodium parasite has developed resistance against it. So the scientists worldwide are working on bioprospecting various plants to find a compound that offers resistance against Plasmodium falciparum. In this regard, Professor Yu Tu of China got Nobel Prize in 2015 for a discovery of artemisinin, a sesquiterpene lactone, which shows anti-malarial properties. So we took this plant because of its high commercial value, and as the most accepted benefit of mycorrhizal fungi is enhanced phosphorus uptake, we carried out pot experiments where plants were given different concentrations of phosphorus so as to get non-mycorrhizal plants with phosphorus concentration equivalent to mycorrhizal plants. And we saw that in spite of similar levels of phosphorus concentration, the amount of artemisinin produced by mycorrhizal plants was significantly higher than non-mycorrhizal plants. Artemisinin is synthesized and stored in glandular trichomes that are present in above ground parts of the plant, and there is significant increase in the glandular trichome density results in significant increase in the artemisinin concentration. There is a strong correlation there. We found that mycorrhizal colonization results in about more than twofold increase in this glandular trichome density. We also found a tremendous increase in the concentration of jasmonic acid in mycorrhizal plants, and hence we went on to see that when plants are given different treatment of jasmonic acid, will it influence these characters that are desired by us? There was strong correlation relation between jasmonic acid concentration and glandular trichome density. And we found that the glandular trichome density increased as the endogenous levels of jasmonic acid increased in plants. And as to our expectations, the artemisinin concentration in non-mycorrhizal plants with equal amount of phosphorus and jasmonic acid concentration also had similar levels of artemisinin. To further confirm the role of jasmonic acid, we treated it with inhibitor of Ibuprofen, and we found that it not only decreased the concentration of jasmonic acid, it decreased the glandular trichome density and artemisinin concentration. Further, as artemisinin is a highly desired compound, its biosynthesis is known. The isopentenyl pyrophosphate that is required for the synthesis of artemisinin is either derived from mevalonic acid pathway that occurs in the cytoplasm or the methyl erythritol phosphate pathway that occurs in the plastid. And we found that though there was not much change in the transcript levels of the key enzymes of mevalonic acid pathway, there was 4-fold and 2.73-fold increase in the levels of enzymes of MEP pathway. Further, there was increase in the transcript levels of subsequent enzymes that are implicated in biosynthesis of artemisinin, which showed that arbuscular mycorrhiza enhances the production of artemisinin by not only enabling the differentiation of epidermal cells to form glandular trichome, but also enhancing the transcript levels of the enzymes that are required for the synthesis of artemisinin. The second challenge is diabetes, and the plant which we took also belongs to family Asteraceae. It houses steviol glycosides, which are 300 times sweeter than sugar, and they are used as alternate of sugar substitutes for patients suffering from diabetes, obesity, and dental maladies. A similar kind of experiments on stevia showed that mycorrhizal colonization also results in transcript levels of all the 11 enzymes that are implicated in synthesis of steviol glycoside and ribadioside. This figure summarizes the mechanism. The dotted line shows yet the mechanisms that have to be confirmed by more experiments. Any technology, if limited to laboratory, is not of much of human service. We struck collaboration so as to test the efficacy of our fungus in the fields. For this, we collaborated with IPCA Ratlam, an NGO in Madhya Pradesh, which is internationally renowned for their ability to standardize agronomy of Artemisia. And the farmers nationwide, they bring their harvest to them and they assess it and assign a value to the harvest. The experiment consisted of 100% recommended doses of fertilizers, arbuscular mycorrhizal fungi, and arbuscular mycorrhizal fungi with 50% recommended doses of fertilizer. And the field experiments were done. And the results that we obtained from them were really very encouraging. Though the performance of fungus was not as much as what we found in the pots, the experiments were done in sterile autoclave soil. Similar type of collaboration was struck by Institute of Himalayan Bioresource Technology, and I want to put in record the help of late Professor P.S. Ahuja, for extending his hands for us for carrying out these studies. And we found that the yield of stevia was also economically more favorable for its cultivators by use of arbuscular mycorrhizal fungi. The third plant which we are working on is Ocimum teniflorum. We all know this plant because it is even worshipped in our mythology. It is sacred tulsi. Its essential oil is an amalgam of flavonoids, terpenoids, and phenylpropanoids. And we found that colonization of arbuscular mycorrhizal fungi results in increase in the production of eugenol, which is a phenylpropanoid, and also terpenoids such as beta-alanine and beta-caryophyllene, which are valued for various medicinal properties. This Venn diagram shows that the two cultivars of Ocimum that we took, they were different in the spectra of the metabolites that were present in the essential oil, and the effect of arbuscular mycorrhizal fungi on them was also variable. What results into this variable response of arbuscular mycorrhiza? What increases and what at the cost of which metabolite is need of the hour? For this, we are carrying out non-targeted metabolome profiling to examine effect of on plant primary metabolites as well as secondary metabolites. Now, what are the challenges that we face in mycorrhizal technology? Lot of literature is available, and we know that arbuscular mycorrhiza, they provide solution to so many problems that farmers face. So from here, the research can move towards two directions: better understanding of the interaction between the arbuscular mycorrhizal fungi and the plants, so as to optimally use this interaction, and secondly, to use this technology in sustainable cultivation of crops. There are several roadblocks in use on mycorrhizal studies and their implication, because the next question that occurs in mind of any student or a person hearing is, if they are so good, they help in abiotic stress, they help in biotic stress, they help in alleviation of transplantation shock, then why do we not get easily AMF inoculum across the shelf, and why not much studies are being carried out? Because arbuscular mycorrhizal fungi, they are obligate symbionts. There are no confirmed sexual stage found in them. The spores are multinucleate containing thousands of nuclei, and they are heterokaryotic, and they lack uninucleate stage in their life cycle. So molecular studies or studies only carrying out on pure culture of these group of fungi still remains a challenge. Response to AM is contingent to both participating symbionts and the site. It depends upon the genotype of the plant that has been chosen and also the strain of arbuscular mycorrhizal fungi. So in this regard, various AMF ecotypes should be evaluated for identifying the most effective strain for a particular plant genotype based on the requirement. And to what extent responses of plants differ when interacting fungi are phylogenetically related. That is there any relationship on the effect of arbuscular mycorrhizal species and their phylogenetic relationships among themselves? And there is a need for considering and responsiveness while carrying out breeding efforts in the high-yielding cultivars. Now, responsiveness to AM should also be one of the traits when breeding cultivars with high yield. And lastly, site-specific case-by-case studies are required to assess the performance of mycorrhiza for the importance for the local value or for the nation's use. Understanding the role of arbuscular mycorrhizal fungi in the complexity of agrosystem is also required. For this, there is need to carry out experiments under natural conditions in a commercially viable way. Economical production of large amount of viable AMF inoculum to enable extensive open field usage, assess the inoculation with numerous AMF species or number of isolates to resemble agriculture ecosystem that harbor diverse AMF and individual plants, and selection of native AMF isolates can give more fruitful results. Once the biodiversity of AMF is reinstated, then we should move towards adaptation of agronomic practices that are friendly for the existence of arbuscular mycorrhizal fungi in the soil. And my last slide says that AMF crop interaction in the tropical soil is also needed. Perusal of literature shows that more of the studies have been carried out in temperate soils. Tropical soils are characterized by relatively low nutrient availability. Tropical soils have high phosphate fixing ability and hence show low phosphate bioavailability. Introduction of AMF in tropical agricultural soil would have immense role in increasing or optimal usage of arbuscular mycorrhizal fungi. With this, I would like to summarize the work that is being carried out in my lab as it is shown by various branches. We are working on various aspects of arbuscular mycorrhiza, basically finding the mechanism of alleviation of abiotic stress, especially salt stress and heavy metal stress, and production of secondary metabolite, primarily terpenoids. We are working on plant pathogen interaction, where we have worked on Botrytis cinerea. We have found out the diversity of both morphological and molecular diversity of Botrytis cinerea that is existent in India and Nepal. And we have gone on by using forward genetics to find certain novel pathogenicity factors that contribute to the pathogenicity of Botrytis cinerea. Now we are working on the interaction of safflower with two of its main pathogens, Fusarium oxysporum carthemi and Alternaria alternata. We are also working on effect of climate change, that is enhanced carbon dioxide on interaction of plants with their pathogens, so that the breeding efforts and developing plants towards this pathogen resistance towards this direction can also be made. And very recently, about few months back, we have stepped into the world of macrofungi, and we have started working on diversity of macrofungi in district of Kargil along with Professor Yashpal Sharma. With this, I thank you all for a very patient hearing, and I would like to acknowledge my collaborators, Jerry, Jamia Hamdard University, IPCA Ratlam, Institute of Himalayan Bioresource Technology, and all of you for your continuous support and blessings. Thank you.
Okay, thank you very much, Professor Rupam Kapoor. For the audience and as well as for the organizers, I must tell you, she did not leave anything on arbuscular mycorrhizal fungi, any aspect. She has dealt with all the aspects which are relevant to the society, to the agriculture, and to the scientific community, and for the knowledgeable upcoming youth also. Her lecture has been crystal clear and highly knowledgeable, and she spoke so much on not only the basics, but also the way in which these mycorrhiza will help the sustainable crop productivity and as well as the ecosystem balancing also, that is through biochemistry and as well as physiological aspects, nutrient uptake, crop productivity, crop enhancement, disease resistance, and so on and so forth. And all of you know that 400 million years ago, the mycorrhiza were found in the case of Rhynia chert. And then since then, these fungi are known to play an important role as a nature's gift for the benefit of the mankind. And they have made the plants, crops, medicinal plants to survive on this particular earth. That is the greatest contribution of this particular mycorrhizal fungi. Challenges are many, but she could come out. Professor Lakhan Pal sir is also here. He has worked on mostly on higher fungi and edible fungi, and he was also associated with late Professor K.G. Mukherjee. And he knows that there is one species of Amanita which spreads its mycelium up to 26 kilometers to tap the nutrients. That is the beauty of these mycorrhizal fungi. And there is no time to say much about the presidential address, but nevertheless, I must say that she, from the slide, last slide, I could see that she is also working for the empowerment of the women. There are only two male students who have been awarded PhD, and really it is very contributory type of science that she has made, and she wants to make the women to become the most important mycologists in this particular country, and also biotechnologists with reference to mycorrhiza. Thank you very much, madam, for having given us an opportunity to hear you, and also to MSI. On behalf of MSI, I thank you profusely and wish you bright future and long life and notable contribution in the field of science and technology with your commitment and concern and dedication. Thank you very much.
Thank you, sir.
Thank
you.
Thank you, madam, for your excellent presidential address. Thank you very much.
Thank you, sir.
The next will be the Lifetime Achievement Award by Professor. But I have made his contributions in the citation when I read that. However, I request Professor Manoharachari, the chairperson, to briefly introduce Professor Bhatt.
Professor Raman, you have already introduced him through that
Yes,
achievement award. But anyhow, I know Professor DJ Bhatt, and all of you know, including my co-chairperson, respected Professor Atri, that he is one of the leading mycologists in this particular country, and at a global level he has made an impact. He has dedicated the whole life for the study of fungi. and in particular to a group that is hypomycetes fungi. Whether the taxonomists agree or they don't agree, but you know the majority junk of fungi goes to hypomycetes fungi. And here is a person who has shown keen interest as a student of late Professor C.V. Subramanian, and later on he has become an independent researcher, and he has created such a kind of wonderful field of mycology. He is not only a table scientist but also a field scientist, a scientist on par with like Hebert, on par with Professor John Webster, and so on and so forth. He is of that kind of command on the subject. And here is a scientist who has spent a lot of his time, lifetime, I must say, only in the study of fungi and in teaching. And no other botheration was there for him. And he has not ventured to go for any other things. As earlier it has been said that 50% of credit should go to the family because he has been spared for the study of fungi. Therefore, I don't want to take much of the time. I have visited his lab. I have evaluated his student. I evaluated his DSc thesis also, a lifetime contribution and known for the country. And I really congratulate him on the occasion of this particular MSI Lifetime Achievement Award. And I must congratulate MSI that they have recognized a true mycologist of that particular science. And I wish him all the success, all the best in his future endeavors. On behalf of MSI, on behalf of my organizing committee, on behalf of my co-chair person, I request him to deliver his Lifetime Achievement Award lecture. Professor DJ Bhatt, sir, please.
Thank you, sir. Can you hear me?
Yeah, we can hear you, sir.
Okay. Thank you, Professor Manoharachari, sir, for the introduction. And I am more than delighted to be here to deliver my lecture. Friends, warmest greetings from Goa. I am profoundly thankful to Mycological Society of India for nominating me for this Lifetime Achievement Award for the year 2020. I'm equally delighted that the MSA is conducting its 47th annual meeting with a conference in Punjabi University, Patiala, a center of commendable researches on fungi. I know how much of work they have done to make this conference being scheduled on the right time and everything in an orderly way. Thank you, Professor Avneet and your team. I have planned to talk my own work today for this lecture and titled it as My Enduring Journey with Fungi. There are 33 slides and I'll try to complete this within the stipulated time. I hope this will be interesting to you also. I have given in the first few slides what I am, where I am, and what I did. My parents are from a small village in Karnataka state. Darbe Ishwar Bhatt and Devaki Amma. Both of them are actually illiterate, but they were great scholars. My father is a great scholar in Sanskrit, though they didn't take the formal education. I'm the fifth of the 11 children. And I did my primary schooling in our village. And many things we learned from our parents. I did my BSc, PhD, all these from the different universities later on. I moved out from my place. And my guide, late Professor C.V. Subramanian, a renowned mycologist and a wonderful human being. I learned not only mycology, everything that I know today from him. As Professor Manoharachari sir said, my DSc thesis was evaluated by him and I was awarded in 2017, though I submitted the thesis in 2009. It took nearly 8 years to have this recognized. My student days in Chennai was hugely exciting, intensely learning, and most memorable, because it had an excellent academic and then cultural environment, and I learned many things from there. I met many people, many intellectuals, and I got married in 1981. My wife is a postgraduate in history, but she never came in the way of my working on fungi. My 2 children, both are engineers. Daughter with her family settled down in Bangalore. My son is working in USA. I and my wife, we are now getting old and are presently residing in Goa. I have 5 major work centers in my fungal journey. These are not by design, it is by default. I mean, I went to University of Madras to have my mycological expertise. And I learned everything from there, how to collect fungi, isolation, culturing, photomicrography, literature survey, identification, everything from Professor CVS lab. Then I moved on to Asmara University in Eritrea, where that was my first work assignment, a decade-long stay. And I set up facilities and did first-hand work on fungi. And I published many papers. Later on, I'll come to this. Then I went to Mangalore University, then Goa University, and post-retirement, I worked for a few years in Mayfulviang University in Chingrai. My first work center was in Madras with Professor C.V. Subramanian in Professor C.V.'s lab. My PhD thesis was on developmental morphology of hypochrealian fungi, a group of ascomycetes. Those days, it was purely morphology-based taxonomy. And I also did postdoctoral work, the hyphomycete flora of South India, with the Professor C.V. Subramanian. And what gave me an advantage, an added advantage, was he was writing those days the book Hyphomycetes, Taxonomy in Biology, which was published in 1985, the academic press. And I did a lot of collection of literature, typing, and so on. So it was a great learning those days. And I listed out the papers which we published together in Madras. Then I moved on to Asmara University. It is merely in search of a job. I went, and it is in Horn of Africa. Presently, formerly it was in Ethiopia. Now it is a separate country, Eritrea. So I went there when it was part of Ethiopia. And the beauty of this place was before my work, the earlier work was primarily by Castellan and Ciferi in 1937. So after 37, the first fungal paper was 1983, a new species of Pleochaeta. I have given the picture of Pleochaeta here. And the lab had a wonderful microscope, but they had no slides, no cover glasses. So I requested Dr. Sutton, BC Sutton, from the CMI, the present CABI, and he sent me through British Council a pack of cover, slide glass, stains, everything. So that's how I started my work. And university supported my work in a big way. So not only I established a lab and did a great deal of collection from the Ethiopian highlands, lowland deserts, swamps, Red Sea coast, and published many papers. And most of these papers are published in Transactions of British Mycological Society journal. and many new fungi, and all are morphology based taxonomy those days. Then I came to Mangalore University because in 1988, Ethiopia, the Eritrea region had a civil war, and we thought we'll come back to India. And the first exercise, the first assignment I took in Mangalore University, and my teacher, Professor K.M. Kaveriappa, who was former Vice Chancellor of Mangalore University, prior to this, he was my teacher. He actually arranged this accommodation in the university. It was a small stay, just a 2 years stay, but it was truly invaluable. And when he gave me a lab space to work, Professor Bryce Kendrick gave me financial support. So that's how I started work. And this was the paper we wrote together in 1993, a big paper those days in mycotaxon, and 25 new conidial fungi from the forests of Western Ghats and Andaman Islands. So a great deal of work we did, many new fungi we described. Then it was just a 2 years stay, I said. Then I came to Goa University, joined first microbiology department for 4 years. Then I moved on to botany department. In fact, this was the major workstation I had in my mycorrhizal journey. Not only teaching we did a great deal of work with my students. I guided only 12 students, but my students excelled me in their understanding of the subject and communicate. Not only my morphology-based taxonomy, they are extremely good in molecular-based taxonomy of this fungi. They are all now well placed in different institutes, and they work as ambassadors of mycology. And we wrote about 120 papers those days, and described many new genera, and most of them were from Western Ghats. Then I wrote a book, worked on different sponsored projects, and we also described new methods for isolation of fungi, established a small nucleus for fungus culture collection, and we conducted 4 annual meetings of the Mycological Society in Goa University, along with the national or international conference. And in my mycological journey, I single out these 2 people, Dr. Sheshagiri Raghukumar and Lata Raghukumar from NIO, supported me in a big way besides my colleagues in the university. Now my work. It was primarily on diversity studies of microfungi. Major focus was on biodiversity study of ascomycetes and their asexual moths, hyphomycetes and coelomycetes. And we did field collection across the South Indian forests. And we went around the shola, semi-deciduous forests, grasslands, scrub jungles, streams, mountains, valleys, everywhere from Kanyakumari to Mahabaleshwar. And for nearly 4 decades, we did this search. Most of them are by foot and trekking, and we scanned primarily plant litter, freshwater foam, aerial plant parts, endophytes of leaves, twigs, herbivore dung, entomogenous fungi, soil. The primary aim was to look at the fungi, microfungi. And we brought most of them in culture, and our cultures, identified cultures, are deposited in, apart from Goa University culture collection, we deposit in Agarkar Research Institute and Institute of Microbial Technology in Chandigarh. And we collected from fungi from everywhere. Nothing we left in our survey. Western Ghats forests, coastal regions, freshwater fungi, hills of Arunachal Pradesh, and Nagaland in the eastern region. Then everywhere we looked for fungi. In search of fungi, I traveled all over the country, and I also traveled many parts of the world. Most important, I went to This was post-retirement to Chiang Rai in Thailand, Dali University in Kunming, and I also went to Hong Kong working on these fungi, then University of Waterloo, then in Kew, and so on, all these places. Okay. And I also interacted with numerous mycologists, most of the senior mycologists in the country, and many distinguished mycologists around the world. And all these discoveries, whatever we did, we compiled them in this book, Fascinating Microfungi of Western Ghats. And it has a foreword by Professor Malaria, and I'm very delighted that he is chairing this session today. Sir, thank you very much, sir. And you have been a great inspiration and a great supporter for me all along my mycological journey. It's a well-illustrated book on taxonomy of fungi. It includes methods of collection, isolation, culturing, and describing of fungi, taxonomic and ecological, biological details of about 130 fungi, fungi isolated from plant litter, endophytes, aerial leaves, herbivore dung are documented here, and also from, apart from Silent Valley, mostly from Goa region, Goa and Karnataka forests we have put them together. And this book is published by Broadway publisher. And what we did, I will quickly run through this. See, an ascomycete has an asexual and sexual phase. And many a times, they are not physically linked. So we had two articles in the International Code, Article 49, which allowed the dual taxonomy. And since Sacchardo's time, and in 59, Article 59 gave us the provision that if sexual phase is described, then sexual phase takes the priority or the asexual phase in naming. But from a taxonomic point of view, it was actually a major taxonomical aberration. This system continued for long until Melbourne conference. Now we have one fungus, one name because of with the molecular sequence data. And using morphological characters, we described these fungi. These are the asexual morph characters: mycelium, conidiomata, conidiophores, conidiogenous cells, conidia, and sexual morphs: ascomata, ascomatavale, centrum, or hamathecium, ascus, and ascospores. And in the asexually reproducing fungi, most important criteria are the morphology. Then conidiogenesis or conidium ontogeny. Now, any of the asexually reproducing fungi should come under the purview of these conidiogenesis: arthric, blastic, phyalidic, analytic, tratic, basoxic, or gangliar type. Using these criteria, approximately 21,500 species of Deuteromycotina were described. Many of them have already been transferred to Ascomycota or Basidiomycota, as and when their sexual phase is connected or redescribed with the information on molecular data. So it is hoped that when molecular data is available for all, we will have a natural system of classification. And many new fungi we described. I have listed here 20 new genera which are described from the different regions. Then we also found very interesting conidiogenesis pattern. If you put the newer taxonomic hierarchy here, hypocreales and nectriaceae have only phyalidic conidial fungi. Similarly, Euroteales has only phyalidic. In other words, this conidiogenesis character also is most stable apart from the molecular taxonomy. Now, if you look at this further, phyalidic conidiogenesis, hyaline with wet or slime conidia, all fall together, even in the molecular sequence data. Similarly, Euroteales have all were hyaline, dry, and catenate conidia. They have similar pattern of grouping. I will run through a few interesting and notable fungi from our work. This is the first fungus that I described. Bahusutra Bija Dvaya. In 1977, we published this Canadian Journal of Botany. This is a mononematous fungus with a beautiful conidia having thread-like appendages. Subsequently, this fungus was sequenced and it was found to be in Chaetospirales. Then we described in 1978 a fungus called Pitambara Sundara, an Ascomycetes fungus, Hypocreolin Nectriaceae family. And it is a holomorphic genus collected from my native place and golden colored ascocarp. So we called it as Pitambara. Sanskrit name we gave. And its asexual morph separately we described and called it as Putagravum. Although it was not fair enough, we described those days because such a provision was there. But later on, its sequence data was found out, and Pitambara is now in Stachybotryaceae and Solidariomycetes. Then, those days, we also studied the developmental morphology of a fungus, Fusarium nivale, Monographilla nivalis, with Ascomyces stage, Monographilla nivalis. Now, why I put this slide is, using light microscope, we found out that the conidiogenesis is phylotic, but the conidiogenous locus was not uniform in different conidiogenous cells. We didn't know what's happening. And subsequently, same time, maybe a couple of years later, Walter Gams and Müller found out that it is an analytic conidiogenous cell. So when we studied in 78, we said it is not of the hypocreolin member because it has not a phylotic fungus. We didn't know what it was those days. In other words, light microscope, a careful examination tells you what a fungus is. And subsequently, in Jeevan et al., they did the molecular sequence studies and found out that it is far away from the Solidariomycetes, it is in the xylariales. And we did several such studies. In 2007, we found out the asexual phase. In 2016, sexual phase. So physical connection was made of this Vamshapriya bambusicola. Then Neodytonia subglobosa, described in 1969. We found out the sexual phase in 2016 and made a physical connection. And look at this fungus. It was Bahusandhika indica, described in 1956 by my professor. And we brought this fungus in 2014 into culture and re-described its morphological characters and studied the phylogeny. So what really matters is, as and when we have the newer technology, we adopt them and get the confirmation of this taxonomy. Phylogenetic analysis confirms its taxonomic position. It is Phleosporales member. So such studies can be done. Natarajania indica, named after my another respected teacher, Professor Natarajan. Then Rattania setulifera, described a fungus which we collected from Rattan's cane from the Western Ghats region. And very recently, in December 2020, we were asked to write a letter, tribute letter to late Stan Hughes, by Professor Hawksworth, and this fungus Fusicola hughesii, because Stan Hughes was also a great supporter of my work. I approached him in numerous times for correction of my descriptions, literature, and he would readily oblige. And he expired only last year at the age of 101, and one of the towering personalities in mycology. And I described this along with my colleagues Professor Dr. Singh and his students from Agarkar Research Institute. It's a beautiful fungus, cinnamatous fungus in culture. They isolated it from air, and it's described in Journal of Fungal Research. And we dedicated it to Stanix Nectriaceae, Sardariomycetes fungus. And post-retirement, I spent some time in Mayful Wang University in Chiangrai with Dr. Kevin Hyde and his students. And he has students from all over Asia. And I listed here papers which we published in 2020. Just 1 year publications. You can see what a aggressive lab it is in working in. They not only work with the microscope, but they also do molecular sequencing of all the fungi that they isolate. And I literally look at every fungus that they describe because they keep sending me the picture, descriptions. That's how I participate in their descriptions. And they contribute into several journals and large number of publications they undertake. And friends, ideally a teacher never retires. And presently, besides mycology, I also assist biology students in a high school, part of the Vishnugupta Vishwavidyapeetham, a newly established Gurukulam type education institute in Lachoke, Pokarna, in Karnataka. It's a coastal town in Karnataka state. This institute was started only last year, and I assist them in biology teaching those students. And I'm associated with the Mycorrhizal Society of India from 1973, the year of inception. And because I joined after the International Congress, so I really didn't see its establishment, but from that year, 1973, and worked in different capacities in this institute as a treasurer for a while, then secretary, and also editor of Kavaka for a couple of years. And we conducted 4 annual meetings here in Goa University. 47th year, the members, I must really congratulate Professor Raman sir, because he is responsible for this great contribution of increasing his membership, and his contributions to the society are immense. Many people have worked on this. I sincerely thank the society for recognizing me for its highest honor. I conclude my talk with this slide. I'm truly fascinated by fungi, and quote from my professor's book, Hyphomycetes, in 1970. The beauty of fungal world is so large, so interesting, and so amusing. And I also had an opportunity to write on my professor, late Professor Mohan Ram asked me to write this article, Living Legends in Indian Science, in Current Science, in 106 volume. If you see, you will see an article on Professor C.V. Subramanian. And tracing small steps in this journey, I conclude what Bhagavan Sri Krishna said in Bhagavad Gita, Shraddhavan labate jnanam, meaning sincere and hard work begets knowledge. End of my talk, I thank everyone who made me fit to receive this award, especially my students who contributed, who supported me, and all the senior mycologists who blessed me. I must thank Professor Manohar Acharya and Professor Lakan Palji, because I came to know that these two people saw my thesis, DSc thesis, for evaluation. And I want to thank my parents, teachers, my wife and children, family members, and my students, fellow mycologists and friends, and not the least, the incredible fungal world. Thank you very much.
Thank you, sir.
Yeah.
Thank you. Yes, sir.
Before I conclude, may I request Professor Atri to add his comments, if any? Professor Atri? Is he there, Professor Atri?
Yes, sir.
Atri is there, Professor Atri?
Is he there?
Okay, sir. Thank you very much, Professor Bhatt, sir, for an excellent lifetime achievement award. And I thank Professor Manogarachari and Professor Atri for their very good cooperation and
Professor Atri is there. Professor Atri is there.
Is there?
Yes,
yeah,
sir.
Professor Atri.
Let Professor Atri comment, then I will just thank all of you for a minute.
Sir, unmute kar lo ji.
Hello, sir.
Okay.
I have spoken already.
Yeah.
I'm sorry. I'm sorry. It happens many a times. So
It is your,
yes,
it is
sir.
your home institution, so you have to spend time. Huh? Yeah.
I forgot unmuting. So,
Dr. Bhatt, Dr. Raman, can I say something?
Namaste, sir. Namaste.
Namaste. Dr. Bhatt, before we evaluated your thesis, you were already inside as Doctor of Science. So it was a mere formality. Congratulations, heartiest congratulations.
Thank you. I'm very grateful to you in this small nature. Yeah.
Sir, Professor Bhatt, sir, your thesis was lying for the university for many years. I am happy that I gave the panel and it was the Professor Manogarachari and Professor Lakhanpal. They evaluated the thesis and it was the university. It was delayed. It is not the examiners.
so may I speak, sir?
Yes, sir. Yes, sir.
Yes, sir. Yes, sir. At the outset, I would like to thank MSI specifically for providing me this opportunity to co-chair this session along with esteemed Professor C. Manoharachari, who himself have been inspiring person and he has been aspiring. He is a teacher for all of us. And to begin with, I will thank Professor Rupam Kapoor for coming to our rescue, to the rescue of Mycological Society, in accepting the request of the executive to be president for this particular session, because of the reasons well known to each one of you. And she spoke upon how AM fungi can be profitably employed for making the agriculture sustainable. And she amply exemplified how we can take our programs from lab to land and make them successful for making the whole exercise sustainable and fruitful. And I'm thankful to Madam Kapoor and for the nice words she has spoken about me regarding Kawaka. I did my duty, which was bestowed upon me by the honorable members. I didn't do anything extraordinary. It was just my duty, which I did to the best of my capability. It is accepted and I feel honored by the recognition the society has given me. As far as Professor DJ Bhatt is concerned, he has been an inspiring teacher and I am inspired by his inspiring journey of mycology which he presented today, and I wish that he keep on inspiring the younger generation and future generations for the time to come. He is a living legend amongst us all, and I am really thankful to you, sir, grateful, and my hearty congratulations to you and to all your family members, Mrs. Bhatt, and everybody, to all your students who has contributed to your growth and really highly inspiring journey, sir. Thank you very much. Grateful. It is an honor for Punjabi University Patiala as well.
It's my pleasure, sir. Thank you.
Thank you very much, Professor Bhatt, sir. Now, did you get married with fungi or with Vasanta madam? Really, it is very interesting and excellent lecture. Your voyage has been such a long. Yes,
may I take this opportunity to project the certificate and the award, which
Yeah.
has been sent in the form of soft copy by Dr. Raman.
Yeah,
With-
the MSI is so efficient and it is giving the certificate soft copy immediately.
So
Thank you.
thank you, Dr. Avanith Khattar, Professor Manruchi Kaur, Professor Raman, and every one of you, Professor Yashpal Sharma, Professor Mathivanan, and every one of you, Professor Atri, and everybody for having given me an opportunity to be with you for this particular session. And I will see you tomorrow. To the Professor Raman and Professor Avanith, I'm sending one message to you. Please discuss in general body meeting today. Thank you very much. Thank you.
Thank you, sir, for sharing the session for joining the session.
Thank you, sir. Thank you very much. Thank you, everyone.
This is the certificate that has been sent by Professor Raman in connection with Lifetime Achievement Award lecture. So once again, heartiest congratulations to Professor DJ Bhatt.
That award is there. The award copy is also there.
Sir, I'll show that also. So this is the certificate. Once again, we congratulate Professor Bhatt for this. And then we have the award also. And here is the award. This is the virtual award, and the copies will be sent as communicated by the Honorable Secretary to Professor Bhatt. So once again, heartiest congratulations to Professor DJ Bhatt for the Lifetime Achievement Award.
I'm truly humbled by your presence. Thank you.
Thank you, sir. So, thank you, Dr. Avanith, for moderating the session. And I think Dr. Nitika Shirendi, she will be.
Sir, no, it's Dr. Arneet Garewal. She will be there. I may request Dr. Arneet to kindly moderate with session 2. Dr. Arneet, over to you, please.
Okay, you are also you are coordinating this session also. On behalf of MSI,
Excuse me, sir. It will be moderated by Dr. Arneet Garewal. She will be taking over.
Okay.
Dr. Arneet, please proceed.
Thank you so much, sir. I'm sure there's some kind of a confusion, sir.
Yeah, madam, now I can see you.
We both share the initials. I'm Arneet Garewal, sir, and I'm basically a cytogenicist. But
nice, madam.
I will be conducting this session. I will be moderating this session, sir. And I thank the organizers for giving me the opportunity for moderating the second session of the day. And it is my honor to introduce the chairperson, Dr. DJ Bhagiraj, and co-chairperson, Dr. MS Reddy. I will be reading out their biodata in a very short form. Dr. Bhagiraj is an elected fellow who was in 2008, and he has completed his education from various institutes of Karnataka, and he has excelled in the fields of agricultural microbiology and with special interests in mycorrhizal symbiosis, biofertilizers, and biological control. He's held various administrative positions and presently is the chairperson of CNBRCD Bangalore since 2001. He has, to his credit, large number of awards and fellowships, and some of the eminent ones being the Agni Hotrudu Memorial Lecture Award of MSI in 2011. For Dr. MS Reddy, I would use a few minutes of your time again. He specializes in plant microbial interactions and molecular biology. His research interests vary from biotechnology to the use of fungi in bioremediation and sustainable agriculture. He has had professional experiences from India as well as abroad, and is presently a professor in the Thapar Institute of Engineering and Technology, Patiala. He's held various administrative positions including Dean Associate Resource Mobilization, Head of the Department of Biotechnology, and as Coordinator of Center for Relevance and Excellence. He has been bestowed upon with large number of awards of national as well as international repute. He's held many memberships, and his areas of teaching and interest again vary from biotechnology, molecular biology, bioinformatics, biostatistics, and microbiology, and latest being metatranscriptomics, which is an emerging area. With that, I would like to hand over the mic to Professor Bhagiraj to kindly conduct, and Professor M.S. Reddy to kindly conduct the proceedings. Over to you, sir.
Okay. Thank you very much, Dr. Avneet Grawal. And first of all, I would like to thank the organizers for having given me this opportunity to chair this, chair session 2, and along with the co-chairperson, Dr. And I hope I am audible.
Yes, sir. Yes, sir. Audible.
Sir, audible.
Yeah. Okay. Yeah. I know the time is 12:30. We are half an hour behind the schedule.
So I think the chairperson and the speakers will cooperate with us.
So I think we should. I think this we have 3 speakers. All of them are going to make beautiful presentations because I know all of them, and all of them are the award lectures. So I only request is kindly keep up the time. Right?
Good afternoon, Professor Bhagiraj. Good afternoon, Professor Bhagiraj.
Yeah. Good afternoon, Dr. Professor Manoharachari. Good afternoon. Yeah.
Sir, before you introduce the speakers, I will introduce the memorial award, sir.
Okay, sure.
The award is Professor C.V. Subramanian Memorial Award for Women Scientists. Professor C.V. Subramanian, former Director, Center for Advanced Studies in Botany, University of Madras, Chennai, India, founder of Mycological Society of India and former President of Indian Mycological Association, passed away in Bangkok, Thailand at the age of 92. He is survived by his wife and 2 sons, Professor C.V. Subramanian, CVS as he was affectionately called, earned his PhD in 1948 and DSc in 1957 from the University of Madras. He did his postdoctoral research at the Botany School, Cambridge, UK, and the Commonwealth Mycological Institute, Kew, UK in 1950. He was appointed to the first ever chair in plant pathology in the country at the Indian Agricultural Research Institute, New Delhi in 1957. He was later invited to organize the botany department of the University of Rajasthan at Jodhpur and Jaipur before moving to the University of Madras, where he joined the Center for Advanced Studies in Botany as professor and served as its director from 1973 until his retirement in 1985. After retiring, he was invited as a visiting professor at the National University of Singapore, the University of Western Australia, and the National University of Taiwan. Several awards and recognition came to his way. In 1965, he was awarded the Shanti Swarup Bhatnagar Award, the highest award for science in India, the Rafi Ahmad Kidwai Award of ICR, the Janaki Ammal Award, etc. He was also the recipient of the prestigious Jawaharlal Nehru Fellowship, 1976 to 1978. He served as member of the University Grants Commission, 1977 to 1982. He has been a member of several national and international scientific academies and organizations and committees constituted by the UGC, DST, ICR, and CSIR. At the very young age of 32, he was chosen by Sir C.V. Raman as a member of Indian Academy of Sciences. Later, Professor C.V. has served 9 years as the Vice President of the Academy. He was also a member of the Indian National Science Academy, honorary member of the American Mycological Society and the British Mycological Society, corresponding member of the Belgian Royal Academy of Foreign Sciences, the member of the International Union of Biological Sciences. He started the use of Sanskrit names for the new genera that he discovered. Several of the new fungi that he described now carry Sanskrit names. In recognition of his contributions in mycology, he was elected as president of the International Mycological Association in 1977 at Tampa, Florida, USA. He was president of the International Mycological Congress, Tokyo, Japan, 1983. He founded the Mycological Society of India in 1973 and launched the journal Kavaka, of which he was the editor from its inception till 1998. Kavaka is the Sanskrit word of fungi. His monograph Hyphomycetes, published in 1971, and the book Hyphomycetes, Taxonomy and Biology, published by Academic Press, London, 1983, are widely referred by the researchers all over the world today. He was a great believer and follower of Swami Vivekananda and Ramakrishna Paramhamsa. He was closely connected with the Ramakrishna mission and delivered lectures on Vedanta in centers in India and abroad. His professional interests were literature, philosophy, and music. He was really passionate about music and was a good singer. And Mycological Society of India instituted the Professor C.V. Subramaniam Memorial Award for Women Scientists in 2018. In 2020, Professor CVS family has contributed 50,000 for the Memorial Award, and the request was sent to all students for the counter amount. Professor Savita contributed 20,000. We thank the family as well as Dr. Savita. And Dr. Savita's student Anil Kumar contributed 5,000, and the MSA contributed 25,000. Now we deposited 1 lakh fixed deposit towards the Memorial Award. One request was put by the family of CVS that the award should be for both the genders. So from 2021, the award will be known as Professor C.V. Subramanian Memorial Award. With this introduction, I request the chairperson to introduce the speaker.
Okay. Thank you, Mr. Raman. So it's my
this award goes this time to Professor Sarita Nasrat from Goa University.
Thank you. And it's my pleasure to introduce Professor Sarita Nasrat from Goa University. And Professor Sarita completed her PhD in microbiology and later did her postdoc work in Italy. She started her work with fungi in early 1980s. Her first work was on metabolism of phenolic compound ferulic acid produced by Fusarium amidst a group working with only bacteria. So she could start the work on fungi with a group working with bacteria. Her areas of interest are marine biology, lignocellulose degradation, bioremediation, microbial enzymes, diversity of fungi in mangrove sediments, and halophilic fungi. She served as a member or a chairperson of several prestigious committees, and she has trained several students in mycology and has several publications to her credit. She was the recipient of Shaw Memorial Lecture Award of MSI in 2015. Today, she is going to present Professor C.V. Subramaniam Memorial Award Lecture on allophilic fungi from different eco-niches and their osmo adaptation. Over to Professor Sarita Nasrat.
Thank you, Professor Bhagiraj. Can I be heard?
Yeah.
Yes, okay. I'm honored also to have Professor Bhagiraj to be chairing this session. Honored and humbled. I've known Dr. Bhagiraj for several years. And also Dr. Sudhakar Reddy. without further ado, because I know we are already late, I will start. And I am deeply grateful to the Mycorrhizal Society of India for bestowing upon me the honor to deliver this Memorial Award lecture in esteem of the distinguished mycologist of India, Professor C.V. Subramanian. I'll go straight to my talk without further ado. Since I'm speaking on extremophilic microbiology, just for those, I suppose there are students listening in, that organisms that thrive in extreme conditions are so called. And I'm speaking specifically on halophiles, which are salt organisms.
Excuse me, ma'am.
Share your slides.
Yes.
Ma'am, your screen is not visible.
Visible?
My screen is not visible.
Yeah,
ma'am.
Video.
What do I do? Just a moment, okay?
You can stop it once, then again start sharing.
Sorry, it was there and then it has gone.
No, ma'am,
Okay.
it was not visible.
Now can you see it?
No, not from the beginning. We couldn't see it.
Yeah.
Now can you see it?
No.
No, madam.
Oh gosh. Okay. Just I'm sorry. One moment.
Stop sharing. How this our screen.
While I figure it out, can I proceed with the talk?
Yes, madam.
Yeah.
Hello. Yeah. While I get it sorted out, can I proceed with the talk?
Yeah. Please go ahead. Yeah.
Just to save the time.
Yeah, sure.
Okay. So I'm talking on halophiles, and they have been categorized according to the requirement for NaCl for their optimal growth, either slight halophiles, moderate, or extreme, depending on the salt concentration required for their optimal growth. Now, the mechanism for survival of halophiles in hypersaline environments, the organisms develop a process. As an archaea, it is a salt and cytoplasm mechanism, and they're termed as osmoregulators. Or in bacteria and fungi, you have an organic osmolyte mechanism, and they are termed as osmoconformers. I will come to the organic osmolyte mechanism since this is in fungi. And there are various compounds that are produced or either synthesized or taken up from the surrounding. These are sugars and derivatives like sucrose, trehalose, etc. You have polyols. Polyols so termed because of the several OH groups that they have. Glycerol, sorbitol, mannitol, inositol, arabitol. You have amino acids, quaternary amines. Now I'll skip the next slide, which showed those figures, but I think everyone knows sugars and polyols, which I mentioned, the ribitol, erythritol, etc.
Ma'am, I'm sorry to interrupt.
Yes.
But I think the screen is still not visible, or it is with our side.
Yes, I just asked somebody to help me out with this. I've actually come to my department because this is a new thing for me.
Okay, ma'am. Okay.
But so I'm sorry, I've sent for somebody again because I'm sorry they've not been of much help.
No problem, ma'am. Please
Okay.
continue, ma'am.
So just to save the time, I'm carrying on. Okay? Yes. So I am aware that, yes, can you please get the screen to be seen by them?
The presentation is not visible.
No, please. I'm on the mic. Now I'm coming to the various works that we have done. Please, can you do that? Please hurry up, okay? They cannot see this screen, please.
It is actually showing here that it is sharing.
Shall I exit and restart again?
I think that would be a better option.
Hello, ma'am.
Hello, can you get my screen to be seen, please? The video has gone off. Okay, I think the video had gone off apparently. I don't know how, but so I will try again. is this now? Something else has come up. What is this now? My whole thing has gone. Nuisance really. No, my whole presentation has gone.
Open your pen drive.
Dr. Avneet.
Yes, sir.
Hello, Dr. Avneet.
Yes, sir. Yes, sir.
Can we go to the next presentation by the time Dr. Sarita gets the whole thing fixed?
That will be better, sir. That will be better.
Yeah, I think that's, yeah. So I think probably.
Yes, sir. I will
Yes,
read
yes,
the.
please go ahead. Thank you.
Okay.
Madam, thank you, madam. Thank you for your coordination. Amanit, can you, Arnit, can you
Yes,
focus
sir.
me?
Yes, sir.
Can you focus me? Yeah. Can you focus me? I am audible.
Yes,
Okay.
sir. You're audible. Sir.
Can you see my screen now?
No.
Ma'am,
Still not.
I would like to request you to first leave the meeting and then join back again. Maybe that could solve the problem with the camera on.
Madam, let you be ready. First, we will finish the Agnihotra Memorial Award. We will come back, madam. Certainly, we'll come back to you.
Can you see it now, just so that I know?
Yes, yes.
Yes, ma'am.
Yes, ma'am.
Now we can see, Dr. Sarita.
Yes.
Now we can see.
You can see only.
Yeah. Go.
Yes, ma'am.
Go ahead if we can.
Yes. We can shift to the full screen mode, ma'am.
Yes, okay. I'll do that. I'll just get to where I left off.
Yeah.
Just wait here, no? Yeah, can you see it?
Yes, madam. We can see.
Please go ahead.
Okay. So I had come to where the various osmolytes are there. Okay.
Excuse me, ma'am.
And I'm going on further. Now, what we studied, you can see it?
Yeah.
Ma'am, the slides are visible.
Sarita, make it full screen.
Slide show. Can we go towards the
Yes,
slide
okay.
show? Yeah, slide presentation, please. Yeah.
Yes. Okay. Is it okay now?
Yeah, if we can make it full screen, it will be nice if we can make it.
It is full screen. It is full screen.
The dumbbell shape.
Ma'am, if you go towards the bottom.
Yes.
If you move your cursor towards the bottom, the fourth icon.
It's at the bottom.
The fourth icon. The fourth icon on the right side is actually the slide presenter.
I can't. There are no icons appearing on my screen right now. It's just full screen.
Let her present. It will.
Yes, I think just go ahead because it will take up time. If you can see something.
Yeah, you can go ahead.
Okay, ma'am. Okay.
Okay. Yes, thank you. So what we studied was athalassohaline environment of Dead Sea. Now, athalassohaline is that the composition of the constituent salts is not like that of seawater. Okay, so and thalassohaline is that as of seawater, not to do with the salinity, the degree of salinity. So the Dead Sea actually has a very high salinity, but athalassohaline. And you have the solar saltons, which are thalassohaline. You have polyhaline environments that we studied, the estuary and mangroves, all of seawater consistency. Now, this is just showing where the sites were. As you can see, the Dead Sea, I've shown it on the world map. And this is Goa. Many of you have been there and you know the Mandovi River, okay? So that's along the Mandovi River. The mangroves and saltons from this area. I will skip these slides. It's showing the salinity and the pH, which was, for the Dead Sea, it was slightly on the acidic side. And for the thalassohaline econiches, it was slightly alkaline.
Your slides are not moving.
Yes.
It's stuck.
Oh, God. The slides are not moving. What I will do is I will try to... now just tell me if the slide is on the thing.
No. We see the sugar
It
slide.
is still stuck?
Sucrose, trehalose, it's stuck there.
Oh God. Oh God. I have no idea why this problem, because mine is moving here.
Oh, it's moving
Oh
there.
God. Okay. But you know what, I can only if I'm on will I know that some problem is there, because I would have said, please go on to the next one.
Dr. Avneet, can she send the slides to you and you in turn can project? Will that be okay?
Absolutely,
Yes,
sir.
ma'am.
That can be done.
Yeah, Dr.
Okay, fine. Yeah. So the next one can do that. Meantime, to whom can I send it?
Ma'am, you can send it to the email of the conference.
Okay, okay, sure. Thank you. Yeah, in the meantime, you can go to the next one. Thank you so much.
Dr. Arneet, shall we go to the next presentation in the meantime?
Yes, sir. Yes, sir. I think that would be a good
Yeah.
idea.
Yes, please. Thank you.
Dr. Navneen Kango can go with the presentation. By the time this can be sorted out.
Yes, sir. Yes.
I have a problem that the video keeps going off all the time. That's why you can't see it. Okay, I'll just try. Anyway, carry on.
It's slow at your end, ma'am.
Yes, yes, it is. Thank you. What do I do? I have to get to the...
Dr. Naveen Kango, yeah, you are there. Okay, nice. Namaskar.
Yeah, we can start with sir.
Yeah, next.
Yeah, next.
Yes.
Okay. Now we are moving to the next memorial award lecture. Dr. V. Agnihotridu Memorial Lecture. Dr. V. Agnihotridu, a native of West Godavari district of Andhra Pradesh. Dr. Agnihotridu grew up in Madras, where his father was in service, and he had the entire education in Madras right from schooling. He passed his BSc in botany from Presidency College in 1950 with a first class and first rank in Madras University. He then went on to take his PhD in 1954 from Madras University Botany Department under the guidance of Professor T.S. Sadashivan, working on the microflora of soil and the rhizosphere. This was followed by a brief postdoctoral stint in the same department, during which Dr. Agnihotridu became interested in mycorrhizae and published a series of beautifully illustrated papers under the title Some Slime Modes from Southern India. In 1955, Dr. Agnihotridu moved over to Assam as mycologist in the Toklai experimentation station of the Indian Tea Association. The mycologist in him blessed in the tea gardens of Assam, and he published a series of papers on various fungi that he found there apart from mycorrhizae. These were in the same nature of hobby, while he concentrated primarily on the disease of tea. Based on his findings on tea diseases, he prepared a thesis for which he was awarded the DSc of Madras University. This was a unique distinction so far achieved only by a few time academics working in the university. In 1964, Dr. Agnihotridu left Assam, came down to Bangalore as technical advisor to Rallis India Limited Pesticide Division. During his tenure with Rallis India up to 1985, first as technical advisor and later as director of research, Dr. Agnihotridu became an expert on diseases of plant crops and their management, especially on the usage and the residual effects of fungicides and pesticides. From 1985 onwards, Dr. Agnihotridu worked as director of Tea Research Institute of the United Planters Association of Southern India, and also a consultant to several chemical companies in India and abroad. He was also a member of various selection boards and committees focusing on the problem of plantation crops and pesticides. He attended and chaired various sessions in various international conferences in these topics. As a person, Dr. Agnihotrudu was charming and human in his dealing with people. His wit and charm were infectious. With his demurely beautiful wife, Indra, his lovely son, Subramanyeshwar, and his devoted cook, Rao, he had a happy home, which was always open to visiting friends and relatives. Dr. Agnihotrudu published on the papers on the subjects of his interest, number over 350. His monograph on pesticide residues have been published by Rallis India. I shared the link to Mrs. Indra Agnihotrudu. I hope she will be on the link. And this award this time goes to Professor Naveen Kango. Now I request Professor Bhagiraj to introduce Professor Naveen Kango.
Thank you very much, Dr. Raman. It's my pleasure to introduce Professor Naveen Kango, Head of the Department of Microbiology at Hari Singh Gaur Vishwavidyalaya, Sagar, Madhya Pradesh. He joined this university in 1997 and completed his PhD in 2003 and later did his postdoctoral work at Durban University, South Africa, and also at University of Helsinki, Finland. His research interest is on production and application of industrial enzymes such as cellulases, xylanases, manninases, illuminases, keratinase, etc. from fungi and also bacteria, and valorization of biomass for generation of 2G bioethanol. He has 45 research papers, 11 book chapters, 1 book and 1 patent to his credit. He is the principal investigator of several research projects and has guided several doctoral students. He is also a member of several professional bodies. Today he is going to present Dr. V. Agnihotrudu Memorial Award Lecture on production of fungal manolytic enzymes for health promoting prebiotic manoligosaccharides, MOS in short. Professor Kango, please go ahead with the presentation.
Thank you so much, Professor Bhagiraj. I think I am audible and my screen is visible.
Yes, yes, you are audible, screen is visible.
Thank you so much. Respected Chairperson, Professor Bhagiraj, respected Co-Chair, Professor Sudhakar Reddy, respected President of MSI, Professor Rupam Kapoor, respected Secretary, Professor N. Raman, and my dear colleague, Dr. Avneet Pal Singh, who has put up this program meticulously, and the moderator for this session, Dr. Arneet Garewal, all the senior and budding mycologists. Very good afternoon to one and all. Well, since morning, I have been receiving wonderful vibes generated by the voices of great mycologists, also, of course, along with their videos and pictures. So it is always a pleasure to discuss and learn about the developments in mycology at the wonderful MSI platform. In fact, it keeps us motivated and charged for the whole of the year. At the start of this memorial lecture, I extend my heartfelt thankfulness to MSI for bestowing this honor upon me. At this juncture, I feel that my contributions are too little for this prestigious award and recognition. However, I assure with all my might that I will try to be worthy of it in the coming future. And before delivering this lecture, I express my gratitude to my mentors, my PhD supervisors, late Professor P.C. Jain and Professor S.C. Agrawal, and also to the senior mycologist of this country. To name a few, Professor Manorachari, Professor T. Satyanarayan, Dr. S.K. Deshmukh, Professor Atri, and I would be failing in my duty, but of course I cannot take all the names here, but I do convey my regards to them. And I'm sure that they are serving like a lighthouse to a small boat like me. So to begin with my presentation, this is about the generation of monooligosaccharides using fungal enzymes. Professor Raman has already introduced this luminary, Dr. V. Agnihotridu, whose exemplary work on myxomycetes and disease on plantation crops and their management and usage and residual effects of fungicides and pesticides is being remembered today, and his contributions will be remembered forever. And I am proud to be associated with his name. So for those who have not visited my beautiful university, which was set up in pre-independence time in 1946 by a luminary, by a visionary, Dr. Hari Singh Gaur. This is the beautiful, picturesque, lush green campus. And I do wish to invite all of you sometime here. And this is the gate, and this is the Patharia Hills in which this university is situated. Well, coming to my talk, it is about the generation of prebiotics. So to introduce it very quickly, I am very well aware that we are short of time. So I will try to be as brief as possible. To introduce this, what are prebiotics? Prebiotics are the substances which, when fed to the host, are selectively metabolized by the friendly or good bacteria. In this way, the population of good bacteria is enriched, and they of course confer a number of benefits, and at the same time they drive away the bad bacteria. So this particular slide suggests the crux of my presentation, that we are trying to develop these prebiotic oligosaccharides using fungal enzymes. These are the benefits of oligosaccharides. Number of benefits are known, and many of them are being pursued. And some of these include that they offer anti-neoplastic activity, they enhance the mineral absorption, they reduce the obesity, they have the antioxidant properties, they are immunomodulatory, and they are hypolipidemic, and so on. So a number of benefits which stem up from by the consumption of oligosaccharides, and therefore there is a reason to work out that how we can generate oligosaccharides in a cost-effective and easy manner, so that they are more available and the idea is more feasible. This picture is to suggest that how prebiotics work. Well, these two terms go together, pre and probiotics. So to the naive audience or to the young audience, I must clarify this, that prebiotics are the chemical substances and which are utilized by these probiotics. That means that probiotics are the living bacteria. These are the bacteria, or even some of the yeasts are also known nowadays, like Saccharomyces boulardii. So these prebiotics are selectively metabolized by these bacteria, and then they produce, especially say lactobacilli and bifidobacterium, they produce a lot of short chain fatty acids that lowers down the pH, and then this is how they favor the enrichment of the population of the good bacteria. They also impart immunomodulatory effect, and so on. They also now the probiotics also produce bacteriocins, defensins, and a number of good things like vitamins as well. So this picture is to illustrate the good effect of the prebiotics conferred upon the host. Well, the indiscriminate use of antibiotics has led to emergence of multi-drug resistance and so on, like you can see in this picture. I won't elaborate much on this, but everybody knows about this. So one idea to avoid antibiotics is to promote prebiotics. So once you start administering these prebiotics, the gut health is improved, the good bacteria comes into play, and therefore the bad bacteria or the harmful pathogens dwelling in the intestine can be avoided. And subsequently, the drug resistance can be avoided, which has already become a big issue. So this is a review on prebiotic monooligosaccharides published recently in Food Chemistry, and interestingly, because of kind of a bent or inclination towards the fungal enzymes, most of the reviews published from our lab most of the times talk about the fungal enzymes. And interestingly, in case of prebiotic generation, fungal enzymes, I'm sure they have a role to play, they have a significant role to play. So basically, in our lab, we are exploring various kinds of enzymes which can be used to generate different kinds of oligosaccharides. So you can see inulo-oligosaccharides using fungal inulinases, that is, they hydrolyze inulin, then fructo-oligosaccharides using fructosyl transferases, and then mono-oligosaccharides using endo-beta mannanases or mannanases. So I will be talking more about mannanases, but a little bit about inulinases as well as FTases. So what we do in our lab is, firstly, we produce firstly we isolate the mold, which are usually newly isolated from various niches, and then we use them for production of the enzyme. We optimize the enzyme production. We try to characterize the enzyme, and then more importantly, we characterize the end product, so that because it is the end product which is important, and at the end we characterize the end product. We use that end product. We evaluate that end product for having this prebiotic health promoting benefits like anti-cancer activity, antioxidant activity, scavenging, free radical scavenging activity, etc. So now very briefly, this is a few slides are about inulinase that say that inulin is a fructan, and it is found in various various plants, and this is how you can generate either fructose or the inulo-oligosaccharides using fungal inulinases. So here is an example where I use Cleveromyces marxianus for generating fructose, which is a low-calorie sugar. And at the same time, we use Aspergillus niger endonulinase to produce oligosaccharides, inulo-oligosaccharides, which are very close to other oligosaccharides like fructo-oligosaccharides. So some more research review articles on inulinases published in different journals. Into fructo-oligosaccharides, the enzyme is named as fructosyl transferase. So it conducts two kinds of activities. One is hydrolysis of sucrose, and at the same time, the fructosylation of the sucrose molecule that leads to production of kistose, nistose, and fructofuranosyl nistose. Basically, these are the well-known prebiotic oligosaccharides which are already available off the shelf, and you can, you can always see them. You can always obtain them off the counter, and these are being provided with the probiotic bacterium most of the times, like lactobacillus and bifidobacterium. So these preparations which are visible on my screen are called as symbiotics, and they have both the things, pre and probiotics. So what I am trying to say is that FOS is the most commonly exploited, are the most commonly exploited prebiotics nowadays. And we developed one, I must say, a bioprocess for generation of fructo-oligosaccharides using a mycelial fructosyl transferase. This enzyme is bound to the mycelia of this fungi named as Aspergillus temeri. And I'm not going to talk much about this work because I need to focus on MOS now. But just to introduce that these are the FOS is being generated by Aspergillus temeri fructosyl transferase, and this is how it looks like. You can the enzyme was localized on mycelia, and then it produced kistose and nistose from the table sugar, that is sucrose. So that is the interesting part, that we can exploit this mold that possesses mycelial fructosyltransferase to generate fructooligosaccharides. And then, of course, the characterization of the fructooligosaccharides using NMR and FTIR. And the interesting thing about this enzyme was that the enzyme being mycelial, we never needed any kind of immobilization. We simply kept on recycling the mycelia, and that is how you can recycle the enzyme basically without purifying it. And then we studied the effect of the fructooligosaccharides generated by Aspergillus temeri. You can see how we can purify these oligosaccharides using Biogel P2, and then we use two different bacteria which are known probiotics, Lactobacillus acidophilus and Lactobacillus delbrueckii, for checking the growth promoting activity of these fructooligosaccharides. So the graph shows that yes, FOS could impart, growth promoting activity, growth promotion of these lactobacillus, and at the same time the pH was lowered down. It comes with this, that when the lactobacilli grow, they do reduce the pH because they produce lactic acid. So this is all favorable for the intestinal ecosystem. So these were the conclusions of the FOS work, and we published in a journal named as Food Chemistry, characterization of a mycelial fructosyltransferase from Aspergillus temeri for generation of fructooligosaccharides. So there are a few more research papers on generation of fructooligosaccharides all the way from fungi, and where we produce inulinase, fructosyltransferase, and sucrase enzyme from various molds, various fungal isolates, and of course immobilization and all that. So this was about fructosyltransferase, and then coming to today's topic, that is generation of mannose-oligosaccharides using endo-beta mannases. So mannan is a very interesting substrate because it occurs as structural as well as storage polysaccharide in plants. This is something rare, because either the polysaccharides are structural or storage. But in case of mannan, you can see it is found in pods. Also, at the same time, it is also found in the structural part, like a stem. It's basically a hemicellulose. It's just like xylem. It is a hemicellulose. So this picture shows guar gum, locust bean gum, konjac gum, and these two things which are agro waste, palm kernel cake and copra meal. So they all have mannan. They are all rich sources of mannan, and the mannan is a heteropolymer, and it's a polydiverse kind of polymer which has various substitutions, especially glucomannan and galactomannan. So definitely you require more number of enzymes apart from endo-beta mannanase. You need an exomanosidase, you need galactosidase and beta-glucosidase. So when I'm talking about generation of mannose-oligosaccharides, the major concern is whether the fungus is producing this endo-beta mannanase, and if it is producing, then how much is the titer, and what kind of an enzyme is this? What are the characteristics of these enzymes? So these are the questions we are trying to answer in our mannan work. Well, palm kernel cake is a waste that is generated by the oil industry, and it is generated in huge amounts. Similarly, the copra meal, which is generated by the copra oil industry. And interestingly, not many people know that they are very rich sources of mannan. So we are trying to exploit these agro waste for generation of mannose-oligosaccharides. So the effort is like value addition to a waste. Now, we started all this work, say, in 2015, and one of my students, Hemant Soni, isolated these number of molds. He screened them for mannanase production, and you can see some numbers on the screen, various titers being produced by different fungi, including thermophilic Malbranchia syndamomia and Aspergillus oryzae. Of course, this is an empty CC isolate, and then Aspergillus quadrinatus, which I'm going to talk more about. So the results of screening were published in 2016, and this zymographic analysis shows that fungi do produce more mannanes, and sometimes they are more than one, many times they are 4 or 3 and so on. We optimized the production of thermostable beta mannanase from Malbranchia cinnamomea, and purified the enzyme and characterized it, and then of course used it for generation of manotetraose from konjac gum, and manotetraose again from locust bean gum. So basically it's about generation of small oligosaccharides from gum using the fungal mannanase. Again, some publication coming from Malbranchia cinnamomea. And all these cultures, most of our cultures are deposited with this NFCCI facility at Agarkar Research Institute. Rahul Suryavanshi, one of my students, isolated this fungus named as identified as Aspergillus quadrilineatus, and the fungus grew profusely on this copra meal, which I referred which I told that it is a copra oil industry waste. And of course, all these molds like Aspergillus terreus, Aspergillus oryzae, Aspergillus quadrilineatus are able to colonize on copra meal. And then we optimized the production of mannanase using the response surface methodology, which is a rather simple technique, but a good idea to optimize the production. And many times you get yields enhanced up to 10 times or 5 times or 4 times. Then coming to the products of mannanases of these molds, these fungi, Aspergillus terreus, you can see. And interesting to note is that with different gums, mannans are basically gums. So basically, with different gums, you are able to see different products being produced using the same mannanase. So right now we are pondering on these things, that how to go after one particular mannan and develop a prebiotic preparation which is more meaningful. So you can see from LBG, the Aspergillus terreus mannanase produces manotetraose, manobiose, and mannose, and from konjac gum it produces manobiose, and in case of guar gum it's producing manotriose, and so on. So these are the usual experiments that we carry out in our lab, that we kind of make the enzyme react with different mannans, and then we are able to see different products using HPLC profile, and then we narrow down to which preparation will be suitable. Basically, our interest in this palm kernel cake and copra meal is because that they are the agro waste, so nothing, they don't accrue any costs. This is the idea. So you can see that we are able to produce M3 with the copra meal and palm kernel cake. These pictures are showing the profile of the sugar profile, the profile of oligosaccharides on polyacrylamide gel using a technique called as fluorescence assisted carbohydrate electrophoresis. So this is quite an interesting thing, that you can visualize your oligosaccharides on this polyacrylamide gel. Of course, you have to use a very high concentration of the gel. So this was using Aspergillus oryzae beta mannanase, and we published these findings in bioresource technology, where we produced the enzyme using Aspergillus oryzae and purified it, and then characterized it for production of mano-oligosaccharides. Now, how do we assess the properties of the prebiotic properties of mano-oligosaccharides? So basically, we conduct, we try to find out that whether they are offering us scavenging activities, they are able to scavenge the free radicals, like ABTS is a very commonly used example. Then do they have antioxidant property, where we use DPPH? So the results show yes, the oligosaccharides which are produced in the agro waste, when we use them for scavenging activity or for antioxidant activity, they do give a positive result. Similarly, you can see that the prebiotic activity in terms of the anti-neoplastic activity of the prebiotic oligosaccharides generated using various mannans, including LBG, guar gum, konjac gum, palm kernel cake, copra meal, etc. So this was tested again colorectal cancer cell lines. The idea is that these prebiotics, because they're consumed, can they serve as a neoplastic agent for the intestinal cancer? Then the growth promoting activities, like I already showed it for fructooligosaccharides, that when you compare the effect of MOS on the growth of probiotic strains like Lactobacillus delbrueckii, Lactobacillus acidophilus, then you are able to see that yes, the effect of copra meal hydrolysate and palm kernel cake hydrolysate in both the cases is quite promising, and it is over and above the positive control, that is when you use glucose. So these results were again published in International Journal of Biological Macromolecules that say that characteristics of bioactive properties of mono-oligosaccharides derived from agro-waste mannans, and the results comprise of the mannanase produced by the Aspergillus oryzae, MTCC strain. Now coming to the quadrillinatus work, you can see that again it is generating various oligosaccharides from locust bean gum, and you could see them in better fashion when you use this fluorescence assisted carbohydrate gel electrophoresis. It's more sensitive even than HPLC. We are yet to we are trying to optimize it for kind of having a technique to characterize oligosaccharides. It's under process. Then we did immobilization of Aspergillus oryzae mannanase on the metallic surfaces, aluminum oxide pellets, so that we can reuse it. And yes, you can see that this is how it gets immobilized. It gets hooked onto aluminum oxide pellets, and then you can, Aspergillus quadrillinatus mannanase, and then you can go on reusing it number of times for generating manotetraose, manotriose, and manobiose. So again, these results were published in Food Chemistry. Immobilization of Aspergillus quadrillinatus, a multi-enzymatic, because it has having more than one mannanase, so we call it multi-enzymatic system for generation of mono-oligosaccharide. Of course, it has some uses in fruit juice treatment also. So the prebiotic activities of hydrolyzed mannans by Aspergillus quadrillinatus mannanase. So you can see profiles, HPLC profiles of different mannans being hydrolyzed by Aspergillus quadrillinatus beta mannanase, and you can see number of oligos being generated. So we straightforward use this without purifying. We straightforward, because purification again is a costly affair. So we characterize. Of course, there are ways of various means of characterization. Like in this picture, you can see characterization using FTIR. And we are also trying to study characterization using NMR, where you can, elaborate, you can get elaborate details about the bonding and all that about the oligosaccharide. Okay, so again, the anti-neoplastic activity of the prebiotics generated using defatted copra meal, konjac gum, and locust bean gum using Aspergillus quadrillinatus mannanase. So you can see that they are offering significant, they are conferring significant anti-neoplastic activity, and it is of course maximum in case of the locust bean gum. Well, a simple experiment to study whether your prebiotics generated in different mannans, do they support again, like I showed some data regarding the growth promotion of probiotic strains. So again, those same probiotic strains, Lactobacillus delbrueckii, Lactobacillus acidophilus. So you can see in case of defatted copra meal and locust bean gum, the film formation and the growth promotion of the Lactobacillus strains is evident. Then trying to figure out the mechanism or what are the actual products when you grow your lactobacilli on the menin hydrolysates. So to visualize this, Rahul Suryavanshi, one of my students, could see the formation of short chain fatty acids and branch chain amino acid in the culture broth of the lactobacilli. Again, pointing towards the probiotic activity of the strains. Lately, one of my students, Uttam Jana, has been doing this in silico studies on the beta mayonnaise of Aspergillus oryzae, and we have already communicated these findings to some journal. It's underway, and we are able to figure out how the enzyme. These are the in silico studies on the binding of the beta mayonnaise with the oligosaccharides. So our conclusions regarding the use of beta mayonnaise, fungal beta mayonnaises on different mannins is like we are able to generate short degree of polymerization, DPs for that, short DP MOS from galactomannins and copra meal and palm kernel cake, which do have a number of prebiotic properties, including the growth promotion of lactobacilli and anti-neoplastic activity. Some more publications regarding the evaluation of mannan hydrolysates, like from Aspergillus terreus, and again Aspergillus terreus growth on palm kernel cake, locust bean gum, and guar gum. And this paper from where I worked with a great exponent of this xylanase, Professor Lisa Vicari. This paper formed the basis of my work on manninases. And from this paper, I got interested in working on mannins. So it happened in 2008, got published in 12, and so from there onwards, we are trying to figure it out. Well, to share with the mycologists, I'm happy to share that these are the two upcoming book chapters, or say review articles coming in the Encyclopedia of Mycology, which talk about application of fungal hemicellulases. Again, a lot of this involved mannan work, and application of fungal inulinases, which I referred to in the beginning of my talk. So, of course, our lab is also engaged in some work on cellulases, the generation of glucose, and then its subsequent fermentation to ethanol. Bhanu Pratap is involved in this. And recently we have published one paper on Aspergillus tuberculosis cellulase cocktail for generation of 2G bioethanol.
Can I interrupt you?
Yes. I have come to my last slide only.
Yes. Thank you very much.
This is about acknowledgements. I have already acknowledged my mentors, late Professor P.C. Jain, Professor S.C. Agrawal, my PhD students, and of course the funding agencies, and most of the analytical instrumentation available in the sophisticated instrumentation center at Dr. Harsing Vishwadhyaya. So this is my group working towards characterization of fungal enzymes. So together they are like 8. One is missing in this picture. So without taking much time, I thank profusely MSI for conferring this honor upon me, and I wish that MSI grows and grows well in future. Thank you so much for patient hearing. Thank you.
Thank you, Professor Congo, for the excellent award lecture. Arnit, can you focus me? Arnit?
Yes, sir. Yes, sir.
You focus me.
Sir, could you please stop the slideshow?
Yep.
Congo, sir. Yeah.
It's done.
Yeah.
Yeah.
Yes, sir. Yes, sir. Very nice lecture you delivered. I request the chairperson and co-chairperson to join me to give this award to Professor Kango, Professor Dr. Agnihotra Memorial Award.
Yeah, we are joining.
Yes, sir. Yes,
Thank
sir.
you so much, sir.
Yes, sir. Already in your inbox, the award is there. Your email. It will be posted to you. Thank you very much, sir.
Thank you so much, sir.
Yes, sir. Professor Bhagiraj, sir, you can go ahead with the next speaker.
Yeah, I just want to know, Dr. Amit Grewal, whether we can go ahead with Dr. Saritas. Has she sent it?
Yes, Sarita is ready. She is ready. We can move.
Well, I'm sure we can start off with that, sir.
One lecture.
Yeah, Dr. Aneet, we have the presentation with us.
Okay, sir. Okay, sir.
I'll play from here. I'll put up into slideshow mode right from here.
All right, sir. Thank you so much.
I request from Sarita Nasrat to restrict by 20 minutes, please.
Yeah, I wanted to tell that you have told Dr. Raman.
Yes, thank you so much. And I sincerely apologize for all the delay, and especially to my chair, co-chair, Madam President, and all the organizers, the esteemed, distinguished scientists. Mea culpa, please forgive me. I will go straight ahead. I request Dr. Bhagiraj to give me an alarm after 15 minutes, okay?
I will raise my hand.
Okay. Thank you, madam.
Thank you. Yeah, can we go ahead? Go ahead, please. Yeah, I had spoken on this. Go ahead, please. Can we go ahead? Yes. I'll start after this. As I said that the salinity is, I'll be speaking on that. Yes. Now, yes, the Dead Sea. Can I have the slide before that? Before that, please. Yeah, that's why it helps to. The one before that. Before that. No, you're going ahead, I think. This is going to take a lot of time. You've gone ahead. Go backwards, please. Okay, let it be. Just let it be on this. I'll speak that the Dead Sea has a very. Where are we? Yes, yes. Okay, there's a high salinity at the Dead Sea of 340 grams per liter, or PPT or PSU, as whichever you want to call it. And the major ions are magnesium, sodium, calcium. Now you will see the high, next slide, please. The high concentration of magnesium and sodium. Next slide, please. The next slide, please. Yeah, which makes it very, oh gosh, there are two gone ahead, which makes it very toxic to microorganisms, okay? And that is why it causes a paucity of organisms. Now, the ones we isolated were mainly of Aspergillus and Penicillium. We also had obligate halophilic isolates, namely A. penicilloides. We had just one of fungus. The next one, please. Yeah, so this is the whole list of organisms. I won't dwell on it. We also had Penicillium cladosporium. The next, please. Yeah, this now you will see on this that there's no growth in the absence of salt, and still at some of them, I've grouped them according to the salt at which concentration at which they grew. This is 2%. Some showed a delayed growth, and therefore we put it here. This is measured after 7 days of growth, and some not even at 5% grew, okay, as readily. So you can see that these were obligate halophiles. They essentially needed salt in the medium. And these were all the penicilloides, this one of fungus. And then you have those that were facultative in nature. That means even without salt supplied, they grew, but they grew optimally when a higher concentration of salt was supplied, and therefore we termed it halophilic, but facultatively so. The next, please. Okay, I'll skip this. I'll go faster. The next, please, because I'm losing time with the change of slides. Yeah, this is just the identification of the cultures by the ITS sequencing. The next, please. This was the first publication that we have to say a first record of obligate halophilic aspergilli from Dead Sea. The next one, please.
नहीं.
Yes.
नहीं मेरे
Okay,
मैं
now
तो यहाँ अभी तो सेशन चलेगा मेरा.
Yeah, okay. We studied the organisms from an estuary, which is now, as you know, an estuary has the influx of
अश्विनी को पूछ लेना, वो भी कह रहा था उसको वैसे मना किया था, पर वो ऐसा ना हो कि वो भेज दिया लिया.
Something to me.
हाँ.
Okay, so and so you have a mixing of waters, and the salinity gradually decreases as it goes hinterland. We wanted to see what sort of organisms we would get, whether we would get halophiles. And in fact, we did. And again, Aspergillus penicillium, we also got some Ascomycota. We had Cladosporium. The next, please. We also went to mangroves, which is the polyhaline environment. And that is roughly more or less that of seawater, around 35 PSU. The next, please. This shows all the organisms. I won't go into it. You can see there Aspergillus, Penicillium, Eurotium. Yeah, the next, please. Cladosporium. Next, please. Okay, these once again are similar to those. I've explained to you that some, you can see it's not growing without the addition of salt, solar salt we were using, whereas some grew with the same sort of nature. And I'll go ahead to the next. Could I have the next, please? Okay, this is something similar. Again, you will see all the penicilloides which were obligately halophilic and the facultatively so. This is now, I forgot to mention, this is the estuary. So we took the surface water that is just below the absolute top, the bottom water which is down, and then that using the Niskin sampler, and then the grab for the sediment down. So this is now the bottom water. And the next one will show you the sediment. Okay, once again. Now, one thing I may mention is that these were mainly moderate halophiles, if you can see, growing around optimally at 10% salt. Okay, one from the Dead Sea showed even at 15%, so it's sort of an extreme also, because remember I said 15% salt is the extreme halophile. Okay, optimal growth. The next, please. This was the publication for the estuary, that we had halophilic fungi even though the waters were not hypersaline, as in they were the normal salinity and even going down to fresh water. And we've explained that perhaps the mangroves were supplying nutrients for these organisms to start building up osmolytes, and therefore they had the capacity to grow even in presence of very high salt. And perhaps these are spores that got carried away and are washed away, but they do exist there. The next, please. Also, we did from mangroves. I will stress only on the black yeast that we obtained, Hortea wernecki, which was also published in Tavaka then, because this was normally recorded as being from hypersaline environments of solar saltins. The next, please. If we could have the change of slides rapidly, it would speed up so that I don't lose time. Now, salt concentrations in the solar saltins, the previous one, please. The solar saltins have a very high, the previous one, you're going ahead, if I may say. Anyway, the solar solvents have a very high concentration of about 330 PSU. That is the concentration, especially at the time of halite deposition. And these were organisms that we did. So these are just the cumulative from the mangroves and saltans. The next, please. Yeah, and these are the curves. You will see that these were moderate halophiles, slight halophiles. I hope you can see it on your screen because my screen is lifting up here and I can't see it. Anyway, the moderate and slight halophiles is down on the thing here, moderate, slight halophile, depending on, now this is again from the mangroves, the sediment. Okay, the next, please. Here, the same sort of thing. Again, mainly the aspergilli are moderate, penicillium are moderate or slight, and Hortia was moderate. We will see the curves for Hortia Wernicke later. The next, please. I'm hurrying through this because it won't really interest people, I guess, from the strict background of the other botanical subjects. For us, it was microbiology, and therefore we focused on these aspects. And basically because it was extremophilic halophytes. Once again, from the solvents, the same sort of, the next one. I'm going through because these are very similar, but just showing you the results that we obtained. Now, halophilic fungi require NaCl for optimal growth, but they do not need salt for their viability. So they can survive, and they can adjust and grow in a wide range of salinity from fresh water to extremely hypersaline waters. Now, they adapt to this broad variations by surviving periods of extreme stress in a resting state. Under favorable conditions of moisture, they then respond with increased metabolic activity. Now, this is especially pertaining to the solar solvents, because we must understand that when the salt is precipitated, they are moved out till the time now that, you know, after the monsoons and then slowly whatever, there is a period of desiccation before the whole aspect of precipitation of salt and salt production is again started somewhere just before the May season when it's extremely hot. So meanwhile, those organisms survive. Okay, so under favorable conditions of moisture, they respond with increased metabolic activity and days. Now this is known as poikilophilic halophily, which enables continuous colonization of these places. The next, please. This was just the publication we had. This was in Indian Journal of Geomarine Sciences. And the next one, please. We go to Hortia, as I mentioned. This was in Kavaka. And it will show you that these cultures were urihaline, sorry, may I have the previous? That they were urihaline, as many of those were, but I'll just explain it here. That means they can grow on a wide concentration of salt, and being black yeast, I didn't mention what were Hortia. They are black yeast, and therefore they also begin to have mycelial form, but they don't grow very big. So you will see that the diameter is 1, okay, and less than 1, okay. And so these were various cultures from mangroves and saltans. We put this separately as mangroves because what we wanted to say was that for the first time we have shown them to exist in mangroves. And we can't say they came from saltans to mangroves because the water is let in from mangroves into saltans and then the sluice gate is closed. The next, please. Okay, so now normally in reports, they have been termed as halophilic because they can grow at near saturation salt concentrations of 32%. However, because of its ability to grow in absence of salt, it has also been reported as halotolerant. These are reports. However, we termed it here as a facultative halophile because it showed no obligate requirement for salt, and it could grow even in the absence of salt and grow well. But yet it grew better when salt was supplied. The next, please. Yeah, the next one. So this is just a summary of all, and you will see that they were growing at about 20 to 25. The aspergillus tends to be higher, okay, the obligate ones, the facultative. And then all the rest were facultative. You will see later that I have mentioned the aspergillus species alone had both obligate and facultative, and the only obligate one belonged to Aspergillus penicilloides. This was a remarkable finding that we had, okay? And then Hortia also, so 25. This is just the maximum tolerance that was there, and we tabulated this together. The next, please. These were the various organisms. So this was the estuary, mangroves, and the saltans. I didn't put the Dead Sea picture, but anyway, this is Aspergillus penicilloides. You can see the very crumpled sort of raised thing, which has a very unique colony appearance. And this was Hortia. The others are Penicillium, Aspergillus versicolor, etc. Yes, the next, please. Now, we were trying to study all the Aspergillus penicilloides from different ecosystems. And so we looking at the salt tolerance index, that means the ratio of growth at a particular salt concentration to that at the maximum growth. So that obviously would be 1, and that is mainly at 10%. And you can see that, so this gives a picture of all of them together rather than the salt tolerance curves. Okay, I'll go to the next. I won't spend time.
Hello. We have finished 15 minutes, Dr. Sarita.
15 minutes. Okay. So I will move ahead. Please, the next, please. If we can have, the minute I say the next, it has to be very quick if possible. So this is showing you how the cultures can grow very well in the presence of salt, but they could not grow in the absence of salt. And this will show that they just about germinated or didn't, but they couldn't grow. But in the presence of salt, they grew. The next one. This shows the germination, that in the absence of salt, they just couldn't grow. The presence, I put just representative ones. The next, please. Now, we have described them as true halophiles because of the essential need for sodium chloride. The next, please. Next. The next, please. Yeah, thank you. The next one. This was the proceedings from the Halophiles Conference to 2013, and this was for the special journal on extremophilic extremophilic fungi of Kavaka. The next, please. So it was a review article with all our. The next, please. Yes. Now we went to. I'll go to the next, please. This is showing you the osmolytes. We studied the osmolite buildup that was there. The next. This is showing you the total osmolytes. So this was the sugars and the polyols, and these are the total osmolytes. Okay, just to give a picture of what was happening. Next, please. Okay, now I didn't stop on this diagrams to show you that they had 4 to 6 carbon, apart from the sugars of sucrose and trehalose, they had 4 to 6 carbon chain polyols, erythritol, etc. The total concentrations of 5 carbon and 6 carbon polyols were higher than that. And in fact, the longer the carbon chain of the molecule, the greater the capacity to function as an osmolyte, obviously, okay? Because the hydroxyl groups, as I said, are what contribute to the osmolyte nature of it. Okay, and a mixture of solutes, in fact, enhances the efficiency in increasing water pressure rather than a single one. Besides, avoiding accumulation of too much of one creates a toxic effect. I mean, the accumulation of any one creates a toxic effect. So a mixture helps to reduce the toxicity of any given one. The next one, please. And the next one, please. The next, please. We thought we would also see it at at different salt concentrations, how it grows. So therefore, you can see 5%, 10%, 15%. You will see that the total osmolyte concentration is increasing with the salt concentration. However, though the growth didn't increase, we've not shown that here, because probably the energy was now moved towards synthesis of osmolytes, which helped them to survive. And this is, we just thought we would check the sodium, potassium, iron accumulation, whether there was or not. As in archaea, though it is purported not to be there, we said, let us just examine what happens. The next one. You can move the next 2 slides. Yes, the next one, please. The next. Yeah. Now, they were, as I said, to open the next. I'm hearing the end. I'm hearing the end.
Yeah,
the next, please. The next, please. Thank you. I'm just fighting for time. The next. That's what, if I had it with me, I would have. The next, please. Can I go to the next because my time is up? Next, please. Yeah, now we found that a lot of salt was deposited on the cell wall, and fungi have that capacity because of the negative ions on the cell wall to absorb salt. And so therefore that becomes the first line of protection for the organism. The next one. Yeah, the next one. I will skip all these and I will go just to the, I would like to acknowledge, please, the next one, please. Go to the last slide. I'm concluding. Yeah, okay, this is a summary. I will skip this. The next, please. The next, please. I think I've lost time with the movement of slides. Yeah, I want to acknowledge the work done by my research team in the area of halophilic fungi. And in particular, I want to acknowledge Dr. Valerie Gonsalves, who has been my student for her PhD, but has continued as a collaborator in this area. And she's currently lecturing at the bachelor's program in microbiology. Thank you. You can keep it on the last slide to say.
Lecture. Arnit, can you, Madam, kindly close the, sir, can, Professor Bhagiraj sir and the, Professor Sudhagar Reddy, can you join me?
Yes, yes, we are joining you. Yeah.
So thank you, Madam, for this excellent award lecture. The MSI is very much honored to have you this award, Professor C.V. Subramanian Memorial Award for Women Scientists, and I think this is the last term for this award. So the next term it will be Professor C.V. Subramanian Memorial Award, and the award will be sent to you post, and the copy is there in your inbox email, Madam. Thank you very much, Madam.
Thank you, and I want to thank you, Professor Raman, and the Mycological Society. Thank you to my chair and co-chair, and my apologies once again, but thank you for your patience with me. The award is also due to my students who have contributed to this work.
Yes, Madam. Thank you, Madam. Thank you. Thank you very much, Madam.
Thank you. God bless.
Thank you. The next memorial award will be Professor K. Nadarajan Memorial Lecture. Professor K. Nadarajan, Professor, retired from Center for Advanced Studies in Botany, University of Madras, and past President of Mycological Society of India, suddenly passed away on 21st April 2008, which incidentally happened to his 65th birthday. Dr. K. Nadarajan was born at Kovilur, Tanjavur district in Tamil Nadu on 21st April 1942, and had his schooling at Kadalur. Later he joined at Vivekananda College at Madras and obtained his BSc degree in botany and first class in 1962, and received gold medal from Madras University. After working as demonstrated in the same college for a brief period, he joined the Indian Agricultural Research Institute, New Delhi, and obtained his MSc degree in mycology and plant pathology. Subsequently, he joined the Center for Advanced Studies in Botany, University of Madras, to work for his PhD degree under the supervision of Professor C.V. Subramaniam, and received PhD degree in 1970. Professor Natarajan was appointed as a lecturer in CES in Botany in July 1968 and promoted as a reader in July 1976 and then professor in 1984. A position which he held until his retirement in June 2002. Subsequently, the Madras University designated him as an emeritus professor in CES in Botany, which he was holding until his death. Professor Natarajan was associated with several scientific societies, and his association with Mycorrhizal Society of India deserves special mention. He was founder member of MSI and served as treasurer, secretary, president, and look over the editor of Kavaka from the year 2002 onwards till his death. He devoted his time and energy for the growth of the society and so as to bring the journal in time. His untimely death in the year 2018 is great loss to MSI. Dr. Natarajan is survived by his wife and one son and one daughter. He is always remembered for his pleasant temperament, soft-spoken and helping nature. He trained several young research students from different parts of India on collection, identification and description of fleshy fungi. He was deeply religious and ardent devotee of Sri Sai Satya Baba of Puttaparthi. The discipline of mycology has lost an outstanding scientist in his death on Professor K. Natarajan. I am the second student, PhD student of Professor Natarajan. I have sent a message to his son Sri Ram, the link also. Probably he could have entered the link. And this time the Professor K. Natarajan Memorial Award goes to Professor Rupam Kapoor. And if Rupam Kapoor accepts, she can finish the lecture by 20 minutes, please.
Surely, sir. Surely.
Madam, before you start, whether Professor Lakanpal and Yashpal Sharma as well as Dr. Geeta, the moderator, are ready for continuous presentation?
Yeah, we are ready, sir.
Yes, sir. Yes, I am ready. Dr. Geetika Sarhandi from Patiala.
The secretary is also ready, and after this we are continuously going.
Okay.
Thank you.
Sir, there is a submission.
Yeah.
We can have quick break of 15 minutes for lunch.
15
Sir, the people can in between they can go for lunch, sir.
Okay, sir, as you wish. Okay, sir, done.
They can go. Even myself, I have not taken, but I am.
Okay, sir. Okay, sir. Ma'am, Rupam, ma'am, please.
Congratulations to everyone, and thank you.
Okay, I think we will go ahead with the introduction of Dr. Rupam Kapoor. Yeah, already been introduced. And I'll very briefly introduce her. Professor Rupam Kapoor is the head department of botany at Delhi University. And she completed her PhD in 1998 from the same university under the guidance of Professor K.G. Mukherjee. And later she worked as postdoctoral fellow with the DST Boy Scout Fellowship at Alabama, USA. And her research interests are on plant-fungal interaction, both symbiotic as well as pathogenic. Her work on fungi, especially enhancing terpenoids of commercial crops and amelioration of salt tolerance, is highly cited. Her work on virulence of Botrytis cinerea and pathogens of safflower, especially is well known. And she is the PA of several projects and is on the editorial board of several journals. She has more than 60 publications in reputed journals and more than 6,500 citations. Currently, she is the president of MSI. Today, she will present Professor K. Natarajan Memorial Award lecture on understanding the role of some novel components in modulating virulence of necrotrophic fungus Botrytis cinerea. Madam, I request you to finish in about 20 minutes.
Surely, sir. Thank you. I am thankful to MSI for bestowing this honor to me. What an honor to be associated with such an eminent mycologist. And the cherry on top is that this conference, this session is being chaired by Professor Bhagyaraj. I would not have asked more from MSI. I want to place on record my supervisor whose wisdom and passion for science and authoritative guidance has immensely been valuable in my growth as a person and blossoming the scientific temperament in me over all these years. My greatest degree of appreciation goes out to him for exposing me to the realm of fungi. Today, I would be presenting a portion of my work, understanding role of some novel components that impart virulence to the necrotrophic fungus Botrytis cinerea. Botrytis cinerea is a necrotroph which is capable of infecting about 500 plant species. It is popularly known as gray mold, and annual losses incurred by this fungus amounts to be as high as 100 billion US. It survives as sclerotia, and high humidity, senescing plant tissue, high sugar content, and low temperature are favorable for its growth. It is capable of infecting plants at all stages, starting from a standing crop to a harvest in transit and the ones stored in cold storage. It causes various infection depending upon the host, necrotic spot on apple, moldy growth on strawberry, blemishes on rose and marigold. It primarily stays as an asexual stage. It forms conidiophore, which infects the leaf, later on form perinating organs, which are melanized called sclerotium, and later on which again germinate to form conidiophores. Past literature reveals several virulent factors that have already been reported for botrytis. Being a necrotroph, cell wall degrading enzymes play an important role in its pathogenicity, reactive oxygen species, certain components of signaling pathways, and tetraspanins are involved in enabling the penetration of the pathogen into the host leaf. A positive side of this fungus is when under low humidity infects grapes, it imparts a sweet taste to the wine that is obtained from these grapes, and the wine that is obtained is known as noble rot. A lot of work has been done on botrytis, as it has been named as the second most economically important fungus. But the work has, in pursuit of some novel pathogenicity genes, we carried out targeted genes. We went ahead with agrobacterium mediated tumefaciens transformation. For this, instead of, we took a virulent strain from GB Pant University, Uttarakhand. This strain was found virulent on grapes, marigold, rose, chickpea, tomato, as well as Arabidopsis. So with agrobacterium tumefaciens mediated transformation, which offers several benefits. I would not be going through this now. And our workflow was first to raise agrobacterium mediated transformation transmutants, screen them through several rounds for pathogenicity, and select those mutants that showed impaired virulence, and identify the tag gene, and then functionally validate the genes for their role in virulence. For this, methodology for transformation of botrytis cinerea, which was carried out in National Phytotron facility of IRI. The pathogenicity studies were carried on chickpea, and compared to wild type, the mutants that we selected showed decrease in virulence by 94.5 and 92%, and the other showed decrease in 79.4%, and BCM 45 and 780 showed decrease, or they were totally apathogenic. Transformation was confirmed by PCR of hygromycin resistance, and southern blot hybridization was also done to confirm and take only those contents that only single copy insertion of tDNA. That gene was identified by thermal asymmetric interlaced PCR, and mutant BCM425, remember, which was totally avirulent, had insertion, or what the gene disrupted was superoxide dismutase. Superoxide dismutase is the gene that is involved in detoxification of intracellular superoxide ions and secretion of hydrogen peroxide. However, this gene was already reported earlier for its role in virulence of botrytis cinerea. So we went ahead with the other genes that were disrupted by insertion of tDNA. One of them was DGAT2, that is diacylglycerol orthoacyl transferase 2, nucleoporin complex protein, kinesin protein, and predicted protein. So we went ahead with 4 mutants for further characterization. The characterization of mutants was done by target deletion and complementation in the background of targeted deletion. And the mutants so obtained were morphologically characterized and biochemically characterized for the virulence factors. Diacylglycerol orthoacyl transferase is the enzyme.
हमारा तो ये हो सकता है थोड़ा लेट चले सेशन, तुम्हारा क्या प्रोग्राम है?
That catalyzes the acyl transferase from diacylglycerol to triacylglycerols, which are required as repository of lipids for synthesis of membranes. Targeted deletion and complementation study was done, and the expression of the gene was confirmed by RT-PCR, which was almost negligible in case of insertion mutants and deletion mutants. The expression was restored in the complemented clones. Since DGAT2 is involved in the synthesis of triacylglycerol, the concentration of lipid was also determined, and it was found that concentration of lipid was less in insertional and deletion mutant, and that of triacylglycerol was also less. The TLC showed a very faint band of triacylglycerol in insertional and deletion mutant, whereas the concentration of triacylglycerol was restored in complemented clones. There was not much difference in the morphology of the fungus. However, its ability to form sclerotium was lost. I would like to add that sclerotia are the organs in which the lipids are stored, and presence of is essential for inducing the differentiation of fungus to form sclerotium. Sporulation rate in the mutants were reduced. Pathogenicity were tested on various host plants in vitro as well as vivo, and it was found that it has response of mutant to stress in terms of osmotic stress, oxidative stress, membrane stress, and cell wall stress was also assessed, and it was found that, as the slide, the mutants were not able to tolerate membrane as well as cell wall stress, as it is evident in the slide. So plants' response to mutant was also determined. Plants were treated with wild-type conidial suspension and mutant conidial suspension, and response was determined, and it was found that while the disease progressed in case of wild type, there was very few lesions found in case of mutants. The oxalic acid production and production of cell wall degrading enzyme polyglucosidase was less. The reactive oxygen used in mutant the stress produced in response to the mutant in plant was less. And the enzymes, so in wild type, it's found that while the synthesis of triacylglycerol results in robust hyphal growth. Video bhi mute ho gaya. Fungal membrane. In case of mutant, when the triacylglycerols are not synthesized, there is aberrant hyphal growth, deformed fungal membrane, reduced mat-like growth, no sclerotia formation, impaired penetration by the fungus, and incompetency to grow under stress, and reduced oxalic acid and polyglucoronase activity. The second gene that we identified was nucleoporin complex protein that is involved in selective trafficking of proteins, nucleic acids, and other factors between nucleoplasm and cytoplasm. And we confirmed its expression in the mutants that were raised. Phenotypic characterization was done, and its in vivo pathogenicity assay was carried out. And ability to stress of mutants was also determined. Biochemical characterization was done, which showed reduced production of oxalic acid, pectin methyl esterases, while there was not much difference in production of there was decrease in polyglucoronase activity as well. And the response was same. We summarized the study that while the protein NUC184 was being expressed, the cargo from the cytoplasm could be transported inside the nucleoplasm, and from the nucleoplasm, with the help of exporting protein, could be exported into the cytoplasm. In case of mutant, when no NUC184 was expressed, there was no transportation, and hence the factors that were responsible for the virulence of the fungi were also not being transported across nucleoplasm and cytoplasm. That might have contributed to impaired virulence of the mutants. What is exactly is being transported is yet to be deciphered. The third protein that we studied was kinesin protein. Kinesins are microtubule-based energy hydrolyzing motor proteins that facilitate cell division and intracellular transportation. KLP7 that we found in this case has been found unique to phylum Mycorrhiza. The deletion mutant and complementation mutant were raised, and expression of kinesin protein was determined by reverse transcriptive PCR, and the virulence and infection related lesion size was smaller in mutants, the disease severity was less, the sporulation was less. Wild type, though it formed a uniform and broad hyphae, in case of the deletion mutant, the hypha was irregular and showed irregular branching. The ability to colonize was also studied on onion epidermis, and that also showed impairment in the ability of the mutant to penetrate and colonize the host. The concentration of oxalic acid, polyglucoronase activity, pectin methyl esterase activity was also less. We summarize here wild type, which was expressing KLP7, It helped in the transportation of the secretory vesicles, and these vesicles were probably bearing virulence factors. However, in mutants, since kinesin was not being expressed, there was no transportation of secretory vesicles that resulted in stunted and irregular hyphal growth, spore formation, decreased conidial germination, and impaired ability to penetrate the host plant. Next is a small experiment, and we are yet to validate molecularly the role of predicted protein that is uniquely found only in Botrytis cinerea and not on any database. This work which I am presenting was published in Kavaka. The wild type was totally impaired, and the mutant was totally impaired in its virulence. And we found that this mutant caused no damage, but was able to induce defense responses in plants. So we hypothesized that inoculation with this non-pathogenic mutant might equip the tomato plants to deal with subsequent biotic stress in a better way. So we first treated the tomato plants with wild-type conidial suspension and that of non-pathogenic conidial suspensions. And then these plants were then touched with a nectar that was Botrytis in itself, and separately with a biotroph, Pseudomonas syringae, and it was found that the plant showed resistance to subsequent attack both by necrotroph as well as by necrotroph as well as a biotroph. So this is what we summarize, that this work has led to the identification that have never been implicated earlier in virulence of any microorganism. The identified gene are conserved and can be extrapolated to investigate their role in other fungi. Predicted protein that is just present only in Botrytis cinerea can be exploited to develop biomarker against the pathogen. An apathogenic mutant that has been identified can be used as biocontrol agent against phytopathogen and aid in development of a sustainable disease management strategy. With this, I thank all the MSI members, including Professor Bhagyaraj, Professor Manorachari, and other distinguished members for the patient hearing. Thank you very much.
Okay, thank you very much, Dr. Rupam Kapoor, for taking care of the time and not losing the subject. Both you have taken care of. Yeah.
Thank you, sir.
Thank you very much, sir. Thank you, ma'am. I'm sure our delegates have. Yeah. Now I would hand over the audio to Dr. Raman. He's presenting the award for ma'am. Sir, you need to unmute yourself. Raman, sir, you need to unmute yourself.
Unmute.
Yeah, Professor Rupam Kapoor, thank you very much for an excellent award lecture. In time, I can say in time. So on behalf of MSI, I am very glad to hand over this, and the chairperson and co-chairperson are joining me, and in another few minutes, your award and the certificate will be in your inbox. Thank you very much. Thank you very much. Sir, on behalf of MSI, I thank Professor Bhagyaraj, the chairperson, and Professor Sudhakara Reddy, the co-chairperson, for excellently executed this session. Thank you, sir. Thank you very much. Thank you very much.
Pleasure. It's a pleasure. I would like to thank the organizers for having given me this opportunity, and also the co-chairperson and all the three speakers, the award winners. Excellent work they have done. My hearty congratulations. And I also want to thank the moderator, Dr. Arlene Grewal. Thank you all very much.
Thank you. Thank you, sir.
Thank you. Thank you, madam. Thank you.
Thank you, sir. And a hearty congratulations to all the recipients of the coveted awards. Thank you very much.
Thank you, Dr. Arnit, for good moderation.
Thank you, sir.
Thank you. Thank you. So the next, I request the next moderator, Dr. Geetika. She is available.
I'll just check, sir. I'll just check.
Dr. Geetika will be with us in a moment, sir.
So till then, can you take over that one?
Okay. Meanwhile, she joined. Let me invite the most respected mycologist of the country.
Yeah.
And a father figure to us, rather a grandfather figure to us.
Yeah.
Our very own and always there at our rescue, Professor T.N. Lakhanpal, as the honorable chairperson for this session. May I request Lakhanpal sir to kindly chair this session, and at the same moment,
Thank you.
And at the same moment, may I request the dynamic editor-in-chief, new editor-in-chief of Kavaka, Professor Yashpal Sharma, who is also equally motivating us, all of us, with his enthusiasm, with his planning, with his execution. So may I request Professor Yashpal Sharma to kindly join us as the co-chairperson for this session.
Yeah, thank you, Dr. Geet, and my humble pranam to everyone. And really, it's a wonderful, moment that I have been, given this opportunity to be with one of my mentors and one of the greatest mycologists of the country, Professor T.N. Lakhanpal, and it's wonderful having a galaxy of mycologists across the country, from across the country. And I have been, hearing and watching all the proceedings since morning. And, one word which I would describe is the quality, is the scholarship which has, been presented over here by one and all. So this is a great moment for all of us. And my heartiest thanks to the organizers for giving this opportunity, and also heartiest congratulations to the people who have been awarded, particularly Professor DJ Bhatt and other award lecture awardees. So thank you very much. Lakhanpal sir, namaskar.
Namaskar. And thank you very much, Dr. Raman, Dr. Avneet, Dr. Manruchi, Professor Manorachari, Professor Bhagyaraj, Professor Rupam, Professor Bhatt, and Professor Yashpal.
Yes, sir. Yes, sir.
This is my good fortune that I am associated with you all, and I have been receiving your love all through, and I hope this continues both ways.
Okay, sir. Okay, sir.
Yeah.
Sir, before introducing the candidate, I will introduce the memorial award lecture. The award lecture is Dr. S.K. Som Memorial Lecture. Dr. Sarit Kumar Som was born on 15th February 1933 at Lucknow. He obtained his master's degree in botany in 1955, PhD in medical mycology in 1959 from Lucknow University, under the guidance of Professor S.N. Dasgupta, and was appointed as an assistant professor in the same university in 1960. In 1961, he went to Instituto de Mycorrhiza University de Recife as research visitor, and was appointed as a mycological specialist. from September 1961 in the same institute. During his stay in Brazil, Dr. participated in the Brazilian Dermatological Congress in 1961 and the 13th Botanical Congress of Brazil in 1962 on behalf of the Institute de Mycorrhizae. He was invited by the Ministry of Education to deliver a course of lectures on mycology and its applied branches at the Escola de Agronomica. On return to India in 1962, he joined University of Kalyani, West Bengal, and shifted to National Institute of Communicable Diseases as Assistant Director of Microbiology Division in the year 1967, where he established the Medical Mycology Laboratory. His practical contribution to national public health includes investigation of mycotic infection in the rural areas of Uttar Pradesh, West Bengal, Delhi, and Haryana in India, Recife and Manaus in Brazil and South America among the population under tropical conditions. His outstanding work on mycosis in these areas bears testimony to his dynamic drive and dedication to work. Dr. Shom attended a number of conferences and seminars and presented several papers. In them, he was invited by Director of TB Demonstration and Training Center, Agra in 1975 to help in developing their mycology unit. He was a special invitee to in the Symposium of Human Mycosis and Myconocole organized by Ethanol Limited in 1974. In a short span of his lifetime, Dr. Shom published over 60 papers in various journals of the national and international repute. In addition to these, several of his works remained unpublished to his sudden demise. Dr. Shom, survived by his wife, daughter Anuradha, and son Anupam. Anuradha and Anupam, they contributed 95,000 to the MSI to make it 1 lakh, and it has been deposited in the bank as a deposit on this memorial award. And Anupam, and this year for this award, SK Shom Memorial Award, Dr. B. Venkateswara Sarma was selected. I request the chairperson to introduce Dr. Sharma, please. And if Sharma accepts, kindly take 20 minutes, please.
Okay, sir.
Let me, sorry, Dr. Sharma, to interrupt you. Dr. Geetika is with us. May I request her the honor of introducing the chair and co-chair briefly. Dr. Geetika, please.
Thank you, Dr. Avneet. So first, good afternoon to all, to honorable members of MSI and as well as the delegates who joined online this conference. So it's my honor to introduce two beautiful souls as a moderator for this session. One is Professor T.N. Lakhanpal sir and Professor Yashpal Sharma sirji. I welcome you on this online conference, virtual conference. So first about Professor T.N. Lakhanpal sir. He is so beautiful human being that I have no words to introduce him. He is himself a personality. Everyone knows, particularly mycologists. They know that what a beautiful person Lakhanpal sir has. Welcome, Lakhanpal sirji. So as a moderator,
thank you, Geetika.
Thank you, sir.
Thank you very much. I think let it be brief because of time.
Okay, sir. Okay. I try my best, but I have so much, and I am so pleased that I get this opportunity to introduce you.
He is well known, madam.
Okay, sir. Okay.
Well known.
So, Professor Lakhanpal sir, He is a retired professor and head of Department of Biosciences, and he also served as Dean Faculty of Life Sciences as well as Director Institute of Integrated Himalayan Studies. So he's a great, along with a great human being, he's also a mycologist, and he has so many papers, books, awards in his life. Get this, and he guided 28 PhD students, and he is also served as chairperson or member of various national committees, and he is also an editorial board of number of journals. I think, if I'm not wrong, with plant pathology, microbiology, and biotechnology. He's also, I think, serving as on the editorial board of Kawaka. So I welcome you, Professor Lakhanpal sir, as a chairperson of this session. Second one who is associated with in this session as a co-chairperson is Professor Yashpal Sharma Sarji. Professor Sharma sir, please, can you make your video on? Thank you, sir. Thank you so much. So next person I am going to introduce to this mycological conference is the Professor Yashpal Sharma ji. He is from Jammu University, as you already know. Great scientist, great mycologist, plant pathologist, ethnobotanist, and along with he is very fond of rare endangered species conservation. And because of his efforts, he make a beautiful garden established. I think it was already established, but he nurtured that garden beautifully at Jammu University. I saw that and I visited that garden too many times. So welcome, Professor Sharma sir, to this session of this MSI. Now,
Geetika,
Hanji, sir.
that is cactus garden, but he is not cactus, he is rose.
He is rose, yes sir. He is rose, beautiful rose with beautiful fragrance. Everyone who come in touch with him, he get fragrant from that. I really agree with
and
you.
You are the spirit, sir. And you are the spirit
of cactus.
Not at all.
Yes, sir. So I hear Professor Sharma his talks on radio and all that, and so with these beautiful, but he has got so many awards, so many achievements, so many feathers in his hat as a scientist, as a academician, as a social person. He's very good personality also. So I welcome you both, and I hand over you this session for further proceedings. Thank you, sir. Thank you so much.
Thank you very much, Geetika, for kind words and very kind introduction.
Thank you, sir.
I think we will start with the proceedings so that we utilize the time. And but I don't have the biodata of Dr. Sharma, but nevertheless I know him. I hope he would not mind the brief introduction that I give. Yeah, Dr. Sharma, I know he is very enthusiastic, very good organizer, and you know, always he is with very, very high spirits, and he is a very good worker. He is very amicable and you know, very enthusiastic right from the beginning. I have seen him known him for many years. And I think he is a very right choice for Shom Memorial Lecture. And I will request him to start with his presentation. And Dr. Sharma, please try to finish it in 20 minutes. And we would be grateful, because let us economize on time so that it doesn't go beyond time. So Dr. Sharma, please.
Thank you, sir. Thank you for your kind words. I'll directly switch out to the presentation mode. Can you see my presentation now?
No, it's not visible.
No, it is not coming, Sharma. It is only Geetika is there. Now Professor Lakhanpal.
Yes. Now we can see it.
How about now, sir?
Yeah, we can see it now. It's
Okay, sir. Thank
visible.
you, sir. Yes, sir. Thank you, sir. So this is work on diversity of ascomycetes fungi from Andaman Islands, which we have taken up, sponsored by SERB DST, Government of India. So, although more than 29,000 species are known from mainland India, hardly less than 1% is known from Andaman Islands. When Hosegaudar and Matthew, they published a list wherein they mentioned that 103 species are known from Andaman, including 52 genera of fungi from Jegomyces, Hymenomyces 25, Gastromyces 1, Ascomyces, etc. So, of these 103 species, 47 species were from soil fungi, one mangrove host, Soneratia acida, and the remaining 56 are terrestrial, from the terrestrial animals. Of course, in 2000, they missed another important work of Chinaraj from marine environment. 64 Mangelicolous fungi have been missed. If that is added, actually I could not see the. So is this okay for you?
No, this becomes too small.
Oh, okay.
Sir, press F5.
F5, huh?
Dr. Sarma, F5. No, sir. You can make a slideshow otherwise.
When I say a slideshow, other windows are opening and it's masking the.
Sir, no.
Content.
On top, it is after animation slideshow is written there, na? You can click that.
Yeah, I will try this F5. Okay, I will go for this one.
Yeah, now it's okay.
So, Butt and Kendrick have reported 25 species from India, including 4 from Andaman Islands. Together with these two reports, that is the Chinaraj work and Butt and Kendrick work, actually by 2000, year 2000, there were 171 species known from Andaman Islands. Then when we embarked on a project in 2015 to 2018, we just surveyed and prepared a checklist of fungi from Andamans. Then we found 446 by the year 2018, excluding our work, which belong to 96 families, 44 orders, and 216 genera. And these 446 mainly belong to meliolales and some lichenized fungi. That is the major contribution that has come, which has increased the number to 446. Meliolaceae, 47 members were recorded, and then actually this mode is masking my this thing. Okay. 3 main contributions are from Hosegaudar, 2012, Meliolaceae, this Chinaraj, and lichenized fungi from the Botanical Survey of India in Port Blair, like Sethi et al. And Jagdish Ram. They have reported a considerable number of fungi from there. And 37% of these 446 fungi belong to these two groups, meliolales and the lichens. The other fungi were sporadic. So recently, Rajamani et al. Under Professor T.S. Suryanarayanan, they have reported endophytic fungi. They have added another 20 with the species names from Andaman Islands, endophytic fungi. So it is against this background, I have taken up a project sponsored by the SERB, and we started looking at the wood degrading fungi. That means the twigs, the whatever woody litter fallen on the forest floor, those samples were collected by us. Around 2,120 samples were collected by us and examined from forests, reserve forests of North Andaman, Middle Andaman, and South Andaman. And we have surveyed through 7 field trips, and 67% of those samples showed spore-lating fungi. And totally 316 Ascomystus fungi belonging to 172 genera and 79 families, 30 orders, and 7 classes were reported during these 3 years of our work. Xylaria was the most specious with 19 species, and Sordariomacetes had 47% of the 316 fungi, that is 148, followed by Deuteriomycetes, 122 fungi. So we have reported 1 new family, Paralophiostromataceae, and 2 new genera, Murinitria and Paralophiostoma. 78 new species of Ascomystus fungi new to science were also reported. And of the 78 new species, we have already reported 26, as the list is given here, which I will also be showing through the slides. And Tuberculosis is the family which has shown a lot of novel fungi. This is the map of India, the Andaman Islands. So we went to the South Andaman, Middle Andaman, and the North Andaman. And these are the areas which we have covered, the forest areas. And the samples which we collected in the plastic bags. And these were the single spore pure cultures we have isolated through this method. And the germinating spores were taken into for further processing for DNA isolation. And these were the primers we have used, the nucleotide primers and the protein coding primers were selected by used by us. And we have used different softwares for our phylogenetic analysis. This is the snapshot of all the procedures: axonic culture, extraction of DNA, the PCR product, agarose, Sanger's, and NCBBF blast. From there, we retrieved sequences, and then further multiple sequence alignment were done, and phylogenetic analysis was carried out. And then we have deposited in the gene bank and the tree base and other resources. These are the depositories where we have deposited our information. And with this earlier, our checklist showed 446 fungi. With our 316 fungi, the number rose to 762 of 388 genera presently from Andaman Islands. We have deposited around 53 cultures in FCCI and MTCC, 68 herbarium specimens of the new ones. And these are the genera which are specious, means large number of species belonging to Xylaria, Aneloxyl, Hypoxyl, and Mazarine, etc. 22 host plants we could connect to the samples, and Calamus was supporting more number of fungi. These are the groups, the Deuteriomycetes and the Sordaromycetes represented large number of fungi. These are these 2 Sordaromycetes and the Deuteriomycetes. This is a list of new species. Some are already published, 28. Remaining are in the pipeline. We are communicating. They are proposed. This is the new family for Paralophiostromataceae, which has been described in the journal Mycosphere in this large paper. And it has hysterothecoid ascomata, but deep-seated ascomata are like this. And it has a unique feature of part spores, splitting into part spores, and it has appendages and colored and hyaline spores also. So when we compared, it is showing like this. It has a relationship with Lophiostromataceae, which has a short pedicel, and Paralophiostromataceae has a long pedicel, unlike Lophiostromataceae, which is having again a medium pedicel. And these, you can see the appendages and the color of the ascospores. That's what has made us to propose this new family, Paralophiostromataceae, which has been published in Mycosphere. And this is Taxterileopsis, another new species from the Taxterileopsis genus. It has oblique septa, so that is why we have given the name obliquus. This is the oblique septum it has, and we have also proved through the molecular analysis, which is cladding separately from the other species of the genus. So this is another new genus. We found Murinitria muriforme. Generally, Nectria species have single septum, and 3 species out of 1,000 species of Nectria also have muriform spores. When we analyze it through molecular analysis, it was clade separately, branching separately from the other species of Nectria. Then we proposed a new genus, and it has been published, Murinectria muriforme. In fact, we also transferred 2 other muriform forming species to this new genus, which were cladding with this new genus. So this is the new genus, where with other new species also been transferred. So this is another new species, Aloe cryptovalsa truncata. It has a truncate ascus. And then these are the other fungi from this. Diphrolomyces is a genus with distoseparate ascospores. And this also has been proved through the molecular phylogenetic analysis. This is a known fungus, Edenia gomepense, and it has varicose ascospores. And then we have Dichotomoptilus ramosimus, belongs to Chitomiaceae. And then we found Diaperthi eucalyptorum, which is a known fungus. And then this is a Trichoderma erinaceum, the ascomata and assay of the Trichoderma. Actually, the sexual state of Hypochria has been merged with Trichoderma after the one fungus, one name. Now it is called as Trichoderma erinaceum. Earlier it used to be Hypochria erinaceum. And this is Daldinia aeschylosae, a very common fungus in Andaman Islands. Huge fruit bodies. This is Nemenia bipalata belonging to Xylariales. These are known fungi. This is Brevicolum hylosporum belonging to Neo Hendersonia with hyaline ascospores. This is Cytospora fusispora. Once again, the Valsa species have been merged with the Cytospora. Since the Cytospora is oldest name, for one fungus, one species name, they have taken Cytospora. So all the Valsa group of fungi have been transferred to this Cytosporaceae. This is a new species. And this is Botryia bambusa, belonging to Botryospiraceae, Deuteriomycetes. And we also got Bruno Apispora pendiculata, belonging to Xylariales. These are published in Kavaka as 12 new species, including this Coeliopelia macrospora of Lophiostomataceae and Cryptoascoma Shodasa bj, because it has 16 ascospores. So we have given a Sanskrit name Shodasa bj. This genus was raised by Ananta Padmanabhan in 1989, Cryptoascoma. After that, no other species has been described. We have recently described Cryptoascoma Shodasa bj, published in Kavaka. And then Diatripella macroasca, it's a big, huge ascus with polysporous assay. And Leptospora sub-bivijana bija, which has 7 ascospores. This is Leptospora verruculosa and Montagnula vakra bija. It has somewhat distorted spore shape. So we got inspired by Professor C.V. Subramaniam and started giving these Sanskrit names. Anstrichnian bija koshe, because it has a sheath kosha. So it belongs to Hysteriaceae. And then Rigelia falcata, it has only one species earlier. Then we added another species, Trichospiraceae. And this is Rosalinia attenuata. Rosalinia, generally you have ellipsoid ascospores. This is an attenuated ascospore. Similarly, Rosalinia mostly eats spores. Here we have tetrasporate condition. So we raise it two new species published in Kavaka. And Physurama kavacha bj, again, it has a sheath, and hence the kavacha bj is the name given. This is published in Cream Journal, Current Research in Environmental and Applied Mycology. This is Physurama microsporum and Neoastrospirilla alankarta bija. These are new species which are published in Cream, belonging to Igalaceae. And this is Astrospirilla stellata, known fungus, Astrospirilla tornata. Astrospirilla uniseptata is the one new fungus we have published, along with the Pithomyces hylospore, which is Astrospirillaceae member. And we have this Patiala striatispora, because these spores have striations, longitudinal striations, you can see, and we have published this in Kavaka. And this is another fungus, Adenia verruculosa, which has already been shown with the molecular sequencing shown here. And this is Clypeosperia with the ascospores. This is Paralophiostoma. We got another new species in the same genus, and one more this lichen forming fungus. We have got a few lichen forming fungus or fungi also. This is Xylaria. We got some 6 species, but all are known ones. Xylaria officialis, Xylaria longipes, and Xylaria hamera. So 6 species we got, which have been also confirmed through this molecular sequence analysis. And then this is a fungus in the Sodaria Soderomycetes, Dictyospora. And then this is one more lichen forming fungus. We have got this Camelomyces tuberofia species. Camelomyces, there are a couple of species described from India, and we have got one more new species. This is belonging to Melanometaceae, Bisosperia jamaicana, a known fungus, and Bisosperia kitty, known fungus, and these are known but recorded. See, all these 316 fungi are new records for Andamans, not necessarily for India. So this is a Diaperthi fasciolarum, which has been approved at the molecular analysis also, along with this Utipa flavovirens. This is Rhytid Histrion rufulum, which has been proved at molecular analysis. And this is Trichoderma peltatum. Again, this is also confirmed through molecular work. And some more lichen forming fungi, Marcelatum. And of the 316 fungi, we found this Utipa flavovirens, which is having more than 10% occurrence, Baldinia 5 to 10%. Most of them are infrequent, and in fact, around 299 are recorded only once or twice. That shows the long-term investigations of these fungi from Andaman Islands. More than 214 are recorded only once also sometimes. So Calamus has been found to be the most rich host genus, supporting 21 fungal species. And all the 316 species are recorded are new. That shows the poorly investigated areas of Andaman Islands. And Hyde et al. Reported that 96% of fungi in northern Thailand are maybe novel, and he attributed them to these agarics, but also mentioned that microfungi are 55 to 96% are new. In 2 decades, they reported 500 new species. So from 3 years of our study, 78 new novel species have been recorded. So this is a good number. I think 25% are new for us. There are 2,200 species are known from plant species. Host species are known from Andaman Islands. If one can apply Hawksworth's formula of 6 unique fungi per plant, host plant on an average, there could be 15,000 fungi which one can expect from Andaman Islands. So there is a huge potential for fungi from Andaman Islands. This is a summary of the study. We published already 1 new family, 2 new genera, and 28 new species have already been published, and others are proposed. And these are the most species families, new species, Tuberophiaceae, Xylaria dominant genus. Most frequent fungus is Utipa, and Calamus andamanicus is incidentally a native of this Andaman Islands, which is a fungal rich host, because we got some 26 fungi from this host. So I profusely thank the Mycological Society of India for considering me for this Memorial Award lecture for this year. I thank the Department of Biotechnology, Pondicherry University, for providing the facilities. I thank Serb Government of India for funding our work. I also thank the Forest Department of Andaman and Nicobar Islands, and Middle and South and North Andaman Islands DF Office, District Forest Office. And I thank Niranjan, who has worked in this project as Junior Research Fellow, and it is most of this contribution comes from him. Thanks. And I thank all the senior mycologists. With your blessings, I could continue my studies in mycology and contribute a little bit. Thank you very much.
Thank you, Dr. Sharma, for a very good lecture and maintaining the time. Can you close your slide, please?
Yes, sir. Yes, sir. Yes, sir.
Yes, I request the chairperson as well as the co-chairperson to join me to award this Dr. S.K. Shom Memorial Award to Dr. V. Venkateshwara Sharma. Sharma, you will get this award by post. Your awards and the lecture certificate is already in your inbox, Gmail. Thank you very much.
Thank you, sir. Thank you,
Thank
sir.
you very much, Dr. Sharma. Thank you for keeping the time and
Yes, sir.
making us a bit comfortable with time.
Thank you. Thank you.
So I think we go to the next.
Yes, sir. The next memorial lecture will be Professor K.G. Mukherjee Memorial Award for Middle-Aged Scientists. This is a new memorial award for exclusively for middle-aged scientists. On behalf of MSI, I profusely thank Professor Rupam Kapoor and her team for creating this memorial award. And they contributed 1 lakh, and that has been deposited in the fixed deposit and for this memorial award. Professor K.G. Mukherjee, Krishna Gopal Mukherjee was a distinguished mycologist and microbial ecologist, a person of high repute for his research contribution all over the world. He was on 14th May 1934 at Lucknow as the 4th child of Mr. Devendranath Mukherjee and Mrs. Leela Mukherjee. He started his teaching career as a lecturer in botany at Lucknow University in 1955, from where he obtained his PhD degree in botany in 1962. He joined the Department of Botany, University of Delhi as a lecturer in 1964 and later shouldered the responsibility of the head of the department from 1985 to 1988. Professor Mukherjee has been keen interest in culture collection and received advanced training in taxonomy of fungi at the Commonwealth Mycological Institute, Surrey, England in 1968. Where he was also offered a permanent post of mycologist in 1968. He also worked at the Institute of Seed Pathology at Copenhagen, Denmark, Institute of Miroforseng, Handelshofen, Brummen, West Germany, and the Central Bureau for Smear Cultures from Netherlands. He visited various institutes, culture collections, and laboratories in Australia, Austria, Canada, Denmark, England, France, Germany, Hong Kong, Italy, Japan, Malaysia, Netherlands, New Zealand, Philippines, Sweden, Switzerland, Thailand, and USA as visiting professor scientist. Publications of Professor Mukherjee show significant contribution made by him in different areas of mycology, plant pathology, microbial ecology, and microbial biotechnology. He has authored and co-authored more than 600 research papers in these fields. He also co-authored, edited, and co-edited more than 35 books on various aspects of microbial ecology and microbial biotechnology. Professor Mukherjee had about 40 years of research experience on taxonomy and ecology of fungi from soil, rhizosphere, root, including the mycorrhiza, plant surface, dung, and seeds. During his research, he investigated and discovered several species and genera of fungi and mycorrhizal fungi from Indian soil. About 55 PhD students and more than 100 MPhil students were awarded degrees under his able supervision. Professor Mukherjee is survived by his wife, Abba Mukherjee. We thank her for the contribution also. who stood by him all through his career and has been virtually the backbone of for his academic and professional achievements. Their son, Mr. Amit, pursued the career of his own choice and did his PhD in plant molecular biology from University of Alberta, Canada, after completing his BSc, MSc, and MPhil in botany from University of Delhi. And this year, this award goes to young scientist, none other than that of the organizing secretary of this conference, Dr. Amanitpal Singh. I request the chairperson to introduce the Dr. Amanitpal Singh, please.
Thank you very much, Dr. Raman. You have made my job simpler. First, I would like to speak a few words about Professor Mukherjee because I happen to be his first student.
Yes, sir. I know that. I want it from your mouth.
And I don't know why Rupam has given this as middle-aged, because he was ever young.
Sir, we are
he never aged. Sir, and
actually,
so is Dr. Avneet. He is ever young, ever youthful, ever blissful. So, you have a right choice. You cannot find a better human being than Professor Mukherjee.
Yes, sir.
He was a friend, philosopher, and guide for all. Very caring, very sharing, honest in his dealings, and lovable person. Ever helpful. Anybody coming to him would not be disappointed. He was such a type of individual. And the same, you have the right choice for this award, first award, as Dr. Avneet Pal Singh, because he has all these characteristics which I mentioned. Age has no effect on him. He is youthful. He is blissful. He is ever helpful, hardworking, sincere, honest, and a young man with the deep integrity, honesty, and dedication. You must have seen in the organization, and along with Manruchi and all other people. So he deserves this award very much, and I congratulate him from the core of my heart, because the award happens to be named after my illustrious teacher, Professor K.G. Mukherjee. And now I will request him to deliver his lecture.
Dr. Amanitpal Singh, 20 minutes, please.
Sure, sir. I'll stick to that.
Please. And you are an asset to MSI.
That is an honor, sir. The credit goes to Dr. Atri.
You maintained 20 minutes, please.
The credit for this association, sir, that goes to Dr. Atri. When I joined the department in 2011, the first conference which I attended, that was MSI at Amravati. And that is how, even though the membership was earlier, but the sort of reintroduction that was made by none other than Professor Atri. So I am extremely thankful, and I am equally thankful to Honorable Professor T.N. Lakhanpal-ji, who is really a guiding force for us, and who has been inspiring us right from the day one when I was an MSc student, and that continued during the PhD and further on. Now, to begin with, I am extremely thankful to the Mycological Society of India for considering me as a candidate for the prestigious Professor K.G. Mukherjee Memorial Award Lecture. Although it is just the beginning, but still the magnanimity of the executive members, that is quite evident. And I'll try to justify, though it looks impossible, but I'll try my level best to be somewhere near for deserving this particular award. Now, the title which I have chosen is the Polypore Diversity in Northwest India. I am here because of this man. He is the person who introduced me to this field. And right back in 2003, when I joined when I thought over joining PhD, he was the choice, and he introduced me to this field. And still, he's inspiring me a lot to do whatever I can do in this field. So coming to the topic, polypores, as evident from their name, these are the macrofungi which belong to the phylum Basidiomycota. They are very, very unique in having macroscopic fructifications. These fructifications, they are quite variable as far as their morphology is concerned. But the most interesting feature is that on their hymeneal surface, they have very narrow openings. Those openings are called as the pores, and after that term, the polypore group has been proposed. And these fungi, they not only play an important role in the recycling of the organic material, but at the same time, some of these, they have been used in the classical medicinal system for the cure of many of the ailments of human beings, particularly in Asian countries. We have so many examples. These fungi, they have been kept in 17 families within 7 orders of the class Agaricomycetes within phylum Basidiomycota. And the traditional group that was proposed in order Aphelophrales, but after the intervention of molecular studies, now we have this particular arrangement where they have been spread in different families of Agaricomycetes. Mainly, they are involved in causing different types of rots, both white as well as brown rot of both the coniferous as well as the broad-leaved tree species. They are caused by these polypores. So this was the introductory portion. When I started my research career, initially the concentration was on the corticoid fungi, which grow in the form of sheets on the wooden substrate. But when I joined back this department, we wished to diversify the group, and on the basis of the inputs which were given by Professor Gurpal Singh Dhingra, we opted for exploration of these fungi primarily in the state of Punjab. After that, it was extended to other parts of the Northwest India. And of course, he's still guiding us, and he is with us right in the field. So because of him, we could contribute something to this group's diversity from Northwest India. So when we talk about Northwest India, these are the 4 primarily areas which we have surveyed: the state of Uttarakhand, the state of Himachal Pradesh, Punjab, and some portion of the present Jammu division of Union Territory of Jammu and Kashmir. And these fungi, as discussed even earlier during that introductory portion, they grow in association both with the coniferous as well as the broad-leaved tree species. So we have surveyed different types of the forests ranging from subtropical to the temperate forests. They have been collected thoroughly, and it is for the last almost 9 years, the survey work that is continuous, and it is still going on in different parts of Northwest India. So the advantage of studying the diversity is that you have the chance to visit new and new places. Along with your group of interest, you come across so many different other peculiarities of the vegetation, the forest type. You are witness to the very graceful landscapes. You enjoy the nature. And most importantly, in our group till now, the contribution has been from the girl students. So those girls, they were brave enough to be in the field along with the other team members for hunting these fungi. We have collected them in association with different types of substrates. This is how they basically grow. This picture depicts the nature of these fungi. This is how the polypores, they grow in association with the wooden substrate. And we have visited even Punjab. In Punjab, we could get some very interesting species which are new to science, because this was the area that was not explored by the earlier workers. We have this type of, so ranging from this type of scrub vegetation, dry deciduous vegetation, to the subalpine zones, we have surveyed all these areas for the collection of these fungi. So these fungi, they have been collected. There is no rocket science in that. With the help of simple hammer and chisel, they have been removed. This is the very first picture which I would like to share. This is dating back to the year 2003, when I joined my PhD and we were in the field for the very first fungal foray. So this is how the experience was there. And from that date till now, the hunting goes on. And these fungi, they prefer to grow over the shady areas. This is a species of genus Ganoderma, and this is how the logs shaded area that is being preferred. These are, you have to have some basic skills, getting their details preserved. Then what you need is a good microscope and to examine these. So on the basis of these very simple techniques, for the last 8 to 9 years, our team has been exploring different states of Northwest India. Until now, about 550 different polypore specimens, they have been examined. And these 550 polypore specimens, they have been deposited in the herbarium of Department of Botany, and they belong to 165 taxa. Out of these, as many as 16, they have been proposed as new to science. Some of them, they have been published. Few of them, they are in the pipeline. And more than 100, they have been documented as new additions to the mycoflora of India or the respective study areas. Now, if you look at the 17 families, I will here go through quickly some of the important or significant families of polypores where we have some economically important members also. The most important family that to mention is family Hymenocrhizaceae. Now, this is a family which you can encounter in the field very easily with their brown surface of hymenophore. And when you put a drop of KOH, the surface turns black. So this is a very simple test. We have different genera to our group. We have the members of Philoporia, Phalinus, Nomensularia, and Fomitiporia. They have been collected and described in this. Now, I will here restrict to some of the interesting findings. The very first on this right is a new species of genus Phalinus. This new genus of Phalinus has been named as Phalinus hemalecus. Now, we ignored this collection when we were in the field because of its dusty appearance, but the researcher, who is a girl student, Dr. Napreet, she was interested that we should take this collection and work it out in detail. And after her intervention, we made the collection, and but when it was described, the macro and microscopic features, they were entirely different. They were different. They did not match with any of the earlier described species. In Phalinus, we have this grayish brown or brownish hymeneal surface, but here the appearance was different. The microscopic characters, there was presence of very thin-walled ellipsoid basidiospores, and in combination with the presence of these long CT is the characteristic feature of some of the members of family Hymenocrhizaceae. Now, it was compared with Phalinus senex, but the size of the spores, the morphology of the basidiocarp, and the context of this pileus that was entirely different. And it could not be placed in Phylinus senex. Hence, this was proposed as an independent species, Phylinus senex. Sorry, Phylinus himalaycus. Then after this, just a moment. Apna Ashu?
Ashu?
Sorry for the interruption. This is another species on the left, which again belongs to Phylinus, genus Phylinus, because of the presence of diametric hyphal system. We have typical skeletal hyphae and the generative hyphae, and with some sort of rooting base of these CT, and very unique, these obtuse margins, which are brightly yellowish to golden brown in color. And these macro and microscopic features, they could not be matched with any of the species, hence a new species that was published. With this, within genus Phylinus, we have good variation even in terms of morphology of the basidiocarp. Here you can see a typical pileate basidiocarp, but in case of genus Phylinus, we do have some members which form sheet-like basidiocarp. And one of them is Phylinus morai, which was proposed by Dye. And this has got somewhat undulating, somewhat irregular sinuous, the outline of these CT, along with the presence of thick-walled basidiospores without oily contents. And this is the first report of Phylinus morai from India. This is very, very interesting specimen, Phylinus papillatus. We were on our way to Manali, and near Nerchok, we stopped for a cup of tea. And when we stopped for a cup of tea, there was a dried populus tree, which was growing near that tea shop. And on that, there was a specimen which was growing on. And this specimen this was comparatively softer. In genus Phylinus, we have comparatively woody, hard specimens. To our surprise, it was comparatively softer, and it was having comparatively wider generative hyphae and thick-walled ellipsoidal basidiospores. The CT, they were small-sized, and some of them, they were having the rooting base also. So this unique morphology, even the hymenial surface, that was violet brown in color. So these macro and microscopic features, they formed a base for the proposal of a new species. While conducting our fungal forays in different parts of Punjab as well as Himachal, we came across a unique collection of the genus Phylinus. This collection of genus Phylinus was always collected in association with Melia azidirec. For the first time, we collected it from Pathankot, that was near the border of Punjab and Himachal Pradesh. And it was growing high up on the branches. We removed it, we described it, and it could not be assigned to any of the known species on the basis of unique macro and microscopic features, hence a new species. Interestingly, from that area, it extended to almost all the localities towards Dalhousie. We made these collections regularly, and that extended even up to some portions of Chamba also. And that was named as Phylinus meliensis as a new species. Then we came across another characteristic collection, that is Phylinus pecularius. We were in our collection tour in the area of Palampur. And in that Palampur, we made these bright golden colored collections. And we brought it to the guest house of IHBT. We were examining its morphological features. Interestingly, one of the caretakers of the guest house, he visited us and he said, "From where did you get it?" And we said them, "It is so and so locality from where we got this collection." He said, "You are very lucky." He named it as Kathi, and he said that people who are lucky, it is a sort of blessing of the God. And interestingly, we got this specimen in the compound of a temple. So this was correlated. We were very much interested in examining its details. The CT, they were large sized. There were some sort of cystidioles and this unique cracked surface that formed very unique set of macro and micro morphological features which were unique to this species, and we named it Phalinus peculiaris. We were also interested in knowing this, why this was called as the luckier one or the blessing one. The girl who was working on this family, she was also exploring the antioxidative potential of family Hymenocrataceae. So she took it for the antioxidant activities, and interestingly, we got the maximum radical squelching radical squelching activity in this collection. So that is what the diversity can lead to. Now, in this family, we have this genus Philoporia. Normally, family Hymenocrataceae that forms huge or large sized fructifications. But you have to be very careful. We got, you can see this is the centimeter scale. This is probably half a centimeter. So about 1 centimeter in breadth, we got these very small poroid collections, very interesting. And it belongs to the genus Philoporia. The species is Bibulosa, and this is a new addition to Indian hymenocrotide mycoflora. We also got some interesting specimens. Here I'll take up only one, that is family Schizoporaceae, genus Oxyporus. Now genus Oxyporus, it forms bright, this very, very bright white fructifications with the resupinate or sheet-like fructifications. And it has got these globose spores, these cystidioles with encrustation on its tip. And this has been placed as Oxyporus obducens, and it is a new addition to the Indian mycoflora. The largest order within the Agaricomycetes that houses maximum number of polypores, that is order Polyparales. We have some interesting families, Ganodermataceae, Fomitopsidaceae, Meruliaceae, and Phanerocrataceae, whose members, they have both economic as well as the ecological importance. The first one to go is the family Fomitopsidaceae. We came across the genus Entrodia. There are 2 species of Entrodia. Entrodias, they are unique because they form resupinate fructifications. And their basidiospores are ellipsoid and they have very prominent oil contents. The 2 species, Palvinescens and Lucina, they differ in their cystidioles. We also tried to study, since these are causing brown rot, we were interested in studying the invasion of the hyphae, and we could cut and trace the course path of these hyphae, which were intracellular in case of this Entrodia palvinescens. One of the significant genus of the polypores in Northwest India, that is Ganoderma. And we have about nearly 38 different species of Ganoderma available with us. And these Ganodermas, they have a diversity, they have a spectacular display of their fructifications. Now, they are basically divided into 2 groups, the shining laccate ones and the non-shining non-laccate ones. On the basis of fructification appearance, one can distinguish between these 2 species. And to confirm, you have these truncate spores with very prominent markings on the intine. So these 2 species, Australe and Amboense, they have been collected. They are new to the state of Punjab. We also tried to see the SEM images, and we got some good results just to see the surface of these basidiospores and how the ornamentation that is there This is in case of curtici. This is in case of...
Can I interrupt you?
Yes, sir.
Oh, yes. So that kindly conclude so that the next
Okay. Yes, sir. Yes, sir. I'll conclude in 2 minutes.
Okay.
These are the other Ganoderma species. We have all these available with us. We have Seriporias, the typical resupinate poroids. We have Entrodelia, this one new species of Entrodiella. One new species, Entrodiella indica, that has already been published on the basis of these macro and microscopic characters. A new species of Bondarzevia indica that is also there. Now, since we have entered into the molecular systematics, so we got this collection of Gloeophyllum longisporum from Jammu. One of our research scholars, Brijbala, she's working on that. She got it. The morphological features were very unique, and further it was confirmed to be a different species within this and has been named as Gloeophyllum longisporum. Besides this, we have also performed these histopathological studies, and we could trace how the hyphae they traverse in case of brown and white rot species. Now, this has been possible because of the contribution and help extended by these people. Nils is there, Dr. Riverden is there, Dr. Langer is there, and we acknowledge the financial grant which is extended by DST under SERB, this Young Scientist Scheme. And the onus that lies on these people, this is the team. These are the 7 people who have contributed towards this. So I am extremely thankful to all these people for making me capable of standing and delivering this lecture. Thank you very much.
Thank you, Dr. Avanit Pal, for an excellent award lecture. And I request the chairperson and co-chairperson to join me to hand over the award virtually to the awardee. So the award, Professor K.G. Mukherjee Memorial Award for Middle-Aged Scientist, and the award goes to Dr. Avanit Pal Singh, the first award in this memorial award, and this will be posted to Dr. Avanit Pal Singh. The soft copy is already in your inbox email. Thank you very much, Dr. Avanit Pal.
Thank you very much, sir. Thank you.
Yes.
Thank you very much, Dr. Avneet. And thanks to Dr. Gurupal Singh Dhingra, who trained you, and to Professor Atri, who groomed you so well. And thanks to Rupam, who thought of diverting this money to Mycological Society of India and making it a prestigious award on the name of our revered teacher, Professor K.G. Mukherjee.
Thank you, sir.
Thank you and congratulations, Dr. Avneet. Thank you.
Thank you very much, sir.
Oh, yes. Okay. The next award and lecture and the last of the series is Professor P.C. Jain Memorial Award Lecture. At the outset, on behalf of MSI, I thank Professor Congo and his team for organizing 1 lakh's contribution to Mycological Society of India that has been deposited as a fixed deposit. And for this Professor P.C. Jain Memorial Award Lecture, the Professor P.C. Jain Memorial Award, the Professor P.C. Jain, then head Department of Applied Microbiology and Biotechnology, Dr. at Arising Gaur Vishwavidyalaya at Sagar, was a worldwide known mycologist and microbiologist and fellow of Mycological Society of India. Professor P.C. Jain was born on 1st February 1949 in a village called Tikaria at Madhya Pradesh. During his schooling, he was a meritorious student and received several scholarships that helped him to continue his education despite of low economic conditions of his family. He obtained dual graduation degree, BSc and B.Ed from Regional College of Education, Bhopal University, Bhopal, with first class. He did his master's in botany from Sagar University, Sagar, in 1974. Thereafter, he was awarded PhD in botany under the supervision of Professor S.T. Agrawala from Sagar University, Sagar. His main contribution was on keratolytic fungi. In 1978, he joined as lecturer in the Department of Botany, Sagar University, Sagar. Therefore, thereafter, he was promoted as a reader in 1991 in the same department. In the year 1999, he was further promoted as professor in the Department of Applied Microbiology and Biotechnology at Dr. H.S. Gaur Viswavidyalaya, Sagar. Head Department of Applied Microbiology and Biotechnology and contributed immensely in the development department. Under his leadership, the department was graded as one of the best departments in the university, both in teaching and research. He has guided 18 PhD students along with several short-term and long-term projects. He successfully completed several projects, research projects funded by UGC, DBT, and MP CAST, MP Biodiversity Board. He was recipient of Commonwealth Academic Staff Fellowship and pursued postdoctoral work on the development of enzymatic tests for early detection of contaminating fungi in cereal grains at Rothamsted Experimental Station, England, and visited various universities and Commonwealth Mycological Institute, England, during visit, and also his valuable contribution to the scientific world is reflected in his research publication. More than 140 research papers made in national and international journals of high repute, and proceedings of national seminars and conferences, and edited books which I have received citation worldwide. Environmental microbiology, storage, fungi, enzyme biotechnology were his major area of research. He has contributed several scientific reviews in the field of plant antimicrobials, storage, fungi, industrial enzyme, published in the books by national and international publications. By Grant Academy Bhopal. The most recent edited book is the Microbiology and Biotechnology for Sustainable Development, contributed a chapter in e-book Applied Microbiology, published by NISCAR for National Science Digital Library, New Delhi. He was an active member of several scientific and professional bodies. He believed that knowledge and experience possessed by individuals should be unconditionally disseminated to young brains for betterment of society. His keen interest in mycology led to seminal contributions in the field of thermophilic, terpenophilic, and storage fungi. And this year, Professor Karwar, R.N. Karwar from Banaras University has been selected for this award, P.C. Jain Memorial Award. I request the chairperson to introduce Professor K.N. R.N. Karwar, please.
Thank you, Professor Raman. It is indeed a pleasure to introduce Dr. Karwar, but Dr. Karwar doesn't need an introduction.
Yes, yes,
He's
sir.
so well known to all. He is a person who is always smiling. He is an illustrious student of an illustrious teacher from Gorakhpur University, Professor Kamal, and he is a pioneer in the endophytes. He has worked so well, he has been trained outside, and I will just sum up that he possesses 3 P's, that is purity, patience, and perseverance, and that is what you require from a humane scientist, and so he is. So I request Dr. Karwar to deliver his lecture.
So thank you, sir. I hope I am audible to everyone.
Karwar, 20 minutes, please. Kindly
So you
cooperate.
are 10 minutes from 30 to 20.
Yes, sir.
Because of the time constraint.
Yeah.
So, sir.
And we are hopeful you can do it.
He will do, sir. Yeah.
Is slide visible?
Yes, yes, it is visible, sir.
I mean, this fungal endophytes slide or something else? Which slide is visible?
Fungal endophyte slide. It's
Okay.
visible, sir.
Okay.
Yes.
Okay. Actually, there was something else also. So first of all, good evening to everyone. Professor Chairperson of this session, Professor T.N. Lakhanpal Saab, Professor Manohrachari, co-chairperson, my friend Professor Yaspal Sharma, Professor Raman, Professor Atri, and Avneet, MS Reddy, all the MSI office bearer, and my known, and every MSI member. Professor Lakhanpal and Professor Manohrachari, they both are friends of my Guruji, Professor Kamal. So they are like my Guru who are here to, and before them speaking is quite difficult. However, I will try my level best to justify this lecture. I pay my sincere tribute to Professor P.C. Jain, in whom memory this lecture has been instituted, and I am thankful to MSI who chose me to deliver this first Professor P.C. Jain Memorial Award lecture. So the topic of my talk today is fungal endophytes, a hidden treasure trove of structural and functional diversity.
Sir, you can, excuse me, Kharwar sir, you can make it full screen.
Okay.
Yeah.
Full screen.
Yes, it's on the right side, sir. Right side, last.
Okay.
Dr. Kharwar, you can directly press F5.
Yes, sir.
F5 at the top, sir.
F5. Okay.
Yes, sir. It's
Yeah,
perfectly all right.
Okay. Okay.
Yes.
So this is we all know. So I'm going very fast because Professor Raman has cut the time, so fungal endophytes are the organism which reside inside the healthy plant tissue without causing any detectable symptoms, and it establishes with the host endophytism, not the pathogenicity, which is unbalanced association, but they develop a balanced antagonism. And these endophytes are so well organized. They are good enough loyal to their host also, who live with healthy plants. They protect plants from the different stresses, and when the plant die, so you know they switch their work from endophytism to saprophytism. So when the plants are healthy, then they take nutrients and shelter. But as the plant starts senescence or die, then they change their mode of nutrition from endophytism or parasitism to saprophytism, and they live till the cremation of the plant. So you can see the loyalty of these endophytes, for whom plant provide nutrients and benefits. So they are not like son and daughters of these modern time. They live with their parents till the cremation. So these endophytes have different variety of roles. So they produce enzyme phytostimulation. They can utilize antimicrobial activity, and endophytes could be used in tissue culture, production of volatile organic compounds, source of bioactive novel compounds. Endophytes with multiple roles, and this nutrients recycling, because they are the best scavenger, also fungi. And in bioremediation, they have the role in the bioremediation and biodegradation. This pigment production also, and reciprocal interaction between above and below ground communities. So, of course, there are many important work carried out. I would like to mention one or two here, that Curvularia lunata mediated thermotolerance generation in Dicanthelium lenuginosum. So this work was done in 2001 and it was published in 2002 by Redman and group in Science. And she proved that in the geothermal area where the temperature is so high and plant denied to grow, so Dicanthelium lenuginosum was infested with the Curvularia lunata, and after that Curvularia lunata infested plant could grow successfully, while Curvularia lunata minus plant couldn't grow. So it was later found that heat shock protein level was enhanced, and that this was the reason that a plant could grow successfully there. Enhancement on antioxidant activity was also done by the Omasini and group. And mutualism to parasitism or saprophytism, this was also done. Resistance against metal and mineral stress, it has been done by Malinowski, and plant growth promoting activity by the releasing of plant hormones, it has been done. And the endophytes, give the 51% new structures and compared to soil isolates, which has given 38% novel structure. So this was also one of the reason to looking for towards these endophytes. And this role of endophytes, as you can see here, that endophytes mediated this Artemisia annua growth, and this is the free control. So you can see the growth. So now you can imagine that plant endophytes mediated growth may be better. Here also we can see that in the non-symbionts, and here is the salt treatment plus symbionts. So you can see that how the symbionts as an endophytes, they may promote the growth of plant against salt stress. Here also against different 0 day, day 3, day 6 and day 10. So differences you can see here in the salt treated, and here is the salt treated with the endophytes. And isolation normally can be done from the insulted conditions. So these type of isolates can work as an endophytes very efficiently. Here another is the salt, cold and drought stress and water uses also. So endophytes may work against this salt stress, against cold stress also. Here you can see the growth of the plants, and here they also in drought stress and the fluid uses, they minimize the fluid uses and they could be utilized in under this condition also. Here we have done some this evaluation of Congo red dye coloration and degradation potential by an endophytic fungus, Trichome, that has been isolated from the Tibetia plants, and this screening we did. And here you can see here that this is the control and this is the after treatment, and the different parameters have been used. So 2 day, 4 day, 6 days. So at least on 6 days, the decoloration was much more compared to others. Here is the other different parameters like concentration of the carbon carbon sources, nitrogen sources, different pH and temperature. So if we see, then the glucose was the best medium, and here is the in West here 20 milligram per liter concentration was degraded successfully. Here is the nitrogen source where the yeast extract has been showed better performance. Temperature was the 30, and you can see here that this is the control and this is the treated with the Congo red. So you can see the deformation in the cell wall and other constrictions has been developed after the treatment. So effect of Congo red, biomass has been decreased after treatment, certainly, but regenerative ability of Trichome has also been assessed, and it was found that after the treatment, successfully decoloration and degradation, fungus has also been revived and grown successfully. So it was concluded that there is no much change. This is the ethyl acetate and this is the RF value of this control Congo red. And after treatment, you can see that the patches that we see that RF value become 0.76. It shows that the compound has been degraded into the smaller compounds. And this is the FTIR spectra, which also shows you this is the Congo red, and this is after the treatment, which show that you can see here the much peaks, while peaks has been reduced, it means that has been degraded. Here also you can see that the HPLC of Congo red, and this is after the treatment, which show the aging different peaks, so that the Congo red has been divided or transformed into some other non-toxic compounds. And this is the treatment with the Congo red, and this is the seed germination with the beans has been performed. And in control, it was 100% germination, while in Congo red degraded products, it was 73%, while Congo red alone, the seed germination was reduced 53%. So it shows that the treatment given by the Trichome is successful, and it has enhanced the seed germination, and this is the plumule and radical length. So the overall conclusion was that the findings suggest that C. gliospiride possesses a good potential for the decolorization and degradation of Congo red, and it had less phytotoxic effect on Phaseolus mungo. And these are the plants which have been studied in my lab, including some more also, because this is one of the plant tea plants. So more than 5,000 isolate we have recovered from this plant, and Phomopsis 3 was Longicola 21%. With 21%, it secured the first position, followed by Phomopsis diporthi with 12%, and the Trichome gliospirides with 11% pollination frequency, and 19 other morphotypes showed abundance about 0.5%. Hitomium globosum and Gignardia species were found specific to only the leaf tissues, while and among 45 morphotypes, 43 belong to Ascomycotina, while 2 to Basidiomycotina. We also studied that location-wise recovery. So there were 4 locations we chose for this plants. Location 1, that was the BHU, log 2 was the Hathinala, log 3 was the Rani Bagh, Nainital, and the log 4 was Mirzapur, and this Rani Bagh, Uttarakhand, and this log 4 was the RGU, Rajiv Gandhi University, Rono Hill, Arunachal Pradesh. And if we categorize it with the tissue specificity, season specificity, so monsoon secured the first rank with the 40% recovery, followed by winter 35%, and least to summer with 26%. If we tissue-wise specificity was also assessed, and it was found that leaf harbored a maximum percentage of colonization with 30%, followed by stem 32 and leads to bark 28%. And of course, principal component analysis we also did, and we found that some species were specific to the bark in winter, and they were segregated in the stem and the leaf, they were segregated. And one positivity we found that 61% got imperfect match. It means it provides us an enormous opportunity to work hard to discover the new species. And of course, Professor Manorachari is here and Professor Lakhanpal. They all know that this time only ITS-based analysis is not being accepted by most of the journals. So we have to consider polyphagic approach, and we have to include morphology, and then the molecular analysis, including some protein genes also, like beta tubulin, HSP, and this translational elongation factors, etc. So this opportunity is with us. This paper was published in Scientific Reports in 2007, and these are some this colony characteristics of the fungi which have been isolated during this period. I'm going fast because of the time constraint. So these are some microscopic view which can be seen, and this is the Preusia, Phomopsis, and the others. So I'm not. So this is one species that we received from the neem plants, naming Chloridium, and this was tested for its antimicrobial activity, and it was found that it has gave very significant activity against some microbes, and then we isolated it and purified it, and luckily we got the crystal, and then crystal X-ray was done, and then RTEP diagram was made, and its molecular formula was this chemical formula was the C15H14O6, and its name was given javanicin, and it was published in Current Microbiology. And this molecule was very, very active against this Pseudomonas fluorescens and Pseudomonas aeruginosa at the rate of 2 microgram per ml. And these Pseudomonas fluorescens and Pseudomonas aeruginosa, they both are pathogenic to plants and human both. And it was also against, active against Cercospora arachidicula, and this Pythium ultimum, Verticillium daily, and some others, but that dose was very high, MIC was high. So, and this is the another biomolecule which was isolated in my lab, and the name was given terine, which has been isolated from the Aspergillus tereus, and the plant was the Echeiranthus aspera. And luckily, we also got a crystal for this compound also. So again, this was given, and C8, H10, and O3, and this paper was published in Frontiers in Microbiology in 2000, and it was a good candidate for anti-cancer activity, which can be seen, and at the rate of 125 ml and 175 ml, it was quite active. So 50% inhibitory concentration, IC50 value of pure compound and crude against 549 cell lines are 170 and 121.91. So for pure, it was higher compared to the crude. So it was concluded that though it has not been elucidated yet that which compound has worked synergistically, which has lowered down the MIC value of crude compound compared to the pure one. And this is the recently published paper in South African Journal of Botany, and we received milbemycin from fungus Penicillium citrinum, and so this is also this also showing antimicrobial activity very significantly against many microbes pathogenic, which are pathogenic to human and plants both. And if we
Professor Karwar, can I interrupt with you?
Yes.
Kindly conclude, please.
How much minutes are still?
Sorry, sir. Please conclude.
Okay. So
Yeah.
okay. So this is one of my review which has been published in Natural Product Reports with the impact factor of more than 12. So why people are focusing towards endophytes? Because if we see, then in from 1993 to 1997, only 6 anti-cancer compound has been reported from fungal endophytes, while in time period 1998 to 2010, it was 5 only, while the most productive year when the number of compound raised, that was the 2003 to 2007, the number became around 28, while in 2008 to 2011, this number went up to 56. So more than 100 compounds have been reported from only fungal endophytes entity. So there is chance to work hard to produce some good candidate which come in the market, because many of them have not come in the market, and Taxol is one of them which has been reported from the fungus and this Taxomyces endriani. Of course, there are so many slides because of the time. This is just I'm running the slides as Professor Raman has given a caution. So what the take-home message of this lecture is that the different dimensions of fungal endophytes. So we can use the endophytes as a plant growth promoting organism. We can use them against different abiotic stresses. We can use them to decolorize and degrade the toxic compounds into the non-toxic ones, and we can also use the epigenetic modulation. Just I wanted to show that how we can enhance the production of known and some cryptic compounds, because this epigenetic modulation do not alter the nucleotide sequence, but it increase or it modulate the gene expression through which newer products are, which is not normally produced under normal conditions, they are produced. And we have published some papers in this line. And this endophytic fungi can also be utilized to synthesize the nanoparticles. So different dimensions of endophytes, and you have seen here different prospects and challenges are also with this. So I'm thankful to all my students who did these works. This is the Gary Strobel, who is one of the pillar of this endophytic research, under whose mentorship I worked almost a year. And again, I'm thankful to MSI for choosing me to deliver this first Professor PC Chan Memorial Award lecture. And I'm thankful to the chair of this session, Professor T.N. Lakhanpal, and my co-chair, Dr. Professor Sharma, and Professor Chari, and all senior members. Thank you, everyone.
Thank you, Professor Karwar, for an excellent award lecture and for your kind cooperation. Can you close the slide, please?
Hello.
Your slide, kindly close your slide so that I can.
Stop. Yes.
Yes, sir. I request the chairperson and co-chairperson to join me in our virtually to Professor Karwar, and what will be sent to you by post.
Congratulations, Professor Karwar.
Thank you very much.
Already awarded in your email. In your Gmail. Sir, thank you very much. On behalf of Mycorrhiza Society,
Thank you, everyone.
I thank all the chairpersons, and now Professor Professor Yashpal Sharma as co-chairperson, and Professor Lakhanpal as chairperson, and also the moderator Dr. Geetika for their excellent cooperation. And with this, all the endowment lectures are over. Now we are moving to the next session, the session 4, the lead lectures.
Thank you, sir. At least,
Thank you, sir. Thank you so much.
Professor
We
Raman, sir,
thank
just a
both
second.
of us, all the three of us thank Professor Raman,
Yes.
the organizers, for giving us this opportunity.
Yes.
And all the presentations, all these endowment lectures were very nice, very explorative, and full of utility. And we thank them from the core of our heart. Thank you very much.
Okay. Thank you very much, sir. Thank you very much.
Thank you very much, sir. For the next session, for the next
Manish Kapoor.
Yes, sir. Sir, let me request Dr. Manish Kapoor to kindly take care of the next session, sir.
Yeah, sure.
Yes.
Just to brief the audience, just to share, Dr. Manish Kapoor.
Professor Avanit, Dr. Avanit.
Yes, sir.
I am leaving. Okay, thank you. Hearty congratulations to every one of you.
Thank you, sir.
Okay, thank you.
Sir. Okay, sir.
Thank you
you.
very much, Professor
Thank you, sir.
Mohanlal. Thank you.
Thank you.
Thank you very
Thank you.
much. Take care of your health.
Yes, sir. Thank you, sir.
care.
Thank you.
Thank you, sir. Very good.
Yes, sir.
So Dr. Manish Kapoor will be moderating for the session 4th, where we have the lead lectures, 2 of the lead lectures. Dr. Manish Kapoor, he is an associate professor in Department of Botany, Punjabi University, Patiala. And his area of expertise is horticulture and floriculture. And he is also working on the cytogenetics of medicinally important plants. He is a multifaceted personality, and he is also exploring the biochemical evaluation of the medicinally important plants. So I heartily welcome Dr. Manish Kapoor and hand over the stage to Dr. Manish Kapoor for further proceedings.
Good afternoon,
Dr.
Professor
Manish Kapoor.
Satyanarayan.
Thank you. Thank you, Dr. Abhi.
Narayana is in.
Welcome, Professor Satyanarayan.
Welcome, sir.
Oh, thank you so much.
I thank the organizers for giving me this opportunity to moderate this session for the session number 4. And I'm privileged that I have to introduce eminent scientist, Professor T. Satyanarayana ji as chairman, and Dr. V. Kumarasan as the co-chair of this session. Professor P. Satyanarayana is a professor in the Department of Microbiology in the University of Delhi South Campus. He has his PhD from Sagar in 1974, PDF from France. He has headed the Department of Microbiology, University of Delhi South Campus 3 times. He had been Deputy Dean Students in the University of Delhi. He has guided 30 PhD scholars, edited and authored 6 books, 150 research papers, and 22 edited books, book reviews, goes to his credit. He is fellow of National Academy of Agriculture Sciences, Association of Microbiologists of India, MSI, Biotechnology Research Society of India, and Andhra Pradesh Academy of Sciences. He is a recipient of Dr. G.B. Manjrekar Award of AMI in 2003, and the recipient of Dr. V.S. Agni Hotrudu Award by MSI in 2009, and Malviya Memorial Award by BRSI in 2012. I welcome you, sir.
Thank you so much.
And Namaskar, sir. Namaskar. And we have our co-chair, Dr. Kumarasena ji from Kanchi Munivar Center for Postgraduate Studies, Puducherry. His area of specialization is mycology and molecular biology. He is actively working on foliar fungal endophytes, on diversity of ectomycorrhizal fungi, diversity of agaric mycota, and diversity of white-spored agarics in Puducherry. He has more than 25 years of research experience and has many research papers of national and international journals. And he has completed 3 research projects. Welcome, sir.
Thank you, sir. Thank you. Thanks a lot. Thank you for
sharing this session.
Thank you.
Yes, sir. I welcome Professor T. Satyanarayana and Dr. B. Kumaresan for this session. Sir, Professor Satyanarayana.
Yeah.
Can you hear me? Can you hear me?
Yeah, I can hear you. I'm seeing you also.
Kindly restrict the speaker's time by 20 minutes.
Okay.
I request the speakers to cooperate, Professor Lakhanpal and Professor Surya Narayanan. And please, chairperson and co-chairperson and the speakers, please excuse me. I am going for my lunch. I will come and join in a few minutes.
Okay.
Thank you.
Thank you so much, Professor Aman.
Thank you. Thank you. Thank you.
For generous introduction. And since there is no time, we have to start straight with the presentation of Professor Lakhanpal, who is very well known for his contributions in the diversity of myxomycetes and mushrooms and endomycorrhizal fungi. He doesn't need any introduction. He has been often participating in MSA conferences and delivering talks. Without losing time, I request Professor Lakhanpal to deliver his talk on health benefits of mushrooms. Please. Not being heard, some problem. Please,
Sir,
Manish, not being heard. Yeah.
I think he has to unmute.
Unmute yourself, sir.
Now I can start.
Yes, sir. Yes, sir. You can start, sir.
Okay.
Please share your screen.
So, Honorable Professor Satyanarayanan, the co-chairperson, Professor Atri, Professor Manruchi Kaur, Dr. Avneet Pal, Professor Manorachari, and all other participants in this August meeting of Mycological Society of India. I first of all, let me thank the organizers for providing me this opportunity to share some of the work that I have carried out, and I dedicate this work to the Almighty, to my parents, and to my illustrious teachers, Professor K.S. Thind from Punjab University, with whom I did my master's, and Professor K.G. Mukherjee with whom I completed my PhD, and to all my students who have been part and parcel of the work that we have carried out for the last so many years. And in some cases, it is still continuing. Next, please. Next, please. So this is a broad topic which I have selected because in the recent times, in the last 2 decades, the mushrooms have become very good health potentiators. So a broad topic is health benefits of mushrooms. Next, please. Now, mushrooms, they have been categorized differently. They have been called mysterious because they just appear and then disappear. They are magnificent because they exhibit a large variety of colors, and then they occur in circles which are called fairy rings. And mythically, it is considered that they represent the path of dancing fairies. They grow on a wide variety of habitats. They grow on soil, they grow on dung, they grow on wood, they grow on litter. And they adopt a wide variety of modes of nutrition. They can occur as saprophytes. They also are parasites. But the most important part is that they occur as symbionts and help in many ways the plants, especially most of the mushrooms, they form ectomycorrhiza, and that is so beneficial to the plants with which they associate. Then they have a large variety of different types. They are called, they are edible species. They are species which are not edible. They are species which are poisonous. They are species which cause daydreaming or psychotropic mushrooms or hallucinogens. There are species which emit light or which are illuminated during night. So they are luciferous. They are called luciferous mushrooms. Next, please. They have a very long history, and there are different myths because they appear and disappear quickly. And that is why they have earned the name mushrooming, because whenever they come in large numbers. So that is what is meant by mushrooming. So Greeks believed them that they provide strength. Some considered them to be the food of gods. Some considered Chinese, especially the Oriental people, they regarded them as elixir of life because the use of mushrooms in the Orient has a very long history. And it is in that culture that they have found uses or medicinal uses or elicitors of mood or potentiators, health potentiators. In modern times, the mushrooms, they were initially consumed only as a food adjunct or a food supplement. So for palatability, and most popularly, they were consumed by people who are vegetarian. But in the recent times, especially after 1995, their nutritional and nutraceutical potential that has been evaluated, and they are found to be of great medicinal or nutraceutical use, and various terms along with other herbals, they have been coined for them. So modern science realizes that it is the diet which controls and modulates the functions of the human body. And it is said in our scriptures that as is the food, so is the mind. As is the mind, so is the thought. As are the thoughts, so is the conduct. And as is our conduct, so is our health. And health, WHO defines health when a person is healthy, when he is physically, mentally, intellectually, psychologically, and spiritually healthy, then a person is considered to be healthy. And good food supports good health or homeostasis. Next, please. At this homeostasis, this reduces the risk of chronic diseases, because the inbuilt system or the immunity of a human being that gives checks and controls. So they are dietary supplements, they are called functional foods, and they affect the health positively. So there are various names which have been given to them. And now mushrooms are regarded as functional foods or dietary supplements of a very good quality. Next, please. Next, please. This is journal, just to complete the presentation, that what are mushrooms, where do they occur, what is that we see them, or when we examine them or when we identify them, what are the characteristics that we have to take care of, and how these all characters are important criteria or taxonomic criteria for their delimitation. Next, please. So there are around 1,44,000 of fungi which have been named. Around 16,000 species of mushrooms have been reported. Around 10% of them, they are prime edible species. More than 100, around 130 species, they have been cultured and cultivated in one or the other way in different countries. But there are only a few species which are commercially cultivated. And there are exhaustive publications for identification or taxonomic delimitation or characterization of different species. Next, please. In India, we have around 2,500 species, and out of them, around 300 are reported to be edible. There are about 15 species for which cultivation technology has been developed or is being utilized, but there are only around 6 or 7 species which are commercially cultivated. Next, please. And these are the cultivated ones: the button mushroom, the oyster mushroom, the shiitake, Calocybe milky mushroom, and paddy straw mushroom. Next, please. There has been a long phase, and Britishers have been the pioneers in explorative studies, and this can be divided into 3 phases. A phase which started around 1825 and ended by 1950s. Next, please. Then there was a phase, the second phase, next, please, which we can say is from 1850 to 1969, and actually the good phase for explorative studies or extensive explorative studies, it started around 1970s. Next, please. And there are so many workers which have contributed, and Patiala University is one of the major contributors for the taxonomic work on all groups of fungi, especially the higher fungi. And you have a stalwart, Dr. N.S. Satri, his guide, Professor S.S. Saini, and they have a lineage which goes to Punjab University, Chandigarh, and we had a father figure in the form of Professor K.S. Thin, who groomed and trained many people in the area of fungal taxonomy, and they are taking, now this is, Avneet Pal is the 3rd generation and he is training the 4th generation. So we have no dearth of good workers, but they are at specific places and specialized in different groups of higher fungi. Next, please. So large number of papers have been published after 1970s. Again, I think the maximum contribution has been from Punjab University, Patiala. Some publications have come from Punjab University also. And the momentum to this was from the visit of Dr. Wartling from Edinburgh. And this is a good publication that came out from on larger fungi of Kashmir. And then Dr. Sathe, unfortunately, we lost him early. And then Manjula, who was Professor Natarajan's student, and Dr. Raman's colleague, she examined all the collections which were deposited there and then provided us a base from where we have taken up. And then Natarajan and Raman, they had excellent monograph of South Indian agaricales. And then we also have published a few monographs on Amanitaceae. Another monograph, I think it is in the press, is by Dr. Manruchi Kaur on Amanitaceae, and it is a very exhaustive work. Similar exhaustive work, she happens to be Professor Atri's student, and I think many more monographs will come from this university on all the larger fungi of at least North India. Next, please. So there is a monograph on Bolitaceae of India, then Rusulaceae of Kumayu Hills. Then there is a checklist by Natarajan. It was in 2002. And recently, the Directorate of Mushroom Research, in which we also, Professor Atri and myself, also associated, that is documentary of Agaricomycetes mushrooms of India, which was published in 2017. Next, please. So next, this is Amanitaceae of India. Next, please. Next, quickly we can go through Bolitaceae. Next, please. Then Rusulaceae by Kannar Das and J.R. Sharma. Next, please. And this was in 2010, Biology of Indian Morels. Next, please. And this is documentary of Agaricomycetes mushrooms of India. Next, please. So the most important, as I said, what are their health benefits? And if you see this picture, which I quite often project, and this is from a book, you can see this Pied Piper from Orient or Chinese origin, and Ganoderma, which Dr. Avneet mentioned, talked about. This is Ganoderma lucidum, and this is called there the elixir of life or panacea of all diseases. And this is one of the most popular mushrooms which has attracted attention as a medicinal mushroom, most exhaustively worked out mushroom, and most potent in most of the diseases. Next, please. So mushrooms, why they are important medicinally? Because they have all the components which make them medicinally important. They have all these which are listed here, all these components, and they all have been reported to have some as health potentiators. Next, please. So they produce antifungal, antibacterial, antiviral compounds. They are insecticidal, nematocidal. So they act as immunomodulators of both humoral and cellular immune factors. Next, please. So next, please. So there are a number of mushrooms we have heard in the lectures that they have antimicrobial or antioxidant properties and how they are useful in one of the award lectures. So there are a number of mushrooms which have been investigated, and they have been found to have very good antioxidant potential. So they are, because of these properties, they are good food supplements or food supplements with the good health benefits. Next, please. And almost all the mushrooms, there are many in small or large measures. The quantity may vary, but all the mushrooms, they are found to be having some or the other compound which is nutraceutically important. So they are good for diabetic people. They are good for people having hypertension. They have been proved to be anti-cancerous, anti-HIV, and they boost the immune system. And I think in this COVID times, the consumption of mushrooms, if it is taken up, if it is administered and seen, because there are experiments which have been conducted, especially with regard to their vitamin D components, with regard to selenium, and some other compounds which have been found to be very useful components for health benefits. Next, please. So the next, please. So anti-tumor properties, as I said, they are anti-cancerous, and many mushrooms they have been worked out and they have been found to possess anti-cancerous properties. Next, please. Next, please. So Ganoderma species, they are very important, and I think it is a vast field which Dr. Avneet Paul should undertake, because we were dependent only on one Ganoderma species. They have identified more than 40 species, molecularly characterized them, and maybe some of them possess better constituents than Ganoderma or Lingxi, what is called in Chinese. And you can see that they have all these properties, which are most important is that they are all immunomodulator agents, antioxidant properties, anti-microbial properties, anti-amyloid properties. So they are good in almost all the diseases, in combating almost all the diseases, or controlling, if not eliminating them, but they provide resistance to the human beings. So Ganoderma species is one. Next, please. There are 13 or 14 species which have been thoroughly worked out. Ganoderma is one. Shiitake, Lentinula edors, or Japanese mushroom, is the other one. Next, please. Cordyceps, this is Kida Ghas, and all of you know its history. This has been projected in many conferences, and a lot of, now this is a fungus which was sold for 50 rupees a kilogram, but now it is around 10 lakh rupees a kilogram, and how it has a long history of medicinal properties. And this is truly a immunomodulatory fungus, and that is what Chinese athletes have been consuming, and the people thought that they were consuming. So dope test was performed, steroids, but it was found that it is this fungus which is giving them strength, both moral strength and physical strength, and it was protecting them from different types of diseases, and it was acting as an immunomodulator and providing them extra energy, and they were not undergoing fatigue. So during gymnastics. Next, please. So Agaricus blazei, this also has a very long history, how it was discovered, and it was found to be better than many of the mushrooms which were prevalent at that time. So this also is very effective, and all these diseases which are listed here, they are from literature. It has been also very thoroughly worked out, and the history is long. We will narrate it some other time. Next, please.
Sir, can I interrupt you, sir?
Yes, sir.
Okay, sir. Thank you, sir.
No, you want that I should conclude?
Yes, sir. Another 3 to 4 minutes if you
Yes,
conclude
yes,
it.
yes, yes. Okay, okay. No, I will not take more than 3 minutes. So next,
Okay.
please. Next, please. Next, please. So lenticidiodes, I have already talked about. Next, please. Gryphola frondosa also is a very versatile fungus, and it has a very good antimicrobial, anti-cancerous activity. And these are the different compounds which have been isolated from this. Next, please. Then Coriolus versicolor. I don't know whether the name is correct. Avneet can correct if it is incorrect. Tremella. Next, please. Auricularia. Schizophyllum, a fungus on which most of the fungal genetics or cytogenetical studies are based and which occurs everywhere, is also a very versatile fungus. Flammulina. So almost all mushrooms, agaricus, volvariella, auricularia, you name a mushroom which is edible, and which all these mushrooms, they possess all these characteristics. Next, please. Even Morell's species, they have also, we have worked out that they have good nutraceutical potential. Next, please. So no, this we can, next please, we can skip this. This also we can skip. This also we can skip. This just gives the statistics from the books. Next, please, or the monographs. How much is the total market? Next, please. The different species. Next, please. You can quickly just go through them. There are different species which have been reported from Himachal Pradesh. These are the common slides which you will find with all of us. So they have been reported from different parts. Most of these are the ones which are edible and which have been traditionally consumed by people in different parts of Himachal Pradesh and elsewhere. And some of these are cultivated, some of these are not cultivable, and but still they have all those properties. Some of these are mycorrhizal. So we have a lot of opportunities and challenges in mushrooms. There is, because species number is so large, and in each region there are species which are ethnically important. So that is why ethnomycological studies on them are most important, which the Directorate of Mushroom Research with its 36 coordinated centers throughout the country, this is one of their prime mandates, to collect germplasm and explore them. And I think each region should have mushroom species which are edible, so that they can aid or help people in maintaining good health. Next, please. So next, please, next. I would just like to show you Shimla. Next, please. Shimla Institute of Advanced Studies, Mall Road, Ridge. Next, please. These are the habitats where we get a lot of mushrooms. And I thank you all for this patient hearing. If I have taken more time, Dr. Raman will excuse me.
Thank you very much for
Thank you very
cooperate.
much, Sir. For a nice presentation. So next speaker in the session is Dr. T.S. Suryanarayanan, who is very well known for his work on endophytes. He's going to speak on association of endophyte plants and their applications. Professor, yeah, please.
Suryanarayanan sir, 20 minutes, please.
Yeah, I'll try to finish in 20 minutes.
20 minutes, sir.
Yeah, can you hear me?
Yeah.
Yes, sir.
Please.
Okay. So let me see. I need to share my screen. Yeah. Yes.
फ्री हो गया अब मेरा ये
find how to do this. So I'm trying to share, but I'm not able to get it. Let me see. Can someone tell me how I do this?
Sir?
Yeah.
Sir, at the bottom of your screen, there is an option of share screen highlighted in green, sir.
Yes.
You just click that.
Okay.
Then there will be an option of share your desktop.
That is the thing that I'm not seeing. There's one this.
First, you open your.
Sorry, you minimize your screen first.
Okay, I'll do that. Yeah. Okay, first I should open the thing that I want.
Open and minimize. And,
okay.
And then share screen. Then.
Okay, I'll do that. Let me see. Now I should be able to do that. Let's see. Yeah, so
Sorry, have you got that share screen?
No,
Hello?
let me.
No, what you do, you first minimize your half screen you make. From the entire full screen you minimize. Then you will see the lower side, there is a share screen button. Press that.
Perhaps I should log in again, okay? I'll try that.
Okay.
Okay. Now I think, can you see the slides now?
Yes, sir. It's perfectly all right. Just put it.
Yes, you can see it.
Yes, sir.
Yeah, and you can hear me?
Yes.
Okay.
Slide mode. Show mode. Slide mode.
Sir, please press F5 to zoom it.
Yeah, I'm trying to do that. It takes a little time here. Okay. So, thank you all. And I thank the chairperson as well as all the office bearers and members for giving me this opportunity. So, this talk is largely based on our following paper, Fungal Endophyte Mediated Crop Improvement, The Way Ahead, which was published in Frontiers in Plant Science recently. Okay, so I don't want to go into the definition of endophytes, we all know that, but it is well known that these endophytes, fungi especially, are able to contribute to the general well-being of their host plant in their growth, biomass accumulation, tolerance to abiotic and biotic stress, etc. So these endophytes, the last 2 decades, are being suggested as candidates for improving crop production. So it is suggested that because they have this peculiar trait, so it is possible to get an endophyte with a very desirable trait and then transfer it to a crop plant so that the crop plant grows better, and you don't have to use pesticides or insecticides because the endophytes could confer tolerance to all these things. Okay, so there are many papers which describe this, these aspects of endophytes, and they are very successful in the lab or in the greenhouse, but when it is tried out in the field, it is not that successful. You don't, so far we have never got a very major breakthrough in using an endophyte as a crop protecting agent or something. So if you look at the number of patents in US alone for the year 2000 to 2018, a number of patent claims are there for different aspects of endophyte, different traits of endophytes, which could improve the crop production. As I told you, unfortunately, we don't see this happening in the field. So why is this? Why is it that the effect is seen in small labs or in greenhouses, but it is not being transferred to the real situation, namely the field? Now, one of the limitations could be due to the fact that we all study endophyte, either bacterial or fungal endophyte, as an individual. So, see, for example, you take the interaction of endophytes in the root system of the plant. So even in the root system, you can see that there are studies would either concentrate on bacterial endophytes, then they would not study the fungi, or schools which study fungal endophytes do not concentrate on bacteria. But actually, in the real situation, there is a mixed microbial community of bacteria and then fungi and perhaps protozoans, and there could be a mixture of all these things, and there is a lot of interaction that is taking place, which we are missing in the study. If you, we did a web search science, this was in the year 2016, and you just put endophyte and bacteria, two terms, and search, you will get, we got about nearly 5,000 number of papers. And if you look at, if you put endophyte and fungi, more, about 5,000. Oh, this is around 5,000. This is around 9,000, 8,500. But if you put endophyte and fungi and bacteria, the number of papers are very limited. And if you put endophyte bacteria and endophytic fungi together, you see that the papers that describe these interactions is very, very low. So again, with reference to number of citations, the same picture arises. So it is very clear that people tackling both endophytic bacteria and fungi, there are not many. And perhaps if we look at these things together, we could get some answers with the help of which we can use the endophytes better in the field. So even among, let us say that fungal endophytes, even among fungal endophytes, there are varied shades of endophytism. Latent pathogens, mutualists, common cells, temporary residents, or even latent saprotrophs could reside as endophytes. So all the endophyte is a very loose term of definition, which says that those fungi, endophytic fungi, are those which live inside plant tissue without producing any apparent symptoms. Okay, but it depends on at what time we are screening the plant. The same plant may have certain fungi now as endophytes and may not have certain, or may have new endophytes at certain other period. So because these fungi which show different lifestyles exist as endophytes, you can expect strategies and counter strategies evolved by both the plant and the endophytic fungi for their continued presence, and this could lead to a lot of interactions. Then genes expressed during phases of endophyte. See, when an endophyte penetrates a plant, I'm talking about fungal endophyte. So let us say it penetrates a plant just like a pathogen to enter the plant. So at that time, there is a pre-penetration stage, then there is a tissue penetration stage, and there is a colonization stage, just like in pathogens. And among fungal pathogens, they have identified that different reactions take place, different genes are shut, different genes are prompted during these stages of infection. What about among endophytes? Is this the way? Why is it that an endophyte enters like a pathogen but is unable to cause a disease? Another example is maize tissue is rich in antifungal compounds called benzoxenoids. But when you say antifungal compounds, they inhibit a lot of fungi, but still maize tissue has fungal endophytes. That means there are fungal endophytes which can tolerate these compounds. And secondary metabolite spectrum of different plants differ. And therefore, the defense metabolites can exert a sort of a selection pressure in the evolution of endophytes among them. So that is a question that has not been answered. So I may get a very good endophyte which shows excellent anti-insect property. I can isolate it from some weed plant or whatever. And then if I put it in a crop plant to use it for protection in the field, the reaction could be different, and maybe we will not get the same results. So then we do not know how plants respond, how endophytes respond to infection with reference to post-infectionally formed defense chemicals like phytoalexins. So these are many unanswered questions. And then the influence of high density of endophyte infection in leaves on photosynthesis is not known. So I have to go a little fast. And in any leaf, for example, leaf endophytes, if you look at leaf endophytes, you take a leaf and then study the endophyte present in a single leaf, invariably you see that it is densely infected by endophytes, but all species are not equally represented. You always see one or two species which are dominant. They are present in very large number in that leaf, and there are many fungi which are present in very small numbers. So every leaf is densely infected. The isolation is very high, but species diversity will be low. So, for example, you always get Trichome phomopsis, Pestalozziopsis, Philosticta, or Xylaria as the most common species in almost any plant. Now, the question is, why among the millions of fungal species, why only these 4 or 5 genera are dominating the endophyte assemblage of almost all plants? See here, we did a study, we published this in Current Science, about 200 plus plants from different parts of, all belonging to 60 different families of angiosperm, growing in Andaman and Arunachal Pradesh, Kerala, Tamil Nadu, and we collected leaf and then we used the same sampling technique and all that. You find Trichome, Philosticta, phomopsis, and Xylaria are dominant in all these plants. So what is this, why this generality? What is characteristic of these species? Not only that, whenever we do work with the endophyte of a plant, we always look at the endophyte in a particular sampling. We just do it once in there or maybe 2 times, but we don't see what happens to the endophyte population, what happens to the endophyte assemblage in the plant as time goes by. So here is our paper on fungal diversity. This plant is this plumeria, and you know that it sheds the leaf during the month of March, April, and then new leaves are formed, and then they grow up to next March, April. So we chose a single tree and then looked at the leaves and the endophytes they have during all these months. And you see in the same tree, you will see that during certain months, you have some fungi which are dominating in the leaf, and then suddenly there is a fall in that group, and you see the other group dominate. So with the time scale also, the endophyte community could change. So these are things which you should consider when we try to use endophytes for protecting crop plants. Similarly, as I told you, endophytes could survive, latent pathogens could survive as endophytes. So we still do not know whether the latent pathogen is genetically different from the endophytic fungus, is different from the pathogenic strain, or is it the same pathogenic strain that survives as endophytes in plants under certain conditions? These are things that have not been known. See, for example, we showed that there is a Leptospirillina crassyasca, which causes a very serious disease of groundnut or peanut in the USA. in all the peanut growing areas in USA. It is the black spot of peanut. But here in Tamil Nadu, several varieties, the leaves have this endophyte, Leptospirillina crassiasca, which causes this leaf spot in USA. But here it survives as symptomless endophyte. It is not a major disease at all in groundnut. But every leaf has this fungus. Around 60 to 80% of the endophyte in the leaf of groundnut in varieties of groundnut in Tamil Nadu has this fungus, but not as a pathogen. Why is it behaving as a pathogen in US and why not in Tamil Nadu? Same fungus. So we all know the work of Major, where they say that when an endophyte comes in, several defense-related genes in plants are highly upregulated. So how does the plant respond? Is the same way? Will the crop plant respond in a similar way when you put the endophyte in? So here, in another paper from our lab in 2018, we showed that an endophytic Trichoderma species, which was collected from a non-crop plant, when you spray it on a crop plant, it is very good in controlling this most common and devastating agriculture pest insect called Helicoverpa. So you can see that when the plant is sprayed with that spores of this Trichoderma, the mortality rate of the feeding insects increase, so it looked like it is a very good biological control agent. Unfortunately, when you spray this spores of this Trichoderma in say Abelmoschus aspirantus, that is that lady's finger, you find that after 28 days, the concentration or the isolation frequency of the endophyte goes down in the leaf. So you need to spray it again. The same thing happens with reference to brinjal. The same thing happens with reference to chili. So we have found a very interesting endophyte from a totally different, a non-crop plant, and it is effective against insect pest of a crop. But if you spray it, it is not sustained in that new crop host. So
Dr. Suryanarayan, please close in the next 2-3 minutes.
Sorry?
Could you please close your talk in the next 2-3 minutes?
Yeah.
Please. Yeah.
Okay.
Sorry for interruption.
Yeah, thank you. The reason for this could be that we are forgetting the interaction that can happen in a situation inside. For example, in the leaf, you may get, there are what you could see as dominant endophyte species, there are minor endophyte species, then there are keystone species, there could be keystone endophytes, then there could be bacterial endophyte, and there could also be fungal endophyte having endohyphal bacteria. These could interact in a sort of a network, and all these could be interacting with the host plant and then also with the environment. So with all this, it's difficult to just to see one endophyte at a time in the lab, and then looking at its properties, we cannot say that it can be easily transferred to the crop plant to protect it. It is like observing just the butterfly in a place and say, oh, this is butterfly. But if you don't look at the other stages of the life cycle of the butterfly and look at only the imago, this is what would happen. So it is strongly suggested that we study the interactions existing, the net of interactions existing, and the strength of these interactions existing in a plant and between its holobiome, all the endophytes that are there, and also the environment, before we can use them for use them gainfully for controlling or using improving the crop varieties. Thank you very much. With all this, I thank the MSI for giving me this opportunity, my present and past students for their sincere work, DBT for funding various projects of ours, and my collaborators, and all those who gave me a patient hearing. Thank you.
Thank you very much, Dr. Suryanarayan, for a very nice presentation. Sorry for interruption, and lack of time.
Sir, can you come out of your slide, please? Professor Satyanarayana, sir, can you hear me?
I can hear you. I wish to thank Kumaresan for giving us this opportunity to share the session to you and also
Thank you, sir. Thanks a lot to you, sir.
and organizers. Thank you so much.
On behalf of MSI, I thank Professor Satyanarayana and Dr. V. Kumaresan for efficient conduction of this session, and the moderator also, Dr. Manish Kapoor. Now,
Sir, please, sir, please. No, on the behalf of organizing committee, I am extremely thankful to the chairperson, Dr. Satyanarayana, to the co-chair, Dr. V. Kumaresan.
Thank you, sir. Thank
for
you.
sparing their valuable time. Professor Satyanarayana and Dr. Kumaresan, they have been excellent in managing this session. And more importantly, we are extremely thankful to the two eminent personalities. Listening to both of them is always a treat, is always a chance to cherish. So we are extremely thankful to Professor T.N. Lakhanpal and Dr. T.S. Suryanarayanan, who could spare their valuable time to be with us during this evening session. It is an absolute delight, sir. The entire organizing committee is extremely thankful to the resource persons as well as to the chairs for this for making to this particular session. Thank you very much, sir.
Thank you. Thank you for the opportunity, sir. Thanks. Thanks.
and all the sessions have been over. Today's session, and on behalf of MSI, we thank all the chairpersons, all the speakers, and all the organizers for the very good cooperation or conduction of these first day sessions. And the executive committee will be at 5 o'clock, and the general body meeting will be around 5:45. Already the links were shared to every member of the society and executive members also. Thank you very much. Thank you very much.
Raman.
Thank you.
Sir.
Dr. Raman.
Yes, sir. Please.
Yeah, and especially to Dr. Avneet.
Yes.
Thank you for using the spares.
Yeah, the organizing people, Professor Mundru Chikar and Avneet Paul and their team, they have excellently, this is a breakthrough for MSI as well as for their department, and conducting the virtual conference. It is efficient one, and we enjoyed it. The MSI enjoyed, and certainly the MSI members also enjoyed this session. And it is interesting that some of the scientists in the abroad also, they joined these sessions. And I thank all of them also for the joining the conference. Thank you very much. Thank you.
Sir, you are indispensables, not the. It's really you all are indispensable for the cause of mycology in India, sir. And we are extremely thankful for your precious and valuable time, sir.
You have really conducted the whole day's deliberations very efficiently. I congratulate you and your team.
Support, all your support and cooperation.
Ma'am, the motivation comes from all the seniors like you. We have been observing you all throughout these years, the entire executive of MSI, the senior mycologist, observing for the span of 10, 12 years how the things are being conducted. So trying to learn from all this, and it is possible only because of the cooperation and guidance. And especially to Dr. Atri, the way he has been there for the last 10 years, that is, I fall short of words in explaining that. Thank you very much. So we will be.
We are not leaving a tree. I will tell you.
That will be great, sir. That will be great. So we will end this session of this conference right here. For executive meeting and for general body meeting, separate meeting IDs, they have been shared. So we will be switching on that ID soon. So thank you very much, one and all, to all the key speakers. Big congratulations to all the award winners. A very, very warm thanks from all of us. Thank you very much.
Thank you. Thank you. And we will assemble 5 o'clock executive committee.
Yes.